I invite Dr. Milind Bharadia, Chief Organizing Secretary, MAHAPEDICON 2022, to give his welcome address.
Good morning, everyone. I just declare we will start the proceedings of the MAHAPEDICON 2022 sharp at 9 o'clock. Thank you.
A very good morning to all of you. A warm welcome to all the delegates to this pious city of Nasik. We hope that the lush green mountains and the picturesque scenery on your way from the airport to this city must have mesmerized you thoroughly.
However, most of us sitting in this audience may have spent decades in this profession as pediatricians. The science of pediatrics is a literal mother as also our breadwinner. Many of us should admit that with the passage of time, the writing on the wall has blurred. And although we're still strong with the basics of the subject, finer things have become hazy. Lectures, seminars, conferences are means available to us to wipe off this haze and replenish the letter and spirit of pediatrics. MAHAPEDICON, the Maharashtra State Conference of Pediatrics, under the aegis of Indian Academy of Pediatrics, is being brought before you in this heavenly city of Nasik. We have complete 3 days packed with academic material from best in the business. The 3 days program will be interspersed with other activities which will keep you amused as well.
On behalf of Nashik branch of Indian Academy of Pediatrics, I, Dr. Shalaka Bagul,
and I, Dr. Prachi Kiran Birari,
welcome you all to this 3-day extravaganza.
For the very first session of this delightful morning, I request Dr. Anil Kasliwal and Dr. Abhay Tilak, both from Nasik, to chair the session. I also invite the speaker of this session, Dr. Jagdish Chinnappa sir, from Bangalore, on the dais as speaker. Thousands and thousands of research material must have been published since decades about the work done by people involved in childcare. Writing a paper is not like writing an essay or film song. It takes years to lifetime of research, which is then put into words in the form of a paper. Not only writing, but also reading a paper is a skill. And this I say from my own experience of reading a paper. Dr. Jagdish Chinnappa has done a huge volume of work in pediatric research itself. And today he is here with us to give us synopsis of the last 10 best papers in pediatrics. Over to you, sir.
Thanks. Changer.
Am I audible? Clear? Yeah. Good morning. At the outset, I must start off this talk with 2 basic words. One is gratitude and the other is congratulations. I am extremely grateful not only to the organizers of this MAHAPEDICON, but also to the state from which I passed my MD and which has nurtured me with so many teachers, so many colleagues, and so many students now. So I am extremely grateful to the state and for the branch of Nasik for inviting me here today. The second is I must congratulate all pediatricians. And I congratulate them for many reasons. The first reason is basically amongst all specialties, it is the pediatrician who makes a difference and the largest difference in a person's life. And you will know that it is how you manage children that makes a difference to their entire life. You are part of the family, and how this is what is the most important thing in that child's and family's life. So what I'm going to do today is take you through, can you put the timer on, if you don't mind? What I'm going to take you through today is a very interesting thing. As to 10 landmark papers, my good friend Dr. Anil Kasliwal wanted me to speak on this topic. But what I'm going to do is to look at what are the 10 things that have really changed, because there are numerous papers which have come in. So I'm going to synthesize all this and see how I have changed, and maybe some of the seniors here have changed in their thinking over the last so many years. So as you know that we are at the present moment, we need to look at our past, we need to look at the future. Unfortunately, we cannot predict the future to a large extent, but we can look at what's going to happen in the next few years at least. At least that much is possible. So let's start off with the first main thing. Nature India published this very interesting paper on what are the challenges before India in the healthcare sector, and especially in women and child healthcare sector. And they put down these points. Neonatal and infant mortality was number 1. Infectious disease was number 2. Antimicrobial resistance was number 3. Malnutrition, NCDs, poverty, poor hygiene and sanitation, environmental health, mental health, and the lack of data, research, and training in healthcare. So let's look at each one of these challenges and see what has happened as far as the publications are concerned. I will start with neonatology. I think we must agree, I think the seniors here will agree, that in the last 15 years, neonatology has been turned upside down. And why? Basically, because of this philosophy of less is more. That is, modern neonatology is going back to the simplicity of neonatology. It's not getting into the more complex things. Yes, you have high frequency ventilation and all those things, agreed. But for the vast majority of children, it is the simplicity and less is more. And what do I mean by this? Basically, we know that these are the things that happen. One is delayed cord clamping is now a norm. 2, non-invasive ventilation. We have moved away from invasive ventilation to non-invasive ventilation. No suction for meconium stained liquor. Lower levels of oxygen saturations are tolerated. Kangaroo care is the norm in most of the neonatal ICUs. Parental involvement for developmental outcomes and screening. We all know that screening is something that is a norm today. Screening for critical heart disease, screening for hearing loss, screening for inborn errors of metabolism, NBS, all this is part of our routine neonatology. And I must pay tribute to my teacher, Dr. Daga, Subhash Daga, who started this concept many, many years back. And when I worked with him as a lecturer, he was propagating the same principles which are coming back into work today. So I think we need to look at more gentle care of neonates. Together, I would add this story. This is a story of a young ophthalmologist, Dr. Anand Vinekar from Bangalore. And he is a person who said, okay, newborn care is done. Now let's look at disability prevention, how we can prevent disability. And he's a ophthalmologist who, along with a tech company and the internet, devised a system of ROP screening at every small town and village in Karnataka. And this has been lauded by most international agencies. You can see this is the recommendations, and that is Light of the Eyes program, which he has implemented in Karnataka. So it is now spreading right across India. It is to screen even at the most basic level. You don't need an ophthalmologist. All you need is a technician with that camera. You send the images on the internet, and there is an ophthalmologist to look at that and say whether there is a chance of ROP and referral. So it's a magnificent program, all because of the initiative and drive of a single young ophthalmologist. So I think this is the future of our country. Next, we'll come to vaccines and immunization. The less said about it, the better. I'm sure all of us have seen the enormous impact that vaccines and immunization have made on our children. And as you know, when we started off practice, this is a landmark paper, which by Dr. Andrew Pollard, who has been on many of our platforms. And he has spoken basically of the advances in the technology of producing vaccines. Initially, we had only 2 or 3 kinds of vaccines. We had the live attenuated vaccine, we had the killed vaccine, and the toxoid vaccine. That's all we had. But now, if you look at it, these are all the vaccines you can see on the board on that side of the slide, which are DNA vaccines, RNA vaccines, subunit vaccines. You have the outer membrane vesicle vaccines. You have so many virus-like particle vaccines. So technology is changing. And over time, we are going to see more and more vaccines for disorders that are killing our children. If you look today, I'm sure all of you will agree with me that RSV is a major problem in your ICUs today. Okay, and the reason for that, now in the next few years, we're going to get RSV vaccine. So it's going to change completely. So this is, again, a landmark paper. The other thing which has happened is we are starting to understand the fundamentals of immunology of vaccines. And why is this important? As you can see, we initially started with 3-dose HPV vaccine. Now we are down to 2. And over time, I'm sure we'll be down to 1 vaccine alone. And you just imagine if you can give a single vaccine to prevent cervical cancer, single dose of vaccine to prevent cervical cancer in our population, it's something unimaginable that we are going to eradicate one more of the main causes of death in women. So I think this is another thing which has changed completely. As you know that ultimately the rights of the child says that we should give them the best health. And this extrapolates to adequate immunization. And we must congratulate our healthcare system for providing a fantastic response on COVID. And also not only providing this fantastic response, but also documenting it, to following it up. We have integrated delivery of vaccines to IT so that we can trace any single individual anywhere and get the data on that person. Then all of us, I'm sure, are involved in public health. And I must say that every pediatrician sitting here would have been involved in the pulse polio program somewhere down the line. And this is something that has been achieved with the cooperation of every one of us. It's not one person. So there are numerous publications on how polio was eradicated. But I'm sure that this is the most important achievement of all of us in the public health sector. Next, we'll come to dengue vaccine. Now, dengue is a major problem in our country, and we have had a couple of vaccines that come out. But what is good on the horizon is this new vaccine called Odenga, which is going to be implemented in Indonesia shortly, but probably will come to India also. And it has shown in fairly large trials to be relatively safe. So I think this is exciting news in the vaccinology sector, that we have basic vaccines being manufactured more easily. And also we are going to get vaccines for disorders that are killing our children right now. Coming to the third field, genetics. I can't tell you how much this has made an impact over the last few years. We have the most exotic genetic investigations available. And thanks to our president-elect, Dr. Upendra, who's going to take over next year, we are going to have a genetics module for all private practitioners, all practitioners. Okay, so that we are updated in how genetics is going to impact us. This is a landmark paper, again, from the New England Journal of Medicine, which shows you about the molecular change genetics, which has changed practice. It all started many years back in the 70s. In the 90s, they started with the Human Genome Project. And this was one of the first projects which was finished far in advance of the scheduled date. So therefore, genetics is going to be the future of our practice. We are going to have a large number of disorders. This is what we used to do when we were students and when practitioners. We used to put on these pedigree charts. But now that same pedigree chart can decide whether the next baby is going to go there or there, a happy child or a child somewhere in the grave. So there are a lot of changes which has happened, and this is how the sequence goes. You have an index child, you have an exome sequencing, you identify a pathogenic mutation, you go to a Sanger sequencing, confirm carrier status, do an extended family screening. I was with Dr. Mamta Manglani recently, and she was very vehement on saying that every relative of a thalassaemic child should be screened. That's the only way you can prevent thalassemia, rather than trying to treat it at a later date. So pre-pregnancy evaluation, pregnancy evaluation, prenatal testing, family identification, and decision. So appropriate genetic counseling is a very, very important part of our practice, which we may need to know the rudiments of. We don't need to be experts, but we need to look. The other thing which has happened is appropriate therapies available. I will come to this a little later on. And also it's more and more available, because initially it was an exotic kind of availability, but now it's more and more available. And therefore, I hope our future charts will identify the parents much before. We look at early diagnosis, on treatment, treatment will be available, and we can give a happy child to the family. Not only this, we have now the omics revolution. This is another landmark paper which I think everybody should read. They looked at how you can identify death from COVID, from survival from COVID. So they looked at all parameters, whether there's any predictive parameter. The one thing which came out was a unique proteomics panel. The proteomics panel in those who subsequently died was very different from those who survived. So this is again another big thing which is going to come into our routine practice, the ability to look at proteomics and how to look at it. Now, again, some people may say, well, genetics is something that is, only for people who stay in Mumbai or Nasik or Bangalore or something like that. But this is a beautiful book, Earth is Flat. All this is changing completely. This is my good friend, Dr. Mahesh Kore at Nippani. Okay, he practices out of a small place. I had the good fortune of visiting and staying with him. It's just about 2 hours from Kolhapur. And he has reported the most exotic genetic disorders because he can do all those genetic tests sitting in Nippani. And today you can identify a genetic disorder. You don't need to go anywhere. You can do it from your house because there are so many laboratories which will come and pick it up and go. So I think we are on to a revolution as far as genetics is concerned. The fourth point is the rapid diagnosis of infectious disease. Now, this has changed our practice completely. I'll give you a small story. About 8-10 years back, I had a child who came to me with a peculiar rash. This was a 2-month-old baby with a peculiar rash, some, vesicobullous lesions on the leg, irritable. And I didn't know what it was. Luckily, I had access to a PCR laboratory and I sent the fluid for PCR. It turned out to be chikungunya. So the point is that we have a large number of disorders which are now infectious diseases which can be detected by the PCR technology. We have the BioFire, which is made a huge difference to our detection and management of disorders. A recent paper presented from, again, Bangalore was a study on the current respiratory viruses, which are causing a havoc in practice. Okay, and what they have shown is that you have at least 11 viruses circulating now. Not only influenza, parainfluenza, and RSVs, which we know, but we have the Boca virus, the human metapneumovirus, and others. And if you look at the unique, the clinical presentation of these, they are very different. For example, parainfluenza virus is primarily you have an upper respiratory croup kind of a problem, all the croupy coughs which you get. Metapneumoviruses tend to be a little prolonged. So there is a unique manifestation which we can identify more and more. So I think this has changed completely. It's a very simple method. You can get the results in a couple of hours, if not an hour. And now I think all hospitals are putting their own homegrown PCR systems in place. I know for sure at least a couple of hospitals in South India put it, the Child Trust Hospital has put it in place. And now they're looking at even diagnosing, rickettsial disease through a PCR. Tuberculosis, I don't need to mention. I think everybody is well versed. We have Dr. Vijay Avle here. So tuberculosis diagnosis, okay, has changed completely. I'm sure Dr. Abhay and Dr. Anil will agree with me that we used to sit around trying to do BCG testing and Mantoux testing. Today, nobody is going to believe tuberculosis unless you demonstrate not only the presence of MTB, but also the resistance patterns. So I think this has changed completely. Coming to radiology. Radiology, I can give you a talk for 2 hours on what has happened in the last few years. But what are the main things that is going to affect us as practitioners? I think the one thing which has changed completely is this, the point of care ultrasound. And the point of care ultrasound is going to help you in so many ways. It's going to help you in resuscitation, going to help you in diagnosis, going to help you in symptom or sign-based and therapeutic. Again, our good friend from Bharatiya Vidyapith, neonatologist, okay, So Dr. Pradeep Suryavanshi, I think he has given a lot of training programs on how to use point of care ultrasound in management of newborn babies. So I think this is now a given. This is like our physical sign now. Unfortunately, we have the PT&D Act, and therefore we are unable to use it in our practices. But over time, this also will change. The next thing I'm going to touch about in radiology is the artificial intelligence. There are a lot of things that are routine, and those routine activities can be overtaken by AI. Recently, we had a very nice talk about AI in pediatrics by a very accomplished person. And he was talking that AI is going to come into almost everything in your practice, whether it's clinical diagnosis, whether it is decision making, whether it is laboratory, whether it is radiology, AI is going to come in. Because a large part of what we are doing is routine. So all we need to do is those that require thinking is what we require the human being. The rest of it can be done by a machine. So I think this is another thing which is come to. Then coming to information technology. I think the last 10 to 15 years has changed us completely, especially in the last couple of years, as far as IT is concerned. The promise of digital health, then and now, is a landmark paper published very recently in 2022. And you can see here that what all it can do, starting from health information, knowledge, integration, personal health devices. Very interesting story. Just a couple of days back, there was a report that the Apple Watch worn on a child detected a malignancy. And the reason was there was an abnormal elevation in the heart rate. So they didn't know why this heart rate was elevated. They investigated, and this is a catecholamine producing tumor in the appendix. So just by the elevation of the heart rate, we could get this. Diagnostics, of course, imaging, so many features. I think that's going to be like the basic center of what we are going to do. Coming to what we do in practice, I think DIAP, I think all of you all must have fallen in love with this and got addicted to it. During COVID times, this was our lifeline, and I think all of us were on it. Electronic medical records, I think all of us are used to now. Teleconsultations is going to get more and more refined, so that you can probably just pick up the phone and do your consultation and your prescription. We all turn to Google and update up to date for our updation of knowledge. Then you have mobile solutions, WhatsApp related consultations and opinions are the rule of the day. If you have a difficult case, put it on WhatsApp and you'll get 10 opinions in the next 1 hour. You'll have no problem at all in your diagnosis or management. So I think this has changed completely. The digital health has changed our lives completely. Of course, a lot of caveats. We have to look at how personal this data is. We have to look at the safety of this data, effectiveness, whether it is equitable, efficient, accessible. A lot of things are there. These are all the fine print part of IT, which we need to look at. Then I'm coming to the next important thing is immunology. Now, immunology again has changed us completely because there are so many things that have happened both in diagnostic immunology and therapeutic immunology. Like, for example, in diagnostic immunology, we are now recognizing more and more inborn errors of immune function, or what are called as primary immunodeficiency disorders. We have the T cell defects, the B cell defects, the combined defects, the phagocytic defects, and the complement defects. And there are many other defects which also we can pick up now because of very easy diagnostic modalities like flow cytometry and others. Another challenging thing which has come up right now is the treatment part of immune disorders or the immunologically based treatments. Today, apart from IVIG, you have monoclonal antibodies. I'm sure all of us are familiar with the rabies monoclonal antibody, which has been introduced in the last few years. Again, made a very big difference in our management of rabies. We have vaccines. We have so many other possibilities. And today, cancer treatment is primarily immunological. Previously, it used to be chemo and radio, but now it's more and more people are tending towards immunology because of the relative less side effects of immunology. Another part is the allergy part of it. All of us used to say, don't introduce cow's milk protein and peanut protein till late in the age because you might induce peanut allergy. This landmark paper, the LEAP study, showed that those who had introduced peanuts early in life had less chance of getting serious peanut allergy later on in life. So now we are probably going to move more and more towards early introduction of allergens. I think this is another landmark study which is going to see a lot of applications in many other allergens over time. Then we'll come to advocacy. Another story now. We need to be advocates for children, and we are all advocates for children. And what we have seen in the last few years is we are getting more and more advocate for adolescents, because we are trying to understand their problems and we are trying to put their issues in place. And we have to transition them. We have to put them into the adult community. So this is again, people are thinking of how to transition care. I'm just going to talk about a small story about this gentleman here. He is Prasanna Shirole. He's from Hubli, the border of Karnataka and Maharashtra. He had a child who had a rare disease. And he came to us. This child was managed at our place, went through a lot of problems, and he set up what is called as the Rare Disease Foundation of India. Now, this Rare Disease Foundation of India has done some outstanding work. This was set up in 2007. What it has done, I'll just put you in place, is this. It has lobbied and advocated for what is called as a national policy for rare disease. And because of this, they have stratified rare disease into group 1, group 2, and group 3. And a large amount of money is available from the government to manage these children. There are centers of excellence, as you can see there. In Bangalore, there's what is a place called as the Indira Gandhi Institute of Child Health. It's a government sector hospital where there is a special ward only for rare disease. And you can see all these children with LSD, Pompe's, and others being given virtually free treatment. So this is another great thing that has happened because we started looking at rare disease as a possibility. Therapeutics. Huge amount of changes in therapeutics have taken place. And you can see infectious disease. We know newer molecules, newer ways of looking at it. Rheumatology, I'm not going to go into it. You see, Dr. Raju Kupchandani would definitely tell you that there are so many exotic treatments for exotic disorders. Oncology, I mentioned very briefly. Rare disease, I've already spoken about. Now, transplant. Now, last few years have seen a tremendous increase in transplants, not only in the traditional liver transplants, but bone marrow transplants. And why has this happened? Because of advances in immunology, because we know how to handle rejections better. We know how to look at it, and the advocacy for more donors. And you can see here, in appendicitis, I'm going to talk about what is new here in appendicitis in therapeutics. Previously, what was the treatment for appendicitis? All of us in medical college said the only thing for appendicitis is refer to a pediatric surgeon and get the appendix out. This is what looked at. Now, what happened here was, this was a study in Ireland they looked at, and they showed that because of COVID, these children couldn't reach hospital. So they said, okay, let's look at this and see whether it makes a difference, whether medical treatment or surgical treatment is better. And they showed that there was virtually no difference between non-operative and operative treatment for appendicitis. So in the routine care, this is another thing which might come in, that the standard of care may move away, especially in those who are uncomplicated, to medical treatment rather than surgical treatment. This is something totally different. So something totally outside our norms of thinking. But this is what is coming now. Let's talk about another disease called cystic fibrosis. This is a chart in 2013. And I have had the good fortune of treating a few of these patients. And one of my landmark patients is a patient whom I have followed for the last 30 years. And I have gone through every single problem with this child. And I can tell you, we have done all this. Initially, the challenge was diagnosis. Then the challenge was managing with her with all these things which you can see here. And we were posting her for a lung transplant. Then what happened? The triple therapy for cystic fibrosis came. Again, one of the most fantastic things which has ever happened for a cystic fibrosis patient is this ivacaftor, tezacaftor combination for Delta 508. Because the previous one, ivacaftor alone, was for a different mutation, which is not common in India. This has made such a difference in the child's life. She's been taken off the list for lung transplant, and she's on treatment. Her FEV1 has gone up from 21 to 40. She's doing exceedingly well. She's a practicing lawyer. She's at 30 years, got married recently. So what I'm trying to say is it has impacted the lives of people phenomenally. This is the transplant program. As you can see, that was one of the first transplant, liver transplant, which was done in 1998 and 1998. And this young man is now going to be a pediatrician. Okay. So I think things will really gladden your heart. That is 2000 bone marrow transplants at one of the centers. That is Dr. Mamta, who I respect a lot, okay, who has done phenomenal work in bone marrow transplants. So I think these are people. Then, of course, one cannot miss this pure clinical acumen, pure clinical study. Okay, so now it's become mainstream. And just a few years back, he was awarded the Padma Shri. Of course, things are going to change. It's not going to remain the same. You're going to get pharmacogenomics coming in, which will fit what you want to do, which will see which medication will work for you. And there are so many things in this. I'm not going into the details of this. Lastly, I'll just touch upon three main things: research, ethics, and policy. We now know there is a large amount of push towards moving our very dismal record in the hunger index. We have been somewhere at 94 or so in the World Hunger Index. But now, of course, the government has taken a lot of action on this. But as practitioners, we also have looked at this. We have looked at vitamin D supplementation, which was unknown many years back. We are now routinely supplementing all our children. We are looking at iron supplementation, especially for adolescents and adolescent girls. We are looking at B12 supplementation. Routine deworming is done now. So a lot of things are happening from our side also. In research, what has happened is accelerated research. Previously, in the top part, you can see that it used to take 10 years for a normal vaccine, but it has been accelerated to 18 months. And why? Because we are looking at a different way of providing ethical clearance. Of course, we have to be careful because now we have got newer challenges in, let's say, CRISPR technology for gene editing, and you'll have problems there. So in conclusion, this long journey has been possible due to the efforts of numerous researchers in numerous fields throughout the centuries who have contributed to their work, creativity, success, and failures to advance our knowledge. The great combined effort of all these people, including my teachers, my colleagues, and others, has paved the way for achievements that are currently being realized. So we started with a slide where there were so many problems, but I'm sure with the resilience of the pediatricians and resilience of the medical fraternity, we have a lot of things that have changed practice and a lot of things that are going to change practice in the next few years. Thank you very much.
Thank you, sir, for this wonderful session.
Hello. 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 Jagdish,
Yes, sir.
one of your cases mentioned about appendicitis in COVID-19.
Yep.
And if you remember, during the COVID epidemic, we had plenty of COVID patients having abdominal pain and wrongly operated for appendix.
Yeah.
What is your opinion on that?
Yeah, you're right. There were a lot of children who came, or rather it was not children, it was more of adolescent adults who came with abdominal pain. But this study had very rigid criteria, okay, between looking at the surgical and the medical part of it, and to look at basically the diagnostic criteria for appendicitis. So therefore, these were truly appendicitis patients and not COVID masquerading as appendicitis. But very good question.
Any other questions, please?
Thank you, everyone. I request Dr. Anil Kasliwal and Dr. Abhay Tirak to felicitate Chinnappa, sir. I request Dr. DC Ladda, sir, to felicitate our chairpersons, Dr. Kasliwal, sir, and Tirak, sir.
May I request our next speaker, Dr. Upendra Kinswadekar, to please step up the dais. And to chair the session, may I request Dr. Anjali Gokarne from Thane and Dr. P.N. Devi from Nasik to please come up on stage. Isn't it ironic that a few hundred bacteria can cause so much trouble, right from a minor infection to major issues ending up in death? And on the other hand, there are not only thousands, but billions of microbes entering into our intestines, pitching their tents and staying put. Yet we do not call them as invaders or encroachers. We call them as friendly microbiota. So much so that the sachet-friendly enemies have become almost every prescription extending from head to toe. They, like our good friends, not only stay with us throughout our life, but also keep us safe from the pathogenic entrance into the gut. To give us more details about the gut microbiota, may I invite Dr. Upendra Kinswadekar. Over to you, sir.
A very good morning, friends. At the outset, let me congratulate Dr. Jagdish Chinappa for a wonderful session before this and all the organizing team of MAHAPEDICON under the leadership of Dr. R.D. Patil, Dr. Milin, Dr. Sharmila Kulkarni, the scientific chairperson, for drafting a great program and seeing you all in great numbers. After 11 years, I think MAHAPEDICON is happening in Nashik. So we are running short of time actually because I'm really happy that we started bang at 9 o'clock. So my responsibility also is to stick to time. That is very important. So when Sharmila told me to talk about something like gut microbiota, food for thought. So I was wondering whether there are two different things that she was meaning, or is it interrelated? I said thought is thinking. So thinking happens in brain. So brain's main food is sugar. All of us know it's the only thing that brain accepts. Then what is it? Anything, something else that brain also gets? So I was wondering, and then gut microbiota. So whether it is interrelated. So that is how the thought process started going down. And then this man, the great man, imagine 2,400 years ago, this Hippocrates tells us all the disease begin in gut. How does he know that? That's such a bold statement to make. And after 2,400 years, I think as Jagdish was talking about, we are now also thinking that probably this is true. Then he said our food should be our medicine and our medicine should be our food. That means anything which is wrong happening, majority of the times, it is related to some faulty diet. So he's correlated almost everything to that. Not only that, he goes one step ahead and he said, probably for every ailment that we are suffering, food has definite role. Salutes to this man. We just talk about his oath, but what great contribution without any evidence-based trials, research, nothing 2,400 years ago. So what if it turns out to be that we actually have two brains? आपण गमतीत कधी म्हणतो की तुझा काय मेंदू गुडघ्यात आहे का? But now we know that actually there is second brain that is called as enteric nervous system. All my MBBS, MD days, I was not aware of this terminology. Always thought of CNS, central nervous system controls everything. And now people are talking about, no, that's not, that's only in the brain, but you have something in the gut too. So what is this? So they said that gut is not only involved in absorption, digestion, assimilation of food, but something more than that. So your physical and mental well-being that all of you are right now having, it's possibly because of your enteric nervous system. It perceives environmental threat and influences our response. The fight or flight response that we talk about is not determined by your brain, but by your gut. That's why we call a gut-wrenching experience. Potat gola ala, bhitine. Before examination, somebody goes for number 2 frequently. Why does it all happen? It's the enteric nervous system. The butterflies in the stomach before the first date. What do we mean by that? All points out to that. Not just capable of autonomy, but actually influences brain. So it is very independent. It is not a subordinate of brain. 90% of signals of vagus, we always thought they come from up to down. It's the other way. It goes from down up. Big part of emotions that I said, and this is not just fancy, fascinating statements. It's been said in American Journal of Physiology. So this is all evidence-based. So 21st century, suddenly everything, it was all known, but nothing was happening here. And then came this lab. That is something called as a germ-free lab, where there is, what they do is, these mice, they lack all microorganisms, completely devoid of, on their skin, on their GI tract, nowhere air, and they're tightly controlled and monitored. They're microbiologically completely sterile, nothing at all. No living organisms you can find. And strict husbandry protocols, testing regimens. This is one of the lab which just hosts germ-free mice and does a lot of experiments over there. And what they found, the first breakthrough research came that there was an exaggerated hypothalamic pituitary adrenal axis which responds to stress as compared to somebody else. So why was it happening? The only difference between others and this was it was completely devoid of a microbiota. This is an astonishing fact which has come to play. Human body, all of us know that we have about 26,000 genes. 50% of them, they get representation in the brain. And a rice plant, bhata's ropter, it has 46,000 genes. So that means it is much more evolved than human beings. How is it possible? So we felt, inferiority complex for a human being as compared to a rice plant. And then we came to know that we have microbiota. There are about 1,000 species, 2,000 genes per species. So 60% of our stool weight for weight, it's actually the microbiota. So the overall predominant bacterial groups in our microbiota are the firmicutes, which are gram-positive, and the gram-negative bacteroidetes. The majority of them, they reside in the most distal part of digestive tract, that is colon, 10⁻11. Initially, we thought they're about 13 times more than us, but now we know that it is almost equivalent to the number of cells that we have in our body. That is the number of microbiota that we harbor. And the number of diseases. So it looked like a superstar. Couple of years ago, we had a debate. Myself and Jagdish had debated on vitamin D and its association in one of the conferences at Surat. And we felt that Parkinson's, Alzheimer's, multiple sclerosis, depression, schizophrenia, higher cognitive functions, autism spectrum, anxiety, everything. So microbiota has some role. And now slowly and steadily, the research is pointing, yes, probably yes, सगळ्या बोलण्यामध्ये काहीतरी तथ्य आहे. So they came out with this human microbiota project. So this is considered, now we have accepted the fact that this is indeed a superorganism and it is mutually co-evolution. I love this word. That means we human beings, we protect them, we nurture them, and they in return also help us. So it's a mutually benefiting synergistic coexistence of microbiota and human beings at various places. So the human microbiome project is a big project. So they set up to analyze the unique relationship to determine the role of this microbiota, not only in health, but also in disease. Now, this is particularly important because all of us are talking about be sterile, keep everything sterile, cleanliness. We do not want any microorganism. Bar, haath dho, use antiseptics, everything. Not only the treatment, but disinfectant use, harsh cleaning methods, wherever possible, clean and destroy everybody is all the microorganisms. They could be notorious. That is the assumption that we follow and which is frequently marketed and sold as necessary for good human health. But is it true? So that's why this human microbiome project that I was talking about, they studied all these microorganisms and there were about 250 healthy people initially enrolled over here. And the samples were taken not only from lymph, blood, vagina, GI tract, abdomen, airways, heart, and everywhere you talk about it. So initially, as I said, they felt that it was more than 10 times. Now it is roughly 1 to 1.3 times. So these microorganisms, their genomes, and the environment. Any disease, we always start off with three things, either a host, either microorganism, and the environment. So there is always a interplay, and whichever has a power superseding other, that wins. That is how we know. And this is now the fourth dimension to this. So all the left things that are known, and on the right side, you can see that almost 80% of sequences are already done. Thanks to, again, COVID, we know this terminology, which I would have, been hesitant to use the called genome sequencing. Somebody would have said, hey, what is theoretical something? But come COVID and everybody knows about genome sequencing. So 80% of them are already known. So only 20% are not matching. Functional unknowns, 40% genes without a match. So they have a great functional database, and whatever new comes, they try to match that, and they try to see what is the new microorganisms which is coming over here. So you would say, Dr. Upendra, what does it microbiota exactly do? So these are the roles that is a fascinating role everywhere, proved beyond doubt. So mucosa or gut-associated immune system. So your immunologic tolerance that we were talking about, epithelial function, motility, vascularity, it is done by them. The commensal microbiota contribute to the homeostatic functions of pathogen exclusion, immunomodulation, upregulation of cytoprotection, apoptosis, maintenance of thick barrier function, all is done by that. Last but not the least, the energy, 10% of your energy. We always used fiber that they do not give any energy. So they're only good for GI tract, to have our motions next day, maybe reducing cholesterol to some extent. But no, this has been proven that they take that last 10%. So if you are completely devoid of any energy, getting anything from food, these are the microbiota, that final 10% from the fiber also, and they provide you. They convert it into short chain fatty acid, like acetic and butyric acid. And then we talked about vitamin K and folic acid. Who gives that? We knew that it is happening somewhere in GI tract. It is the microbiota, metabolism of dietary xenobiotics, so on and so forth. And last but not the least, the enteric neuromodulatory process has a bidirectional association with the brain. And it's, remember, bidirectional. So nobody is boss and nobody is subordinate. So both control each other. So these gut microbiota that do the maturation and development of both, we want to have both equal coexistence of the enteric nervous system and the central nervous system. Now, if this microbial colonization does not happen, it will lead to our altered gene expression. Turnover of neurotransmitters will get completely hampered. And that is why it is important to have them in our gut. So delayed gastric emptying, intestinal transit, that is also happening. And the microbiome influences our gut motility. The fluid handling, which is so important for gut to do so much of fluid that we are getting into our GI tract. Who handles at the end of the day? It is the microbiome, which is sitting over there, which helps us. And then allergies. We always knew that there is something to do with allergies and our microbiota. Is it been proven? Yes, of course, with evidence. 80%. Now, believe me that our immune cells that we are all day in and day out, Dr. Jagdish also talked about the immune cells. Where are they? 80% of them are in and around our gut. So our early life, if you do dysbiosis, and there comes the importance, the practical importance. Early life dysbiosis can happen because of injudicious use of antibiotics will completely hamper this. So it is linked to increased risk of chronic inflammatory disorders. We always knew about Th1, Th2 hypothesis. We knew about the infections and the allergic autoimmune diseases, Th2 association with too much of exposure to only Th2 kind of response. So this is completely altered by them. Birth mode, I'll come to that. How even a vaginal delivery or a cesarean delivery will change into that, and will tell you whether you have an atopic or asthmatic disease. And this is pathogen-associated molecular pattern. This is a great thing, fascinating, and you hear it henceforth from today onwards in any infectious diseases conference, PAMP. So every microbiota has a pattern, and there is a database, like in a police chowki, they have the fingerprints of all the known notorious gangsters in the city, and anytime somebody is caught, they try to match. This is the same way. So the body has, and there is a pattern recognition receptors in the body. So who influences the action of this? It is our microbiota. So this is a great correlation, the vagus and what I was talking about, the CNS and ENS. The intestinal nervous system is not less. If we have trillions of cells over there, we have about 200 to 600 million neurons over there. So this interest in intestinal vagal activity, that will lead into enteric neuroinflammation and enterocytes over there. So two ways, a vagus as well as the perivertebral ganglia. That is how the bidirectional communication takes place. This is a leaky gut, exciting concept of that. So for any disease to occur, this gut thick epithelial barrier has to move, has to get disrupted. And who protects us from that? One of the factors is microbiota. Lipopolysaccharides, all gram-negative infections, how do they enter the body? It is because of production of them. And who protects us? The thick epithelial barrier function. If it is disrupted, these endotoxins are just waiting to enter the intestinal lumen and then into circulation and causing havoc. So this is another inflammatory cascade that would set in, which is also prevented by the microbiota. Gut microbiota, hypothalamic pituitary adrenal axis. When I am stressed, actually what is happening? This is what is happening. So cortisol, we know, is a hormone which is secreted, which causes a leaky gut. This leaky gut, again, we know that the epithelial function is completely disrupted. Then it releases the immunomodulators, interleukin-6. Again, we know because of COVID, it downregulates glucocorticoid receptors on one side and on other side, the HPA axis is exaggerated. And that is how this microbiota play a role there also. And this is a beautiful concept, the brain-derived neurotrophic factors. Why we are happy, healthy, in perfect state is because of them. They make us feel good. And that is also completely the secretion of serotonin, acetylcholine, and dopamine. Acetylcholine and dopamine, they're all controlled by our gut microbiome. The neurotransmitters and gut is an exciting concept. We always thought serotonin is produced by brain. No, 90% of our feel-good serotonin, friends, comes from gut. 50% of dopamine comes from gut. So low neurotransmitter you can make out. There is a depression. And if it is low in the GI tract, of course, what would happen? Constipation, depressed gut. If it is high, what would happen in the brain? Anxiety. If it is high in the gut, irritable bowel syndrome. Got it? Such a wonderful correlation. So bacteria through their intestinal wall, they probably come out and talk to the brain. Oh, do you need more? Oh, yes, I'm feeling low. So, okay, let me see. So this is how the bidirectional communication between the gut and the CNS, ENS and CNS takes place. So if this is low, then what would you do? There's a beautiful book, if you get hands on it, please read, Secrets of Serotonin. Serotonin is a great neurotransmitter. So if you have good, enough, you feel good, great. But if you have low, you would always feel, let me eat something, let me have a peg or down, get a migraine or insomnia, depressed. In fact, millions of people are taking antidepressants without knowing the importance of, they can just modify their diet and come back to their good levels of serotonin. So if there are no microbes in the body, serotonin level, again, poor germ-free mice came to our rescue and they told us the serotonin goes down by 60%. So if you are feeling low, friends, have something starchy, by which I mean have complex carbohydrates, which would give you some food to that intestinal microbiota. So the starches increase our serotonin by 10%. But on the contrary, if you have a very high protein diet, it will reduce your serotonin down by 35%. So when there is anxiety, then at that time, probably you may have, because you want to reduce the level of serotonin. But if you do the other way, that if you have the opposite way, when the serotonin is to be increased and you have a high protein diet, what happens is at that time, nothing would happen. But at the end of the day, actually your levels go down so much that you start having binge eating in the evening. And that time you always choose simple carbohydrates, which actually to some extent improve your serotonin. But then you know what happens after simple carbohydrates overeaten. So that is how one should know our personality and what kind of diet we can do. So healthy diet, which is good of high fiber. Now half plate is vegetables and fruits, everybody knows. So before thinking twice, always have first bite of fruits or vegetables, then something else. 50% has to be there. It has to be high fiber, low fat, some prebiotics. Breast milk, of course, to start off with. This is going to give you a normal microbial community. An unhealthy diet with formula, high fat, low fiber, and this is what would happen. And these are all beyond doubt. The NEC, celiac, IBD, irritable bowel, gastroesophageal reflux, and colonic cancer are all results of dysbiosis. The words are self-explanatory. What is eubiosis and what is dysbiosis? So how can I build a beneficial microbiome, and what is the role of a pediatrician to do that? There's an excellent article in which tells us, of course, the breast milk has to be, it's a birthright of every baby. You get fructooligosaccharides, of course. These are the ones. We always knew about the macro and micronutrients from breast milk, but we never talked about the bioactives and the entire focus. So if you ask me to talk something of breast milk right now, I can talk for half an hour without talking about the macro and micronutrients. So that is how the bioactives have found to be a fascinating role in breast milk. So antibiotics are absolutely a big, destroying factors for microbiome, formula diet, urbanization. But before going ahead, let's pay attention to Dr. Yevla is sitting here, our avoid antibiotic abuse man. Over misuse of antibiotics, early life, especially medication exposure, what it results is less diverse gut microbiota, reduction in risk clostridials, increase in antibiotic resistant bacteria, and benefits of breastfeeding are completely negated. So think 10 times before just giving antibiotic, especially in the infants. Let's be very practical about it. So human milk can do this. So all these microbiota bioactives over there, abundant. So oligosaccharides are so abundant. The companies, the formula marketing companies are coming out with something trying to add on, but nobody can match the breast milk. So it favors the proliferation of specific bacteria in the gut, bifidobacterium especially. And on the contrary, mother's diet also influences. If the mother has a very high fat diet, the energy content in human milk will affect the proliferation of bacteria. So healthy, balanced diet of mother and then the breast milk and then everything. But breast milk, this is a fascinating thing. All of you are practicing neonatologists also. NEC, have you seen it recently? What has changed? We had so much of NEC a decade or 2 decades ago, and now suddenly everything has gone down. We completely stopped formula in all these babies. If at all possible, we are giving only EBM, and that is how it has changed. So what was causing NEC all these years? It's a triad of aberrant intestinal microbiome, immature mucosal immune system, stressors, and formula-based feeds. And the moment we shift completely to only, if possible, milk, that to mother's milk, then we change completely. So this was because of this. The human microbiota that you change completely, influence and modify just because of using a breast milk has completely changed NEC. This is what happens. You can see what happens after a vaginal and cesarean. Before breast milk, let's talk to delivery. Look at the microbiota change in vaginal delivery and the cesarean section. On day 4, if you see the sampling, if it was done, this is where the vaginal delivery, and day 4 vis-a-vis cesarean section, you can see the enterococcus, streptococcus, staphylococcus, klebsiella. The word, we really hate these kind of organisms, and look at their predominance. Fortunately, it's not lifelong. Just because you have a cesarean baby, by infancy, by 21, it comes to almost matching, and by 3 months, the flora becomes good. But first 3 months after cesarean section, there is great, disadvantage for babies, as far as if you overzealous use of antibiotics will be a great disadvantage. The fecal microbiota they tested, 72% of vaginally delivered babies vis-a-vis 41% so C-section. Look at the change. It's a huge change, isn't it? So that's why our babies, as far as possible, should have vaginal delivery. Then they started doing research that C-section babies are missing key microbes. So you know what is happening? Fascinating research that they're painting after C-section, the moment baby is out on mother's abdomen, they take swab from the mother's vagina and just paint the baby. So that's why the baby gets at least whatever she would have got by vaginal delivery, those microbes baby gets. So this is a scientist swab C-section babies with mother's microbes. Instead of that, let's try to have more vaginal deliveries. So till now we have understood, if I have a normal microbiota, I'm going to have a normal development. If I have a missing or dysbiotic bacteria, I can have altered immune development, altered metabolism, altered ENS. So this is all, and the disease is a correlation. Second, before that is placenta. We always thought placenta is sterile, isn't it? No, placenta has huge number of microorganisms. So if you correlate that microorganisms, you would say it could be, if you are saying microbiota, then mother's GI tract. Is it true? No, look at this, what is happening. World wants to have less number of premature deliveries. So this is what happens. The inflammation, bacterial invasion, inflammatory mediators, from where do they come? Look at this periodontal infection. So the placental microbiota correlates with mother's oral health, not with mother's GI tract. So what has that caused? Look at this, this is happening. So oral microbiota and pregnancy. So the obstetricians, to prevent premature delivery, what they are doing, that having a dental screening, so have better periodontal hygiene to prevent prematurity. So that is how they're getting the fascinating research, bidirectional relationship proposed between oral microbiota and pregnancy. So these are a couple of papers which I came across, and everything had, it was not a single standout paper. Everything had something to say, and these are all 21 and 22. That obesity, fascinating evidence over there, change of microbiota, possible role of in autism spectrum disorders. I'm not going to in details about dysbiosis, but only thing one should remember, if you are managing autistic patients, they all have tremendous GI problems. So why do they have it? And 1/3 of them, they have not been able to have genetic link. So probably they could have some role there. The current understanding, ASD, irritable bowel, absolutely logical, rational, that probably a microbiome gut-brain axis disorder. And so that's why they are trying to do that. And finally, we are all worried about, we are marching towards the other extreme. And each one of us sitting here is worried a little bit, I should not have Alzheimer's. Of course, touch wood, we'll be keeping fingers crossed, we should not have. So neuroinflammation is found to be the pathology for even Alzheimer's also. So this is considered Alzheimer's is now talked to be as type 3 diabetes, something like that, dysbiosis. So finally, pseudomembranous colitis. All of us knew it is antibiotic associated diarrhea is the only condition where probiotics have some role. We have oral vancomycin and metronidazole as a drug of choice therapy, but fecal transplant, 100% cure rate for pseudomembranous colitis. Why does it happen? So fecal transplant is coming in a big way. Fecal microbiota transplantation, bacteriophage therapy, psychobiotic effect. So these are the different ways in which the science is progressing and the role of microbiota is getting established. So what should I do as a pediatrician after listening to all this? Quite some theoretical, but quite some practical also. Exclusive breastfeeding. Before that, vaginal delivery not under our control. Exclusive breastfeeding has another advantage today we have understood, and timely introduction of complementary feed. Variety of diet is important. Balanced diet, which is full of carbohydrates, proteins, fats, micronutrients. Intake of indigestible carbohydrates is so important. Fiber is not only for diet, but also because they provide all the food for that microbiota, the inulin resistant starch, fructooligosaccharides, which increase the short chain fatty acids. Foods with natural prebiotics, the garlic, onions, dark green leafy vegetables, bananas, everything, fruits, vegetables in general, beans, whole grain, wheats, oats, barley. Introduce these foods in small amount if you have never eaten, but still, because even as adults, we need to build our microbiota. 60,000 foods over shelf in USA, out of them 72% have added sugar, and that may not be labeled that this is sugar containing. So they completely disguise you by labeling faulty labels. So please be careful about eating these kind of foods. And now final one comment about Americans can go to any extent. So that's a great project. I talked about human microbiome. So American Gut Project, you know what they did? 12,000 samples, University of San Diego, they did this, and what they said, they've advertised. So you are supposed to take this kit and you can take your own fecal sample. If you want, you can take it from your dog also. You can get from gut, mouth, skin, from anywhere. They tell you how to parcel it, how to put it inside and send them to, and then they'll give you sequencing and analysis, and they have done a great thing. And what was the final outcome of this? The 4 things which I would like to, it's a big revelation, but then plant-based diet. So even if you want to have proteins, have plant-based proteins. That is number 1. Antibiotics, definitely. Overzealous, injudicious, indiscriminate use of antibiotics definitely completely hampers your microbiota. Third, very exciting. Look at this. Detection of agricultural antibiotics in people who claim that they haven't taken antibiotics in the year prior to their sample collection. अरे last 1 साल से मैंने एक भी antibiotic दिया नहीं, खाया नहीं, कैसे आ गया ये? So it was because of agricultural. So agricultural industry uses antibiotics like anything in all the animals, farm animals, and that is how you get it. So this was a great revelation. And composition of microbiome. So this study extended from US, UK, Australia. They did something in this American Gut Project in collaboration with Asia also. But this was a great thing. US and UK samples, all patients were clinically diagnosed to have depression. The microbiota was similar in both these things. So some substance in it also. So nutrition friends, avoid refined carbohydrates and sugars, have plenty of vegetables, legumes, as I said, eat fermented dairy vegetables, healthy fats, high quality proteins, select meat with little or no antibiotic residue. I don't know how we can control it. Acid in stomach is very important. Another very important practical point, avoid reduced duration of drugs that lower. And one drug which is found to be hampering is PPIs. We are indiscriminately, overzealously using even PPIs. Let's be very careful about long-term use of PPI. A short duration may not have, but long-term, excessive use of NSAIDs. And finally, this is a great statement. It takes at least 3 months for our microbiome to recover after a 1-week course of antibiotic. So this is how one should remember. So friends, to conclude, I would just say, we should have our gut bug cultivation. We should respect them, love them, care for them, feed them healthfully, make sure they get enough sleep. That is the number of papers, your sleep and your microbiota, how it changes. Your exercise, physical activity and your microbiota. You and they will be much more healthy. Thank you very much. Not before. Invitation to each one of you. I take this opportunity always, I never miss on that. 19th to 23rd February, Gandhinagar, our own conference, along with the IPA, which is happening after 45 years in our country. Last was in 75. So imagine after 45 years where you and I would be, maybe 30 years, because there are more than 130 members. So each country doesn't have it. It comes only once in 2 years. So please, if you have not yet registered, you register, encourage your students to make at least send a scientific abstract. We have extended the date for 30th November. Thank you. Thank you so much again.
Thank you
sir.
very
It
much,
was an
Upendra.
excellent talk.
And Dr. Devi and I had a gut feeling when we came here that this was going to be an excellent talk and that no questions will be needed.
Thank you. Thank you. Thank you, Dr. Devi, Dr. Anjali. Thank you so much.
Thank you, sir. May I please request our chairpersons, Dr. P.N. Devi and Dr. Anjali Gokarne, to please felicitate our speaker, Dr. Upendra Kinjwarikar.
Yeah. Thank you. Thank you. Thank you.
May I please request our senior most pediatrician from Nasik, Dr. J.S. Patel, sir, to please come up on stage and felicitate our chairpersons.
My pleasure.
For the plenary session, I request Dr. Vijay Evle, Dr. Bela Verma, both from Mumbai, Dr. Dhanya Dharmapalan, and Dr. Jagdish Chinappa sir again to come on the dais as speakers. I also invite Dr. Tanu Singhal from Mumbai to moderate the session. Century ago, the great man made an invention which is considered the best ever gift to humanity, which was named as penicillin. And today we are using hundreds of such molecules called as antibiotics. Without these wonder molecules, we cannot even think of treating dreaded infections. It took us several years then to learn that these lifesavers could be life killers also, if not used judiciously. Yet we marched ahead using antibiotics right, left, and center and gave birth to a tragic reality of antibiotic resistance. We are all guilty of initiating and now perpetuating the misuse and disuse of gems of therapy. And it is high time now we correct our past mistakes and select the right patient for the right antibiotic in the right form and in the right dose for right duration. So think before you ink. Over to you, ma'am.
Good morning, everyone. And first, I would like to thank the organizers and especially Dr. Sharmila for giving me this opportunity to visit Nashik and do this presentation before you. Okay. So I think while our panelists settle down, and so what we are going to do in today's talk is that we will discuss common scenarios in both outpatient and inpatients. And we are going to talk about empiric antibiotic, not definitive. Like, you know, we don't have a microbiology report before us when we prescribe these antibiotics. And we have, it's like an MCQ form. So you have a question and then you have various choices. And I want all of you in the audience to think about what choice they would want to use when this when this situation comes, and then try and compare their choice with what the panelists tell us. And what I would like the panelists to do, because we have 10 case scenarios, so we have roughly 3.5 minutes per case, to first think about what pathogen we would be dealing with in that situation, what would be the drug resistance likelihood, and then which choice is correct and why the other choices are not very correct. And also, I have provided a set of antibiotic options, but if you feel that there's another antibiotic which fills the bill better, you are requested to add on to that. And finally, there is no right or wrong answer. There may be subtle nuances which we may discuss, and I mean, we'll discuss them as it goes on. So with that, we go to the first case. So this is for you, Dr. Chinnappa, starting with you. So this is a 6-month-old infant who comes with a prodrome of fever, cold, cough, and now fast breathing. The respiratory rate is 60, and he has bilateral creps and V's. Saturation is 90% on room air. So the child is meeting the definition of a pneumonia. He's tachypnic and febrile, and he's having hypoxia. So you will, of course, this child is admitted, but the option is that what antibiotic would you like to use here?
I think I will. Is it audible?
Yes.
Yeah, okay. Thank you, Dr. Tanu, for asking me to go first. Well, if you look at a 6-month-old who presents with this kind of a syndrome, it's more likely viral in nature. This is pneumonia, no doubt, but it looks more likely to be viral, primarily because we have multiple systems involved in terms of cold, cough, and fever. The other thing which we also need to know is that how long this problem has been going on. If it's just a day or two, then probably it's only viral. If this child has had this problem for, let's say, 3-4 days and then goes in for rapid breathing and a drop in saturation, then there could have been a bacterial superinfection. So we need to look at basically the progress of this condition. At the outset, with this child, I would just watch and investigate and see whether there is a possibility of a bacterial infection in terms of a localized lung pathology, and then start an antibiotic. Upfront, if this child were to come, let's say on 4th or 5th day, then maybe I would start with ceftriaxone, pending the investigation that it's only viral or otherwise. So this would be my choice, primarily because if you look at a post-viral infection, 2 organisms
But
predominate.
let us say this is just 2 or 3 days old situation. The patient had 1 day of cold cough, next day had fast breathing.
Yeah.
And he has a generalized pathology on examination, bilateral creps and V's. V's is also there.
Yeah, so I think it depends on where you're located. If you're located in a hospital, I would look for bacterial etiology, primarily with investigations, if there is a possibility. But this seems viral in nature, so I would just wait with symptomatic treatment pending investigations.
Yeah, so you're saying that this is more looking like a viral bronchiolitis, the typical thing, which is mostly viral, RSV and all. But you also mentioned that you would like to do tests. So would you like to actually subject this child to a CBC, CRP or a chest X-ray, or just wait it out?
If you're admitting this child, most definitely will investigate, because no parent is going to just allow you to just admit the child and watch with
Yeah.
IV fluids and all.
So at least a X-ray you would do in
An
that
X-ray,
case.
cut
Yeah.
down, see where
If
we
there's
are.
hyperinflation or something. But I think a point which you mentioned is that, it also depends on how long the child has been sick. This is also a flu-related illness. If the child is sick for 5 to 7 days, then you would, with the prodrome and then deterioration, then it could be a bacterial superinfection. And in that case, you would add.
Hello.
Dr. Bela, you wanted to say something?
No, same thing what sir is saying, that supportive management with maybe bubble CPAP and,
No, so the
oxygen.
baby is
for
like on
90
nasal prongs
saturation.
is saturation, is
yes.
saturating well. So
Yeah, and wait and watch if anything comes positive.
So and in this situation, if you have the facility, you could also do a nasal swab, do a PCR for RSV or a nasal antigen test. And if you find a virus there, then you could probably just withhold antibiotics and just give supportive care, and the baby would improve. Okay. So I mean,
Can I make a point here? See, it's all good that in metros all these facilities are available. For most of the practitioners who are practicing in the rural parts of Maharashtra, 2 things important. Bilateral crepitations very often is equated with a bacterial or a viral pneumonia. Let's not forget that 6-month-old child with a viral prodrome, clinically happy-looking child, is unlikely to be anything other than bronchiolitis. So that should be borne in mind. And then you have hyperinflation, etc. So you
Yeah.
can very well withhold antibiotic in such a child.
Yeah, so and even if one would go for an antibiotic, then let them just stick to the basics, like the ceftriaxone or amoxicillin, and not go, trigger happy with higher antibiotics here. I think the appearance of the child is really important, whether the child looks sick or whether the child looks happy, because most viral illnesses, the kids look well. Okay, so let us come to a different situation. And Dr. Yevle, this question is for you. So you have a 2-year-old allergic rhinitis adenoids. I don't know about you, but in Mumbai, we have an epidemic of allergic rhinitis. That's 50% of my practice is that. And this child has had recurrent otitis media, 3 episodes in the past 3 months. And he has received antibiotics every time. He's been treated with cefpodoxime, cefdinir, then coamoxiclav for a fair duration of 5 to 7 days. And the doses were correct. And now he again presents with high fever along with left ear pain and pus discharge. So let us say that this otitis media which he has merits antimicrobial therapy, because we know that when otitis media you can just give paracetamol and watch, but this is not the case here. So again, I would like to ask you, in this situation, what do you think would be the pathogens? What is the likely resistance pattern? And which antibiotic would you like to give from these choices, or any other one on your mind, and why you would do so?
So first of all, this is a 2-year-old child with fever which is very high, 39 degrees. So it's a severe otitis media. He also has recurrent otitis media, and has used beta-lactam antibiotics in the previous, say, 3 months. So certainly the risk of resistance would be high. So we would like to use something which would cover resistance. If you talk about pathogens causing otitis media, it will be again big three: strep pneumonia, Haemophilus, Moraxella, and of course viruses. This child also has underlying predisposing factors for recurrent otitis media, that is allergic rhinitis and adenoids. So while we address those underlying predisposing factors, said that, since the child has received beta-lactam antibiotics in the past, has severe otitis media disease, and this is the 3rd episode, I would definitely use a BL-BLI combination, and I would like to use high-dose amoxicillin, because this could be the failure of 40 mg per kg in the pneumococcal otitis media. This could be antibiotic failure of 40 mg per kg dose. So I would go in for high-dose, extra strength co-amoxiclav. That would be my choice in this patient.
Okay, and what about sending a pus culture here? Do you think that is of any value?
It really doesn't help much, regardless of the this thing. We know that these are the 3 common bacteria which cause, and it really doesn't impact
And
your
most of the
antibiotic
times
choice.
Grow Klebsiella, Pseudomonas, which will throw
These
you
are
off the track.
You disregard that. You don't really
And
change
if
your.
you have to get a sample, then it should be by myringocentesis. It cannot be the
Absolutely,
sample which you
absolutely,
get in the ear.
absolutely.
So
Yes.
sending a sample. Dr. Dhanya, you wanted to add something?
Yeah, just wanted to add, usually when the otitis media relapses before 15 days of treatment, it is usually considered failure of eradication of that original pathogen. But if it is after 15 days, it could be a different pathogen. But in this case, it could be drug resistant strep pneumonia, it could be beta-lactamase producing and H influenza or Moraxella. So I agree with Dr. Yevle about co-amoxiclav. Regarding the option D, cefuroxime, just want to let the audience know that cefuroxime is not very good for either H influenza, resistant H influenza, or to eradicate drug-resistant strep pneumonia. So in this case, co-amoxiclav, and I think the ratio is 16:1 in this extra strength. So it's
14:1.
14:1, sorry.
Yeah, it's 14:1. So Dr. Chinnappa wants to make a comment because
Yeah.
he's the expert on otitis media.
Like that, I think what is very important in this child is that the child's pain has not been relieved after the tympanic membrane has burst open. You have purulent discharge coming in and the child still has pain and high fever. So that means there is a focus of inflammation somewhere else. It's not the middle ear. So this child has to be investigated for mastocytitis.
Yes.
There is definitely a high scope of mastocytitis in this child, primarily because of persistence of pain after rupture of the tympanic membrane. And in this situation, it is better to go with a parenteral antibiotic rather than continue with an oral antibiotic. Because this is a child who is not looking good. He is having 3 episodes in the last 3 months. He has had some severe otitis media. He has pain despite rupture. So I would definitely investigate this child for mastocytitis and start on IV cefuroxime. I won't wait with co-amoxicillin.
That's a good point.
So I would like to add that it is recurrent allergic and inflammatory stimuli which is causing adenoid hypertrophy perhaps. It could also be causing comorbidity of obstructive sleep disordered breathing, which is
Yeah,
leading
but
to
in this
recurrent
session, in
otitis
this
media.
session, we are
So
mainly focusing on the
whether he needs
antibiotic.
surgery,
Yeah.
whether we should look
no,
for the
so we
surgery. He's
are
about
only-
2 years old.
We are mainly discussing here the antibiotic choices. I agree with your point that we have to take care of the predisposing
Comorbidities
factors.
also.
So I think the point is very well made. that here is a situation where you have a high likelihood of a drug resistance pathogen. And you must understand that the ear is also a sanctuary site like the CNS. And the middle ear, the penetration of antibiotics is less. So you may have to use relatively higher doses. So while in your garden variety case of acute otitis media, it is fine to either start with amoxicillin or maybe co-amoxiclav. In a patient like this, you would either use parenteral ceftriaxone or you would use this high dose extra strength co-amoxiclav, and investigation for mastoiditis is extremely important. We have seen cases where you do a CT and you find that they have mastoiditis and they need longer duration of therapy and higher doses. Okay. Now let us come to the next case. This case is for you, Dr. Dhanya. 1-year-old in Mumbai, when there is an ongoing flu epidemic, he has high fever for 3 days with cough and breathlessness. He's admitted, he's sick, toxic, has tachypnea with low saturations, reduced air entry on the right side, and you see an X-ray which shows complete opacification of the lung field on the right side. So in this situation, what would be your pathogens that you want to address, and what antibiotic would you like to use?
Though there is a localization to the right side, if there is a flu epidemic ongoing and you have a sick child with a severe respiratory, I would add oseltamivir empirically in this child, even if it looks more like a bacterial, and will cover with ceftriaxone. Vancomycin, it is not as urgent as a meningitis that I would upfront cover with vancomycin. I would stick with ceftriaxone and oseltamivir, and then vancomycin will not be an upfront cover for me.
But this pattern of an X-ray looks, and this illness is just 3 days old.
3 days old,
So,
rapidly
it
progressing.
doesn't seem like there is a viral infection which has got complicated with a bacterial pathology.
Toxic
It looks like a bacterial pneumonia to begin with. The situation would be different if you had fever, cough, cold for 3-4 days and then tachypnea and then relapse of high fever. That tells you that you're dealing with a viral flu
Viral.
which has got complicated with a bacterial thing. Here, I mean, I don't know.
I would go with ceftriaxone and vancomycin, considering he's a sick child, rapidly progressed in 3 days, so would cover
So you
staph
would like
also.
to want to cover an MRSA
Staph,
as
yeah.
well. So, Dr. Yevle, what would be your choice between ceftriaxone and vancomycin? Because that is the dilemma amongst people.
So if you look at, I mean, there is underlying flu epidemic going on. So oseltamivir would be added. Ceftriaxone, oseltamivir, and looking at the rapidity of progression, I mean, we may add vancomycin as well. But oseltamivir would be, added in this child on the background of this. I have a question, Tanu. The other lung looks absolutely clean. Isn't that little uncommon with staphylococcal infection? Why can't this?
Yeah,
Could be
I mean, here,
streptococcal
I would worry about staph more than influenza. I mean, in this situation, of course, there is a background of a flu epidemic, but right now the problem looks more bacterial to begin with, to look at. Even in flu, it was odd to have the other lung completely spared and you have just unilateral involvement. That is odd. Okay, so we can debate about the oseltamivir or not, but leaving aside oseltamivir, would you want to cover MRSA upfront in a child because he's like sick, with SATs of 92%, he's rapidly going down. I think most people would, because we are believing and there is an effusion with a pneumonia. So you are thinking that you would be dealing with, it could be either pneumococcus or it could be MRSA. And in a background of flu, you can have these complications.
Yes.
So I think in this situation, and there is also a possibility of you making an etiologic diagnosis because you would be sending blood cultures, you would be doing a pleural tap in this patient. So you would have the option for de-escalation also if required. Maybe if this patient was not hypoxic, not very sick, we could have gone with ceftriaxone alone. But what do you think of this choice, number D? Because a lot of patients who have a pneumonia, who are sick, are put on piptazo, linezolid. Of course, azithromycin is not needed because it's a 1-year-old child and mycoplasma is unlikely.
I think one simple message, piptazo is for nosocomial infections which are not involving CNS. That message should go home. Piptas has no role in such a situation in pneumonia as a respiratory infection, unless it's a nosocomial pneumonia.
And also, yes, Dr. Jagdish, you want to make a
Other choices.
Some people would give Mero-Vanco in this patient.
Yeah, absolutely. I think the one drug which should not be used for a condition like this is meropenem. Because meropenem has very poor penetration into the lung tissue at this stage. So meropenem is a definite no-no.
Yes.
So I think
Amikacin is also a
the
definite
correct
no-no.
choice would have been ceftriaxone, vancomycin, and oseltamivir. I would have not said that you don't give oseltamivir.
Okay.
So even though we are a little late, we are on 3 days,
Within
which is a
48 hours.
bit late. But even then, I suppose that in a patient like this, I would cover with all three because we can't be sure. So I think I
Fair
would
enough.
cover with all three.
Okay, so I think the main messages here are that no matter, even if the child is very sick, you don't have to use Piptaz, you don't have to use Meropenem, you don't have to use Amikacin for treatment. And also in a respiratory illness in India, if you want to use a MRSA cover, you either use vancomycin or use ticoplanin to begin with. Don't use linezolid because that is a very good anti-TB drug. And some of these community-acquired pneumonias, though not in this patient, but a more insidious pneumonia, maybe TB, and then you will mask it. So I think linezolid is something which you would use if you have proven that it is MRSA and now you want to continue therapy later after the initial parenteral therapy. That is when you would switch over to use a oral drug. Okay, this question is for you, Dr. Bela. This is a 4-year-old with gluteal abscess. The pus is drained and sent for culture. Now, what empiric antibiotic would you use here?
So if we are treating this patient on an OPD basis, if it's not too sick
Yeah,
with
this patient is in
systemic
OPD.
illnesses,
Yeah.
I would either give a cephalexin, 20 milligram per kilogram for about 7 to 10 days, or a clindamycin if I want to cover MRSA. If I suspect MSSA, it would be cephalexin. And if I suspect
how
an
would you-
MRSA, I would give clindamycin.
How would you, kind of suspect MRSA or, MSSA here? It would be, you would have to wait for the culture reports, but is
Yes.
there an urgency to cover MRSA here? Because the patient is stable, you have
Yes.
a gluteal abscess. So, I mean, you can even change after 2 days.
Yes,
It's a
we can
OPD
change or
treatment that
step
you're
up, or
giving.
if the child becomes ill with systemic illnesses or high fever, tachypnea, tachycardia, if he is admitted, then we can step up the antibiotic.
Yeah, so I think one important message is the previous case and this case is different. The previous case was a sick child in the IPD where you don't have time to escalate therapy and you would give your best shot in the beginning. Whereas this is a case which is an OPD patient. So even if it turns out to be MRSA after 2 days, you can always modify and you may not need to start. Okay, so I would also want to ask you, there are many people who would use coamoxiclav here and instead of cephalexin. So what is your, why wouldn't you choose coamoxiclav?
I
Because
would
that
choose
would work for
coamoxiclav if-
MSSA as well.
Yes, that would cover the drugs and the resistant beta-lactam drugs, which would be the organism who's resistant. So coamoxiclav is also another option which we can use for about 10 days.
But what are the downsides of coamoxiclav versus cephalexin?
Uh,
Is there anybody, maybe somebody could add?
no, I don't know.
Yeah.
Spectrum, coamoxiclav.
Yes.
It's a broad spectrum antibody, unnecessary exposure to.
Yeah,
broad
you
spectrum
would, it would it would destroy your gut anaerobes. We just heard from Dr. Upendra that you have to protect the gut. It would destroy your gut E. coli and anaerobes. So you would become colonized with more resistant pathogens and it would probably have more side effects like diarrhea.
But abscess in the gluteal region, one may get tempted to use coamoxiclav thinking of covering gram-negative and anaerobes. But most of these abscesses are staphylococcal abscess.
And
So there is no
also-
need to use any other.
And it
agent.
also depends what is the location of the abscess, because if the abscess is very close to the perianal area, then you might want to use coamoxiclav because you want to cover anaerobes and E. coli. But if it is a standard gluteal abscess which began with a boil and then went on, it's most likely staphylococcal.
Anu, if I can come in here, I think I would not give an antibiotic at all.
You would not give antibiotics.
Because the point is the current recommendation for skin and soft tissue is there is no need if you have drained the pus to give any antibiotic.
Yeah. Yeah,
the-
but this child does have a lot of abscess inflammation and
It's not there.
redness.
Not put there.
Not written.
So that's a good point.
So
So-
I think if this is a pure abscess which has been there for a few days, you have drained it and there's nothing else, I would not do anything other than follow this child up. Anyway, you have sent for culture, no? So why can't you wait for 2 days to get the culture
Yeah.
report and
So
then
I
start
think
the
the point is that you may not give any antibiotic at all. But I think sometimes what you also find in these patients are there some other pustules somewhere which are there. Many of them have pyoderma at other sites and you have significant peri-inflammation. Maybe you could use. But okay, what about clindamycin? People are tempted to use clindamycin because it would cover your MSSA and MRSA both. So what is your take on clindamycin?
I think we should not use a toxic antibiotic, potentially toxic antibiotic for a very simple common problem.
Yes. So clindamycin, and the other thing which one must remember is that 30 to 40% of MSSA are actually resistant to clindamycin. That is an important point. So if you're giving clindamycin without a culture report, you may be under treating your MSSA. Okay, fine. I would choose cephalexin if I decided to give an antibiotic, but I would use a slightly higher dose than 20 mg per kg because that is for cefadroxil. Here I would use 30 to 40 milligrams per kg per day. Okay. Now let us come to this next case. Now this is for you, Dr. Chinnappa. This is a 4-year-old who has a history of wheezing in the past, comes with fever for 1 day followed by breathlessness, and the patient has tachypnea, bilateral wheeze, saturations are low. You have started nebulization because he can't take the MDI. I know you're all asthma experts here, so I'm not even going into that area. That's why nebulization is given. Steroids have also been started. And a CBC is sent. Now, you would say that there's no need to do a CBC or an X-ray, but let us suppose it has been done. And the count is 20,000 with 80% polys. So my question is that what antibiotic would you start? Because the resident is very nervous. The white cell count is 20,000. And we are giving steroids. We must cover with an antibiotic.
I presume that you're giving nebulized steroids or you're giving, so if you're giving nebulized steroids, you don't need to worry. Second issue is that
No, we have given systemic steroids also because the case meets the criteria of a severe attack with hypoxia.
okay, fine. So if you're going, even then, the total leukocyte count of 20,000 doesn't scare me at all because this is a response. It's an inflammatory response to any infection, so which may not be bacterial at all. So if this child is going to be observable, if this child is going to be investigated, then I would do nothing right now, not give an antibiotic and wait and watch. I would not start with any azithromycin, ceftriaxone, or with amoxicillin.
And azithromycin is a great favorite for respiratory infections because, and most of it's not only the doctors who are prescribing it, but the patients themselves tell you, we had fever, cold, cough, I've started. I had sore throat, I've started AZ. So it seems like a harmless antibiotic actually, 1 dose for 3 days, no side effects at all. Do you think that is a lot of misuse happening, and is azithromycin really a good drug for respiratory infections like this?
Absolutely, it should be reserved. I think it's a reserve antibiotic because over time, if you're going to have the multi-drug resistant salmonella, you will have to have azithromycin as a backup drug. So I don't think we should use it indiscriminately for any respiratory infection. This child has had wheezing in the past. This child has probably got a viral induced wheeze. A bacteria is highly unlikely to produce an exacerbation of wheeze. So this probably is a viral induced wheeze in a 4-year-old. So I would wait. I won't be in a hurry to start antibiotics in this child. And even if antibiotics, I must have more data. I must have more information, whether there is anything wrong with the X-ray or any such issue, rather than start straight away some antibiotics.
Just want to add that 20,000 could be due to the steroid itself.
But the blood was drawn before the steroid was given. But we know that even in DK, you can have this leukocytosis, which people often think is a marker of infection, but it is a stress response. So I still remember, Dr. Yevle, your presentation once where you said, don't give antibiotics for a leaky nose, leaky gut. And third thing which you said was, don't give it for a wheezy child.
Basically, these are 2 or 3 conditions in clinical practice. One is bronchiolitis and other is this wheezy child, where a lot of antibiotic is used. I would say 99% of the pediatricians would use an antibiotic. That we must refrain from using antibiotic in such situation.
And also for the information of the audience that when you have a bacterial respiratory infection, azithromycin is a poor choice because 30% of our pneumococcus, Hib, Moraxella are all resistant to macrolides. So the only indication of using azithromycin in a respiratory infection is if you're suspecting an atypical infection like mycoplasma, or if you have serious beta-lactam allergy. Those would be the only 2 situations. And the people get better with AZ because it was not needed in the first place, because they will swear by it. I took AZ and in 2 days I'm better. Anything else you would like to add here? Because I think these are 2 situations, upper respiratory tract infections, which are viral, and wheeze, and even bronchiolitis, where a lot of antibiotics are used, which we can avoid. Okay, so we wouldn't give anything. Okay, we come to the next case. This is for you, Dr. Dhanya. This is a 3-year-old with history of grade 4 VUR on left side, and the child is on cotrimoxazole prophylaxis, has fever without focus since 2 days. Urine routine shows 40 to 50 pus cells per hyperfield, nitrite is positive, bacteria are seen, a urine culture is sent, and now we have to start an antibiotic. So what antibiotic would you use in this situation? What are the pathogens and susceptibility pattern you're thinking of, and what would you use?
If I'm treating this on the OPD basis,
Yeah, right now we're
So
treating
this
it on
child
OPD.
is not very sick. I mean, though I understand that there is grade 4 VUR, which makes the child high risk for a resistant bacteria in the urine, but still, since the child is not sick, I would start with cefixime, wait for the culture report, and then upgrade or go for another, rather than upfront covering a resistant bug. Had the child been sick, I would definitely.
And I remember, what are your comments about Faropenem? Because you wrote an editorial or some update in Indian Pediatrics that, you know, if you're suspecting an ESBL UTI, which is what you would be in this patient because he's been on cotrimoxazole and he has had recurrent UTI, then Faropenem may be a good drug to use.
Yeah, but then I would use it as a definitive, not as an
Absolutely.
empirical.
Absolutely. You would use it once you have, and many times you will find that when you get the culture report, there will be an ESBL pathogen which will be resistant to cefixime, but cefixime would have still done the job because of the concentration in the urinary tract. So your point is very well taken that this is a patient who is not sick. So you should stick to the standard antibiotics, either cefixime or even coamoxiclav. In fact, nowadays people say that with the era of ESBL, coamoxiclav, because of the high concentrations of amoxicillin in the urine, may actually be a good alternative to cefixime here, but you would choose either of these. And what about nitrofurantoin? Because many times when you see the reports of these urine infections, you find that the isolate is sensitive to nitrofurantoin.
So the problem with nitrofurantoin is that it is bacteriostatic and it doesn't achieve a concentration in renal parenchyma. But here, I mean, I don't assume the child to be complicated UTI.
But he's
considering-
febrile.
But
So it means
otherwise,
it's pyelonephritis, yeah.
yeah. So it is not at all a good drug for complicated UTI.
It's for prophylaxis more than.
Yeah, or cystitis or
treatment.
something. Anything else you would like to add here?
Danya, you said that your children are at a higher risk of getting resistant infection. Why so?
So repeated, they are prone for repeated urinary infections.
Yeah, so, and also, of course, in this patient, the other risk factor is he's already on an antibiotic like cotrimoxazole, which could also, now if this patient was not on cotrimoxazole prophylaxis, that would have also been a good option for empiric therapy, because you know a lot of ESBL infections are actually sensitive to cotrimoxazole. Dr. Jagdish, you want to make a point?
Well, I would have also wanted to see whether quinolone should have been added here. Oral quinolone.
The sensitivity pattern to quinolones is very
So
poor.
it depends again on where you're practicing.
Yeah,
There may be
but
some
most
centers
of the
where-
times,
because the concentration of quinolones in the urinary tract is very high, and therefore even if it doesn't work elsewhere, it works pretty well in the urinary tract. So I think we should consider that too before using anything else. So
yeah,
I agree with Dhanya that first choice would be cefixime. Probably if quinolone were there, I would look at quinolone. Ferropenem, of course, would be only after you have a definitive diagnosis. Nitrofurantoin is meant for basically a lower urinary infection. And here
Yes.
it's definitely pyelonephritis. So
Yes.
I don't think we should even venture to try nitrofurantoin.
okay. So I mean, I would choose a beta-lactam antibiotic. And about quinolones, most of the ESBL pathogens are actually now resistant to quinolones, and even you have sensitivity to cefixime and coamoxiclav better than quinolone susceptibility. And even the Indian pediatric guidelines really don't talk about using quinolones below 18 unless you have no other option. So for those reasons, I didn't put a quinolone as one of the choices. Okay, now Dr. Yevle, the same patient, but this is now a different setting. This child is toxic and is not feeding, and he has a fever of 104. He's tachycardic, but his BP is normal, capillary refill is normal. So he's admitted to the hospital. Now in this situation, what antibiotic would you choose now?
So again, the child with UR toxic, but not in shock, has pyelonephritis. And on cotrimoxazole prophylaxis, I would think of an ESBL and would use piperacillin tazobactam since he is not in shock. Had this child been in shock, maybe I would have used meropenem.
So one more question is, why not use ceftriaxone and amikacin? Because amikacin works very well for uropathogens and it works well for ESBL pathogens also. It has good concentrations in the urine, and you also have the option of switching to once a day amikacin when the child is going home after 2-3 days if you have a urine culture which shows a ESBL. And whereas if you start this patient on pip-tazo, then how would you continue treatment for 10 days?
Sometimes these ESBLs are not covered pretty well by amikacin.
That I would not agree, because the sensitivity to amikacin is actually even better than carbapenems. But there is one more hazard of amikacin here in this patient who already has a grade 4
Renal.
UR.
Yeah.
Nephrotoxicity, something.
Yeah. So, I mean, it should not be used mainly because of the nephrotoxicity issue, because you already have a compromised kidney. And if you have other options, you would probably want to avoid using amikacin.
What do you discharge these children on if they are
Yeah.
not willing to stay back in the hospital on the piperacillin tazobactam?
Yes. So, good. So, I would like you only to answer the question. What would you discharge them on?
It's a tough call. I mean, we ask them to stay back in the hospital and complete the course of piperacillin tazobactam. Sometimes the sensitivity shows good sensitivity to co-amoxiclav. But
Yeah, so one more important thing, because we are not discussing definitive treatment here, but when you look at a report of a ESBL pathogen, many times coamoxiclav comes sensitive. But remember, it is not the drug to be used, especially if your ceftriaxone is resistant and your coamoxiclav is sensitive, it may not work because your MICs are just at the break point. So if you are really wanting to treat these patients, definitively you look for cotrimoxazole susceptibility, and pharopenem is another drug which you can consider here. But please remember that there is no way to check the sensitivity to pharopenem. There is no disc available. So you have to go by the company which says that, you know, it works for ESBL, whereas, you know, this drug was approved only in Japan and India, but it has not been approved in European Union and USA. I hope the pharma people are not listening to me here. So that is the downside of using pharopenem. But I mean, you could, once you have got patient has got better and you want to use an oral drug, pharopenem is something which you could use here, but you would watch the patient carefully for a relapse, because studies have shown that when pharopenem was used for ESBL UTIs, there was a 1/3 relapse rate.
In fact, just want to add that pharopenem is aggressively marketed for community-acquired pneumonia, for otitis media, which I think should be strongly discouraged.
Yeah, so if there is one indication for pharopenem, like it's a drug in search of a disease, that is actually a ESBL UTI, which is proven. There could be some role of using it as oral therapy. Okay. So I think with that, let us go to the next case. This case is for you, Dr. Bela. This is an 8-year-old who has fever for past 5 days, abdominal pain, loose stools. It's smelling like enteric fever. The CBC is unremarkable, but CRP is high and SGOT is elevated. And we have not done Vidal. We have not done typhi.IgM because it is not really needed here. But yes, a blood culture is mandatory, which has been sent. But now you want to start an antibiotic pending the results of the blood culture, because the whole thing looks like enteric. So what would be your choice as an outpatient? And
So
I mean,
as
I've
an-
kept the choices a little hard.
Yeah, so for the OPD management, if he is not a sick child and we are not admitting him, then I would like to give cefixime at 20 milligram per kilogram per day for about 10 days. Or the second option would also be an azithromycin, but definitely not the C and D. The irrational combinations of antibiotics I would not use. And pending the reports and how the disease progresses, how the illness progresses, to see whether the child responds to these drugs or whether he has a blood culture which is positive, because we know there is multi-drug resistant S. typhi and paratyphi since many years nowadays, who have been resistant to the conventional drugs. But yet, we do have sensitivity to chloramphenicol and cotrimoxazole sometimes.
But would you like to use that? I mean, would you shift once you get the blood culture report and you've started cefixime or azithromycin, would you change to chloramphenicol or cotrimoxazole?
No, I would not.
You
I would
wouldn't,
not.
na? Because these are
No.
side effects with these drugs. You would
Yes,
use them only if you are having a situation where, you have ceftriaxone resistant enteric fever and you can give cotrimoxazole.
Yes,
You would use
yes.
that. Okay, I specifically want to ask you, how do you choose between cefixime and azithromycin? I mean, you said you would use cefixime first, but we should give it for 2 weeks, not 10 days. 2
Yeah.
weeks is
So
the
azithromycin usually is used for relapsed cases of enteric fever, high dose azithromycin. We reserve it for that.
So you would use it, you would like to reserve azithromycin because you think that it is the drug to go in case we have ceftriaxone resistance. But remember that azithromycin is being used left, right, everywhere.
I don't see any response to it. We get patients who are partially treated outside, who come with, haven't taken these drugs for 5 to 7 days. They all are hospitalized for IV antibiotics. So I do not
And,
really know the community resistance
duration is
pattern.
shorter. So the duration is shorter, and also it's just once a day therapy. And maybe it is cheaper than cefixime. I don't know. I've not done the cost calculation. Dr. Dhanya, you want to say something?
Yeah, the advantage, as you said, is that short duration with azithromycin. But sometimes in some children, obese children, older children, when you have a very, when you're calculating 20 milligram per kg per day, say a 40 kg child. So, I mean, I think the audience should know the ceiling dose of cefixime is 1200, but we get only 200 formulations. So then, I mean, you look at the compliance of the patient also to take that specific number of 3 tablets morning, evening for 14 days. So even that would be considered, I mean, looking at compliance also, but I would go for cefixime, but in
And
certain
keep-
situations,
yeah.
maybe I'll think of it.
Dr. Chinnappa, Dr. Yevle, any other?
I think it's a matter of personal preference. If you look at many pediatricians who are, let's say, above the Vindhyas, they would use cefixime. Below the Vindhyas, they would use azithromycin. So I think it's basically a question of personal
So,
preference. I
so
don't think
let's do an audience poll here. All of you would use cefixime for typhoid fever. Please put up your hands. And how many of you would use azithromycin upfront? Oh my God. Okay, so I think
And
that
how
it
many
is-
would use both cefixime plus
that
azithromycin?
is very common. Okay, so we are in an enlightened audience.
But
Okay.
azithromycin will be BD dose here.
Yes,
Somebody
yes.
said one.
And you have to warn the patient that I'm using a higher dose because this patient goes to the pharmacy. Pharmacy वाला बोलता है आपके डॉक्टर को कुछ पता ही नहीं है इसका डोज 200 twice a day आपको 3 tablets twice a day दिया है तो इसका मतलब the patient loses confidence in you, and one patient actually went on Google and wrote about me that this doctor doesn't know the dose.
No, very often this is a dilemma in clinical practice. Here you said the patient is looking enteric, but very often you get patients who have fever for 5 days without focus. CRP is 40s, 50s. CBC is unremarkable. And SGOT is also, say, in this range. And you are not too sure about your clinical diagnosis of enteric fever. Do you start 20 mg per kg or you would wait for, I mean, what I do in practice, I wait for the culture report and then decide.
No, I would start if I'm thinking this is enteric, and you also know the pattern of fever, that the fever is step ladder. By day 5 is 103, 104.
When it
So
is so clear, there were a dilemma. I said there are grays.
But
where
I would
not
still
so
start
certain.
with the proper dose if I've decided to start. And one important point, that places where there is scrub typhus is prevalent. There, for a illness like this, people may want to start azithromycin because it would work for both enteric and scrub. So if you see in Vellore, what is their guideline for acute febrile illness, they would use azithromycin like here because they would cover scrub as well. Okay, so we quickly go to the last 2 cases. We are, we have 2 minutes apiece. So this is a 9-day-old male neonate born to primary by elective LSCS. Sorry, it should be by birth weight of 3 kg, discharged on day 3 of life. So no risk factors for early onset sepsis. Presents with high fever and irritability on day 5. Sick, toxic look, jittery, normal AF, delayed capillary refill. So we are dealing with severe sepsis and maybe early septic shock. You have done a sepsis screen, you've sent a urine, you've sent blood cultures, and you're planning to do a CSF, but this is like an emergency where you would give an empiric antibiotic. So what would you use in this patient? Maybe, you know, anybody, like any of you could, Dr. Bela, yeah.
So this child actually presented on day 5 of life. So considering it to be early onset septicemia, and then I would like to start, he's about 3 kg. So I could start cefotaxamine with amikacin or piperacillin tazobactam if it is available with amikacin. Unless meningitis is diagnosed, then I could, if I have to step up, then I would
Beha.
go to meropenem.
So you would not start with meropenem upfront.
No,
You would
no,
wait
I
for
would
the
not.
diagnosis
wait.
on the CSF.
Because AF is normal here.
Jittery, I put
He's
jittery word.
jittery, he's sick looking. So considering all early onset septicemia, I would wait for meropenem. I would wait
Okay.
for the CSF and
So option
culture.
3 is definitely not there, but this baby is also in kind of septic shock, early septic shock.
Yeah.
Anybody has a different opinion here?
I would start
with
meropenem,
Meropenem.
given the fact that, see, you have planned a CSF, you do CSF and start meropenem. It's a medical emergency. Irritability.
Neonatal
Normal AF
sepsis.
does not rule out meningitis. Normal AF will not.
I
I would
think
start
Dr.
meropenem.
Mridula is here. Maybe she would give comments or something, but we have a lot of ESBL and, you know,
I
other
would use
pathogens.
meropenem.
Yeah, so I don't know, but
And,
this is a situation. So, I mean, rational antimicrobial therapy is twofold. Like you don't use higher antibiotics when you don't need them. And you also don't use lower antibiotics when you actually need a higher antibiotic.
And piperacillin will not cross the blood-brain barrier.
But doctor said that, you know, she
Cefotax.
would not, she would wait for the CSF, and if the CSF shows meningitis, she would upgrade to meropenem.
Another thing is working in a government hospital where the antibiotics are not available. So our first line would
Actually, you
always
know,
be
this
what
is
is
a very-
available.
I think this question really depends on what kind of patients you are seeing. So if you're working in a community hospital, they would even give Cefotax Amica or Ampigenta here. And if you are actually in a tertiary care center where you see all the time ESBL, carbapenem
I'm a-
resistant pathogens, you are actually, you know, you are stimulated to give a higher antibiotic, because here if we go
Tanu
wrong with the
madam, I'm
antibiotics,
afraid to
we may
interrupt
be
you.
dealing with death or neuromorbidity.
Tanu madam, I'm afraid to interrupt you, but we are just short of time. So last 2 minutes.
Yeah, I'm done. Last case. So a 7-year-old with headache and vomiting for 1 day, terminal neck stiffness. So you thinking of meningitis, you have sent us, done a CBC CRP, you are sent blood cultures, you are done a CSF. Now, what empiric antibiotic or antimicrobial therapy awaiting results? And remember, it's not proven bacterial meningitis. It could be viral meningitis, it could be something else. Yeah, anybody?
Actually, just 1 day history, no fever is mentioned, but there is terminal neck stiffness which suggests meningeal irritation. So considering that, I would definitely start an IV antibiotic, which could be a ceftriaxone. And this could be a non-infectious cause for, high BP with a bleed or a viral etiology. So maybe I would add a cyclovir to begin with.
But it's
pending
not
the
an
reports.
encephalitic presentation.
No,
it's
it
a
is not.
meningitis.
No difference. No, there's no change in mentation, no seizures.
Acyclovir is not
Yeah.
indicated here.
Yeah.
That is
I would-
mostly for encephalopathy.
Yeah.
Yeah, I would defer that. I would start with ceftriaxone and vancomycin if there is any slightest doubt of a bacterial meningitis, because that is now what we practice. And then
Yeah.
DFT.
So the thing is that's why this question, that, you know, here you haven't even proven meningitis, you haven't even proven bacterial meningitis. This could be viral meningitis on the CSF. So there is no harm in this patient to just wait till your CSF routine comes back. And if the routine is suggestive of a bacterial meningitis, I take your point and I want to give a message that you would add vancomycin empirically. But we are talking about a situation where you don't even have not even proven. Okay, I think with that we end. So I would thank my panelists, and final messages for the audience: make a clinical diagnosis, distinguish between bacterial, viral, non-infectious causes. If it's bacterial, think of the various things which we thought, choose the narrowest spectrum drug, and then all of the rest you can read from the slide. Thank you so much.
Thank you so much for this excellent plenary session, ma'am. Now I'll request Dr. Vijay Yavle to felicitate our speaker, Dr. Tanu Singhal. I'll invite Dr. Subhash Kashyap sir on dais to felicitate our chairpersons. Sir, to felicitate Dr. Vijay Yavle. Sir, to felicitate Dr. Bela Verma. Sir, to felicitate Dr. Dhania Dharampal. Kashyape sir, to felicitate Dr. Jagdish Chinappa. Now for this session, I'll invite Secretary of Nasik IIP, Dr. Rishikesh Kute, and President of Nasik IIP, Dr. Kedar Malvatkar, to be on dais to conduct late Dr. G.V. Kopikar's oration. We also request senior pediatrician and our pathbreaker, Dr. Subhash Kashyape, to be on stage, and our Central IP President 2023, Dr. Upendra Kinjavkar, to be on the stage for the oration. I'll request Dr. Rishikesh Kute for the proceedings.
Good morning, everyone. I invite our esteemed guest, Dr. Mrudula Phadke Madam, to grace the dais.
Good morning, everyone. On behalf of Nashik IIP, I welcome all the faculty and all the delegates for the 3-day academic extravaganza. Right now, we will be having Nashik IIP's yearly Dr. G.V. Kopikar Memorial Oration. And we are lucky to have, we feel proud to have none other than Dr. Mrudula Phadke Madam with us to deliver the oration. Apart from graduating from a good medical school, there are 10 aptitudes a good pediatrician should have: knowledge base, clinical skills, competence, continued education, compassion, fearlessness, camaraderie, good humor, respect to fellow colleagues, and the killing instinct to keep people alive. We had one such colleague who obeyed all these commandments. A shining alumnus from St. G.S. Medical College and KEM Hospital, Mumbai, Dr. Gautam Kopikar lived and died holding on to these principles of life. In memoriam, Nashik IIP, Nashik branch of IIP, takes pride in paying tribute to him every year in the form of Dr. Gautam Kopikar Memorial Oration. This year, the oration has been bestowed upon none other than Dr. Mrudula Phadke Madam. I invite Rishikesh to introduce Madam in short, and then we'll proceed with the oration.
Hello, everyone. Dr. Mrudula Phadke Madam needs no introduction, but as a protocol, I will have to introduce Madam. Madam is the ex-dean, BJMC Pune, GMC Mumbai, former Vice Chancellor of MHS. She is the first woman Vice Chancellor, former Director of Medical Education Maharashtra, Senior Advisor, NRHM UNICEF, Independent Director, Serum Institute of India, Visiting Scientist, Hopkins Institute of Training and Research, Senior Technical Advisor, Bill and Melinda Gates Foundation, Member, Global Advisory Committee on Vaccine Safety, WHO, Advisor on Maharashtra State AIDS Control Society, Government of Maharashtra. Madam is a topper and gold medalist throughout her medical education and recipient of President of India's medal. She is the second pediatrician in India to receive honorary fellowship of Royal College of Pediatrician. There is a lot to add to her CV, but even 3 days of conference will not be enough to talk about her. She has done more work and dedicated herself for enough holistic neonatal and pediatric child health. Now we invite Madam to deliver the oration.
Before starting the lecture, we will just respect Dr. Kopikar sir by garlanding his photo.
Honorable chairpersons, my very good colleague, 50 years ago, stalwart of pediatrics in Nashik, Dr. Subhash Kashyape, Dr. Upendra, the president of IAP, who just gave us so much of food for thought that I feel that all of you are full and I don't have to talk anything. But I'm just going to give, may not be food, but tea for thought. Dr. Kedar, President today's, and Dr. Rishikesh. And my dear friends, I don't know, looking at all of you and your youth and wisdom, I feel nearly half in grave. But I'm trying to do a little bit of justice on a talk and a subject which we generally don't talk. We know how to bring down child mortality, and we know we have done so well. We think that India has achieved most of the things. And when I heard you talking on meropenem and whether you're going to give a choice of 3 drugs and oseltamivir and with so much, I thought that, well, we have really done well. And then I opened the newspaper yesterday and I realized that there is a big epidemic of measles in a small slum, Govandi, in Mumbai. And then I thought that maybe we are justified in talking. So in the next 15 minutes, I will tell you, this was the pediatric ward of the past. Tuberculosis, meningitis, polio, diarrhea, it went on. And this is the district hospital today. So what change have we made in 50 years? This is an SNCU brought about with the help of UNICEF and government of Maharashtra. And you can see probably we have done much better. And then on the global scenario, what do we find? I hope there is a pediatric surgeon in this audience. Well, pediatric surgery done on a 530 grammer child, tracheoesophageal fistula, repaired, operated well, and the child is a 7-year healthy child now. This is a WHO world record done by a student from the BJ Medical College, Pune, now based in UK, and probably she has close associations with all of you, and certainly a very close association with Nashik. So this is what we have achieved, the strides that pediatricians and pediatric surgeons have done. And so we see that what have we got opportunities, or we have done all our work? Well, we have to do lots. And it's not only pediatricians, it's not only health professionals, not only medical college faculty, but each one of us has a role. Public health professionals and medical colleges are total at dichotomy today. And therefore, I would think that let us see what opportunities do I have. Besides, certainly antibiotics are important. Certainly a wheezy child nebulization is important. Certainly gut microbiota is really hovering on us. But we have 3-4 simple windows. And I'm going to tell you many such windows which are looking at us, but we are not seeing them. First 1000 days of life, all pediatricians know it by heart that we have to look at it from conception to birth, and then first 2 years of life, that comprises of 1000 days, and nutrition during this period, looking at the health of the mother and then the baby is important. But we wonder what is to be done, because no pediatrician goes and does antenatal pediatrics. I believe some obstetricians are now calling a pediatrician right there. We don't have so much of perinatology in existence. So 1000 days need not remain on paper. We have to see that it's a reality and you do them. And then adolescent girl. Well, adolescent pediatrics is a big branch of IAP. But do we see that an adolescent girl who is going to get married tomorrow is given folic acid by pediatricians? I don't think pediatricians do that. And do we know that does a gynecologist do that? No, because the lady hasn't gone to a gynecologist. So adolescent girl is the biggest window of opportunity for pediatricians today. And we are just not doing it. So let us see what we can go and do step by step. Mortality statistics of the last many years. Under 5 mortality 2022 by SRS survey, 32. Infant mortality 28 per 1,000 and neonatal mortality 20. The betterment is more in terms of neonatal mortality. Thanks to the neonatologists in this audience, and thanks to government and other non-government and corporation colleges who are having state-of-the-art, maybe level 3 and some places level 2. And that is why the reduction in neonatal mortality is 76%. So that was probably the first window which government and non-government organizations and private colleges did it. Look at the SDG goal. We have already achieved it. I mean, by 2030, we have to have 12 as our neonatal mortality per 1,000 live births. Maharashtra is already on 11, but we can achieve and go up to Kerala. So 6 states in India have already achieved the SDG goals. Under 5 mortality, we want it to be 25. 11 states have achieved it, Gujarat included. Certainly Maharashtra is a little over Gujarat and we are on 18. And that's how our mortality statistics. In a nutshell, if I tell you, in 1990, and when I practiced pediatrics, 1 in 7 children or 1 in 10 children died before the age of 5 years. But in 2020, globally, 1 in 30 children is dying before the age of 5 years. So survival is good. But we don't want only survival. We want survival and we want them to thrive. I'm told Dr. Kopikar was one such person who had such a holistic attitude towards pediatrics and an encyclopedia of pediatrics that he wanted that children should not only survive, but they should thrive. And therefore, with greatest pleasure, as Dr. Ramakant Patil, the president, has told me, I am dedicated my whole talk to survival and thrive to late Dr. Kopikar. Now, these are all achievements. Why did we achieve all this? Because there were 101 national programs. As pediatricians, have we seen? I don't have to tell you that we have the facility-based newborn care, we have the newborn care corner, we have the JSSK and NSSK. That is the Janani Shishu Suraksha Karyakram. Do we know that women get free transport? Do we know that they get drugs? Do we know that they are diagnosed? None of these things we think are probably making a dent, but they are there and they're all done by the public health department. What we need as private practitioners is we have to contribute to it. So when we say that we want our mortality statistics to be even better, we have to join hands with these people. The RBSK, Rashtriya Bala Swasthya Karyakram. To my mind, this is one of the best programs. And what does it do? It detects, it has 4 Ds. You detect defects at birth, you detect diseases, you have developmental delays, and you have deficiencies. And under RBSK, all this is done. In my next slide, I'm going to tell you what was the impact of RBSK during COVID time. So this is another national program. Then we have the district early intervention centers, HBNC, HBYC, all home-based cares, IYCF. The most important, Dr. Upendra told us so much about breastfeeding, infant and young child feeding, Ma project and mother's absolute affection, known so well after Madhuri Dixit, and the IMNCI, old-timers FIMNCI. We did something to detect neonatal illnesses, and the NRCs established, the nutrition rehabilitation centers established. IAP did a lot of work. You have the continuous NALS and PALS, and INAP, India Newborn Action Plan, and Kangaroo Mother Care has taken a headway. All this is helping us. But now let's see why it has not helped us so well. Pradhan Mantri Matritva Yojana is there, and APJ Abdul Kalam Amrut Ahar Yojana is there. You are giving good food to the pregnant mothers. We have Anemia Mukta Bharat also. But when I say this scheme, can I tell you that 70% of Indian mothers and children are anemic? If that's our statistics, then you can understand the dichotomy. The problem is that the scheme is there, but it is not coming to us and it is not going down to the patients. So if we have 70% anemia, I don't know from where we'll begin. And then we have one good scheme, which the credit goes to everyone, the pediatricians and the private practitioners and also the government. Mission Indradhanush, immunizing 7 vaccine preventable diseases. We are doing well in that, but we realize that the second dose measles coverage at 16 months is just 24%. And why did that happen? Maybe there are many reasons what it has happened. And of course, the rubella rota and the pneumonia added now, only 8% coverage. I would like that pediatricians put a special stress. And so this is my first window of opportunity that we have, and that is to improve immunization coverage. Have the pediatricians done that? The medical colleges don't report to the health services. They are standing in 2 solo buildings, which are absolutely in silos. And so the health services have no idea how much is the coverage done by us. And many pediatricians don't even give immunization advice in several medical colleges to the effect that you want it. So I don't know if newer modalities of immunization practices have come into trends, and therefore we saw that in slums of Mumbai just 2 days ago, there was a measles epidemic and government declared that 17,000 children in Mumbai remain unimmunized today. They have not received a single dose of measles vaccine. So if that's our scenario, and now it has spread not only to Govandi, but to other slums around, it has gone to Bhivandi. And also I'm told that yesterday Malegaon has had many cases of measles. So this would be the first window of opportunity. What are the second and the third windows, or what other things can we do? We have been battling COVID for the last 3 years. I mean, I'm seeing such a face-to-face conference, and I'm really feeling happy because all of us were indoors. The mortality and morbidity of COVID was so enormous. The lockdowns, the joblessness, the supply chain disruptions, inflation, all this has had a long-lasting impact on the population. Fortunately, third wave has waned and we are in a much better situation. But the impact of COVID is going to trouble us. And why is it going to trouble us? The collateral damage, the silent food crisis that COVID has produced. Do you know that 100 million Indians are vulnerable for the food crisis? 52 million children are born in COVID times, and women and children are most vulnerable. We have gone, 30% of people have gone out of their savings. Did we ever think that a 10% loss in the salary, when job losses occur, the thing that decreases first is that there is a 1% decrease in the protein content in the diet and 0.8% in the carbs. And nutrient-rich foods, we can say give all this in your plate, but they have become 10 times costlier. And staple food grains and vegetables, we just can't afford them. So if that's our scenario, Lancet very surely has very rightly said in 2020 that after COVID, there's going to be a 14% increase in wasting. And India's global hunger index yesterday, everyone talked 107. We can't praise ourselves. We are lower than Bangladesh and Pakistan. Politics aside, but this has to be taken an eye-opener for all of us. And as pediatricians, we can contribute to it. COVID did impact on this. Our services for IYCF, our services for ICDS, our midday meal schemes, adolescent and anemia Mukta Bharat, everything had gone down, had come to a standstill. And Purnima Menon had done a wonderful survey, and she said that compounding this has also impacted. And in addition to that, we had the food insecurity, and all this bothered us. Now, what medical impact did COVID have? Lots. On children, a direct impact and an indirect impact. Fortunately, children were spared, but still the direct impact of COVID was that the nutrition got affected, because if you had COVID, appetite was lost, there was no taste, there was vomiting, there was diarrhea. Some people also saw cases like Missy, particularly Dr. Prabhu from Wadia Hospital, Mumbai, had recorded some cases of Missy. Dr. Kinikar showed that SAM, severe acute malnutrition children, and anemia had increased. So that was the direct effect of COVID. They published it. But from another hospital in Pune, Dr. Lalwani, he found that there was no such effect. Now, this happened only because the two areas are drained differently, and probably in Bharati Vidyapith, better socioeconomic status people come. Because of COVID, we saw that healthy children went into severe acute malnutrition and moderate acute malnutrition. Anemia increased, but this is only the direct effect. I want to bring to your attention the indirect effect of COVID, and that is the collateral damage, food insecurity, girl children ignored, girl children left our school, our Bhamabai and Bhimabai, our domestic workers' children, they were married off. In Parbani, today I met some pediatrician from Parbani, one of my students, and I was told that particularly in areas where there was no internet and no teaching going on, people just got their children married at 13 and 14 years. Girls got married. And I bring to you an important data from RBSK, one of the national programs that I mentioned to you 5 minutes ago, the Rashtriya Bal Swasthya Karyakram. This data got published, the credit of which goes certainly to UNICEF, and Raj Lakshmi Nair madam is sitting here. 21 lakh children are examined by doctors under the RBSK in Maharashtra. And what did they find after COVID? That 112 children were born with neural tube defects. What do we mean by that? What is the result of this? 4 times higher than what it was in 2020. Now, why did children get born with neural defects? Similarly, a similar data on these 21 lakh children showed that severe acute malnutrition, SAM, had increased 5 times. Why did all this happen? Simple enough. Girls got married in COVID times, no folic acid available. Nobody gave folic acid to the children. And in BMJ Global Health, this has been published that obviously excess of neural tube defects that occurred were a collateral damage of COVID. And we saw it through our RBSK detection. Students and friends, we have to make a note of this and we have to see that we take care of it. COVID and pregnancy, immense effects. Mortality rates of mothers in COVID times was 20 times or 22 times more. Fetal distress more, lots of prematurity, birth weight was low, and PIH in mothers had increased. So this was the effect of COVID on children. A good JAMA article in 2021 showed, and it was one of the best studies, inter-COVID study of Oxford. But we were no better off. Dr. Malik from Nair Hospital, Sushma Malik, showed a similar study on 925 children published. And we know that lots of premature and low birth weight babies were born. So if so much of low birth weight was born, then we should know what happened. COVID has nearly gone down. XBB strain, Omicron strain is now not such a worry. All of us are moving freely with masks. But the second window of opportunity that we have, and that is the nutrition of children who suffered, the low birth weight children, all these that we have to bother. And that's the second window of opportunity lies with us. I don't want that after 2 years, again, we say that 112 and 114 children with neural tube defects were born because nobody looked at these girls. Adolescent clinics were closed. So what happened? Let's look at the data from NFHS 5 study. National Family Health Survey study showed that stunting 35%, wasting 25%. I hope all of you know the new terminology. We don't call malnutrition as kwashiorkor and marasmus now. We say stunting, wasting, and underweight. And 36, so 1/3 of our population, the child population, is still stunted. So much is wasted and severe wasting. What we are really worried about, the old kwashiorkors and marasmus is 10%. If that is so, and with NFHS 5 data showing this, that it's worse than NFHS 4, and COVID added means it's even more, and 30% of the babies are born low birth weight. So all this is having a compounding effect. And therefore, if we don't look at the window number 3, and that is to look at the children who are going to get malnourished, particularly the low birth weight babies born, then we have to be worried. I also give a word of caution, like a disclaimer, data from government does not show what NFHS 5 data and statistics shows. And we all have also witnessed all this. But somehow central government data says that SAM was only less than 2% in a study done in Delhi on 1 crore babies. This is unpublished data, came only in newspapers. But therefore, Government of India made a good statement for which I was very happy 5 months ago. They said that they are going to involve the Indian Academy of Pediatrics to validate whether nutrition and the scenario of SAM in the country is just 2% or 10% and 35%. I suppose trustworthy data needs to come. Now, one thing that all of us have never seen, neither the government nor the private practitioners, and that is how is severe acute malnutrition in infants below 6 months. You know that so many low birth weight babies are born, 1 in 3. Then we have the 4th window of opportunity. Look at these children. Can you detect SAM in children below 6 months? I think I can't. How do we detect severe acute malnutrition? We do it by taking weight for length. And how do we detect stunting? We do it by taking height for age. And how do we detect underweight? By taking the weight of the child and with the corresponding age for the child. But all these conventional methods are associated with difficulties in children under 6 months. Why? Because the WHO growth charts, if all of you have seen them, they don't have a length of less than 45 centimeters. So in a newborn who is born a little preterm or who is an SGA, you don't have on the chart what to plot because there is no reading below 45 centimeters. Preterm low birth weight babies have difficulty in putting their knees are flexed, and so you can't make them straight, and therefore you have difficulty in bent knees, and particularly in communities and where you don't have infantometers, all this So detecting SAM under 6 months, we have never thought of it. And if you don't treat them, that is the cause of mortality in infants. And therefore, I would insist that let us weigh our children at birth, let us take their lengths and mark them and plot them. We have given you a lot of WHO growth charts, but nobody is doing that. So we have to see that it is done. Now, what is the research evidence that we have? Wait for age. People in communities find it very difficult to take the length. So Dr. Praveen Kumar from Delhi, in Journal of Pediatrics, he published that they want to take just weight for age. And they think that weight for length is not a very good predictor of mortality. What is good is you just take weight for age. And what we used to do 30 years ago, that is growth faltering. Just monitor the growth of the child, take the weight of the child, and that's done. Wadia Hospital in Mumbai also has done a similar study. Dr. Sanjay Prabhu and Professor Irani Madam, probably they found similar results. Ethiopia, Malawi also thought that such a thing is probably taking only weight is good. But WHO and MAMI, they both say that they are not in agreement, and many of us are not in agreement. All of us feel that weight and length of the child is very important. So even in the first 6 months, let us think, are we doing that? And therefore, I bring to you one index, what is called as the Composite Index of Anthropometric Failure. This is called as CIF. Doing it is very simple. It is a good indicator of mortality and morbidity. And Swedberg, an economist, imagine an economist is telling pediatricians that you should do Composite Index of Anthropometric Failure. And it is a very good mathematical index. You can do it. People are doing it in the Jidao mission at the government of Maharashtra. And so I would recommend at least give a thought and try and use the WHO growth charts. Try and see children below 6 months and see that am I detecting a child of SAM? And if I'm not, then all my low birth weight babies are just going without any treatment. So we come back to the next window, the concept of 1000 days and early childhood development. Yesterday, Dr. Ramakant Patil and all the organizers had a wonderful workshop on early childhood development. And that is the most important window of opportunity for us. So period from conception to 2 years of life, one of the critical windows. And in that window, does a pediatrician have a role or an obstetrician have a role? Well, it is a combined role, dual role. But I will come to what is called as preconception counseling. Who has to do it? An obstetrician has to do it or a pediatrician? Because the lady who is in your clinic when she's an adolescent girl, can you put up a chart in your adolescent clinic and share it with your obstetric colleagues that I will advise every adolescent girl, well, don't think of getting married before you are 18 years. If you are a careerist, then don't think of getting married maybe even up to 25. You see that you have achieved a weight of 40, at least 40 kilos, no need of having a zero-sized figure. See that your BMR is above 18.5 and the mid-upper arm circumference, at least in India, may not be 23, but 21 centimeters. Have we ever done that in the adolescent clinic? None of us have done it. And if we don't have the time, let us at least put a board so all the 13 and 14-year girls who are coming to you will remember it. There would be no consanguinity and no consanguineous marriage. Have you taken a rubella vaccine? Government has started a program on measles and rubella coverage even at 15 years, the MR dose. But pediatricians, we have not gone, and up to 4 to 6 years is only the dose that we are giving, which is also missed, less than 20% coverage. No tobacco, no alcohol, no drugs, and proper spacing. All this advice must come from the adolescent clinic of a pediatrician and go on to the obstetrician. I think this is the 5th window of opportunity, but we are not availing of it. What is the 6th window? Is the nurturing care concept, the early childhood development, a holistic care, nurturing care of a child. Look at the physical, motor, socio-emotional, and cognitive and linguistic development of a child. And that is done by interaction, and your own continued stimulation, not only by the parents and the caregivers, but a pediatrician's impact on the whole thing. And look at ECD in 5 domains: the health and nutrition you're going to look at, the safety and security of the child, responsive caregiving, and early learning. Yesterday, there was a wonderful talk on responsive caregiving and early learning. I hope all the pediatricians, I'm sure, are going to get it from Dr. Samir Dalwai, Dr. Uday Bodhankar, and Dr. Anand Deshpande, not to forget our own UNICEF's Professor Pramila Menon and Raji Nair Madam. So this ECD has to be practiced by all of us. It's not a simple thing. That way it's a simple thing, but you have to remember that 5 domains and the nurturing care is important. Practice it at all levels. Do the undergraduates today know of ECG? They don't know of ECD. Do the undergraduates know of how we have to do it in an immunization clinic, how we have to do it in a well baby clinic? So I would think that there are lots of people from the Board of Studies of Pediatrics, from medical colleges. Why don't we include ECD in our curriculum? So if there is a small question on early childhood development, people know how to practice it, people know how to do it, and they know how to write it because it comes in the exams. I have already spoken to 2-3 deans, and we are in a city where the Maharashtra University of Health Science is there. Honorable Vice Chancellor is there. Let us make an appeal to her that ECD must come along with IYCF in our curriculum at the MBBS level. I would say about 1,000 days. Now, what do we say on 1,000 days? Everyone knows it is important, but nobody knows that we have to give folic acid. And nobody thinks that folic acid is paradoxical. So let me tell you how folic acid is to be given. Folic acid for primary prevention of neural tube defects, what we have learned 3 months before conception and 3 months after conception, that is the periconceptional period. And on that time, at that time, you have to give 400 micrograms of folic acid to be given to the woman before she conceives. So even before she is getting married, you have to think. So before she goes for her honeymoon, she has to carry folic acid tablets. And this 400 microgram folic acid is difficult to get. The only folic acid that we have is 5 milligrams of folic acid tablet. So the pure folic acid tablet is 5 milligrams. The ideal dose of folic acid for primary prevention is 400 micrograms. What is the dose of 5 milligrams? When do you give it? Folic acid for secondary prevention. That means if a woman has had a previous neural tube defect and you want to prevent it, then you are going to give 5 milligrams of folic acid. So 2 doses of folic acid. One is primary prevention, 400 micrograms. And second one is secondary prevention. That is, you have to give 5 milligrams. The standard folic acid that comes to us under the government program in ANC to be given for 180 days is iron folic acid or FIFA tablet or whatever they call it, FA tablet. The iron folic acid tablet contains 400 micrograms of folic acid, which is given generally from the 2nd trimester onwards to the mother for 180 days. In some scenarios, in some countries, particularly in the UK and even higher in the US, the dose of folic acid in the antenatal given tablet to the mother is 600 micrograms and 800 micrograms in the US. But we have to remember what happens in India. If we give more and more folic acid, say we give 1 milligram of folic acid, Dr. Chittaranjan Yadnik from KM Hospital, Pune has done a lot of work on it. And he says that most Indian mothers are deficient in vitamin B12. And therefore, if you give folic acid, the neurological effects are increased in the absence of B12. So women who are deficient in B12, you have to give both folic acid together with vitamin B12. And this is what is required. Now, an important study in Journal of Maternal and Child Nutrition in January 2020, one lady has done an excellent meta-analysis And this Jonker lady says that folic acid given in the periconceptional period increases the birth weight of the baby. So look at the opportunity before us. We just go on talking of neonatologists and we just go on saying, take this low birth weight baby, take this low birth weight baby. But have we ever seen that if you had given periconceptional folic acid, which probably is not available here because we don't have 400 micrograms of folic acid. And so the birth weight has not increased. We have two small areas when the baby's birth weight increases to maximum. First is in the first trimester from conception to 3 months. And the second increase is in the last trimester, which we have all along focused. And we think that at that time, it's important. I'm not saying last trimester is not important. Last trimester, the weight increases maximum of the baby, but first trimester is also important, which we have missed. And therefore, folic acid given here, the birth weight has been found to increase by 13%. Simple, since we don't have only folic acid, at least periconceptionally, let us give multiple micronutrient supplement. Some studies are underway, not only in Western countries, but also in India. And one was done in Pate and Sinnar in Nashik district itself. And multiple micronutrients are given to the mother, so our MV tablet. And they found that birth weight had increased. Good to know that this tablet has 400 micrograms of folic acid and 2.6 micrograms of vitamin B12. So good for us. We can think about it, but it's not being done. And so I bring you to some more newer concepts. And all of us know that folic acid is important. When to give, and the paradox is still to be resolved, but we'll try to resolve it. And therefore, you should know what is DOHAD concept. DOHAD concept is a concept, developmental origins of health and disease. And Lancet 2018, one Dr. Fleming, not the Fleming of penicillin, but Dr. Fleming found out the relative contribution of maternal and paternal factors in the formation of the ovum and the embryo. And he says that periconceptional developmental conditioning, he's done a lot of experiments on mice, humans, and the IVF fertilized. Day before yesterday, someone told me that because IVF is increasing, lots of preterm babies are born. And this Dr. Fleming experimented on IVF embryos, and he found out that maternal undernutrition has a great impact on it. And so if the mother is on a low protein diet when she is honeymooning, and we also know the history of Dutch famine, we know Gambia famine, and we know that it is the last 3 days of oocyte maturation, and the embryo at implantation is very important, but we have never looked at it. And so when the mother is probably in her in-laws' place, just married woman, nobody is looking at her protein diet. And if the mother is on a low protein diet by whatever, because she can't afford, then what happens? So this Dr. Fleming found out that low protein diet in the mother, at the time when ovum is getting formed, the insulin is low in her, branch chain amino acids are low, Graafian follicle that is formed and the blastocyst which is coming into the uterus, all of them have very less amino acids. The uterine fluid is deprived of amino acids. And what is the result? Poor implantation, poor placenta, poor fetal growth trajectory, and low birth weight. So we know nobody needs to tell us that the mother has to have low protein, high protein diet. But we now know what the low protein diet is producing on the formation of the placenta. But we have never looked at it. So see how protein is important. And this effect is not so much when you give proteins late in the gestational period. So last trimester, even first trimester or conception is more important. DNA methylation is important on proteins, and paternal factors, which are less clear, we have not looked at them. But this Dr. Fleming found out father's lifestyle, father's obesity decreases the sperm quality and DNA integrity. So we have to think about all this. Then a word on maternal obesity. Obese women, as we know that they have high inflammatory cytokines, they have a lot of blood glucose, low insulin levels, more lactates, more triglycerides, leptin, and all these accumulate around the oocyte, and they hamper the maturation of the oocyte. And chromosomal aberrations can occur. So this is the biggest window of opportunity that we have. And look at these low birth weight babies who become later on in life obese like us. And then they have a higher chance of coronary artery disease, central obesity, diabetes, hypertension, hyperlipidemia, and the intergenerational cycle continues. Mother who is poor in diet, who is low in weight, low birth weight baby is produced, that one again conceives and she becomes low birth weight and then becomes with coronary artery disease. So all this is going on, and the concept of it is the DOHAD concept. Now, after having seen that seventh window, let us come to IYCF. The baby is born. Let's say that the baby is born good in weight, and we have infant and young child good feeding, breastfeeding within 1 hour of birth, exclusive breastfeeding for 6 months, good complementary feeding, adequate diet. We haven't seen dietary diversity. BPNI, IAP, FOXY are making their level best efforts. And let us see that this is not only taught, practiced, but it is included at all levels. Breastfeeding within first hour of birth, 53% in India, and exclusive one is even worse. And only 8% children have good complementary feed in 6 to 23 months. So where are we? We have a big window in terms of all this, window number 7 and window number 8. IYCF practices good and good complementary feeding. Have we seen that there is a dietary diversity in our diets, presuming that there is enough money? Have we seen, do we know about child food poverty? We only know of one poverty, and that is poverty in terms of money, or we may know at the terms of poverty in terms of knowledge, but we have to know child food poverty. And this means that the child is not fed minimum diverse diet, a terminology coined by Victor Agallo from UNICEF. We have not 7, but 8 food groups. To the original ones, we have added the 8th one, that is breast milk. Second one, grains and tubers, legumes, nuts, fish, this, that, dairy. But 8 food groups are there. And you will say that who is a child who has food poverty? If he is consuming less than 2. So if you are having only varan bhat, imagine that you have got only 2 food components, and therefore with 2 food groups, you have severe poverty. If you have 5 groups, food groups, then you have no food poverty, and moderate poverty, 3 to 4 food groups. So have we seen that our programs are directed to our complementary feeding? No. We still think that Varna Chapani and Bhata Chapani is ideal, but I would really see that you attract your attention on child food poverty and see that the child consumes so many food groups. A word on double burden of malnutrition. Double burden is, on one side you have undernutrition, stunting, wasting, underweight, and on the other side you have overweight and obesity. Both exist in the same population, both exist sometimes in the same individual, and we don't want, IAP should see that this poster goes out of India, and we never do we want a child like this in our country at least. And then we have triple burden to both obesity and undernutrition. You add the third component, and that is of hidden hunger or micronutrient deficiencies. Most important of the micronutrient deficiencies that we have to remember is anemia. Zinc is important, selenium is important, copper is important, all are important, but we can focus on number 1, anemia, and number 2, vitamin D deficiency and A deficiency. This chart is showing you how much India lacks in multiple micronutrient deficiencies. India is nearly the highest. So we don't have to feel proud about it. It is highest in children, it is highest in mothers who are 15 to 49 years of age. So we want to prevent that. And NFHS 5 data on anemia, below 5 years, 68%. Pregnant women are also 50% anemic. So this is the reality. We are not getting iron, folic acid. So again, we have to bring back on our focus on anemia. And therefore, I would give and say that unavailability of iron tablets, girls not taking them, vomiting them, gastric disturbance, nothing. Strengthen the anemia Mukta Bharat concept, marriage and periconceptional folic acid to prevent neural tube defect. That would be our plan of action. And then we come to the last window, window number 10, and that I would like to give. That is one good thought that all pediatricians can carry with them. We are convinced that first 3 months of pregnancy are as important as the last 3 months. We are convinced that we don't want to give low protein diet even when the Graafian follicle is getting formed, when the ovum is getting formed. And now I bring to the new concept, what is called as the concept of IGF-2 or insulin-like growth factor 2. Many of us know this insulin-like growth factor because it is involved in tumors and all that. And insulin-like growth factor 1 is responsible for growth in adolescent age and adults. But insulin-like growth factor is important for the growth in the fetal life. And there is a gene for it. And the gene is called as IGF-2 and IGF-2 receptor. And the ability of these genes, or the funny thing of these genes, is that these genes are imprinted. What do we mean by imprinted gene? That means the gene is expressed only, not both genes are expected, the one from the mother's side and the one from the father's side, not both genes are expressed. When a gene is imprinted, only one of them gets expressed. And this IGF-2, the beauty of it is expressed only for the one that comes from the father's side. So there is a paternal expression of IGF-2. And from the mother's side, the expression is negligible. So this gene expression, and what does the father's side IGF-2 do? It makes the baby nice, good in weight. And the mother's side doesn't do it so well. And this gene expression, as we know, depends on methylation. And methylation depends on folic acid. So I give you one very important concept. What is the neuroscience news? A good concept given by Cambridge University, author called Sandovici and Miguel in 2021. They have given this hypothesis and they have proven it in experiments and in humans. And they say that paternal IGF-2 gene in the fetus is responsible for the growth of placental blood vessels. So when paternal IGF-2 is active, the placental blood vessels are becoming big and they are growing. Whereas mother's IGF-2 makes the placenta smaller, it makes the placenta thinner, and the blood vessels also become less. So if the mother's IGF-2 is active, what happens? The baby becomes small in weight. If the paternal IGF-2 is big and active, the baby becomes big. So imagine the tussle between males and females is going on even before birth, and that is IGF-2. And this intrauterine ladai that is going on is because of the insulin-like growth factor. And this happens, when does this happen? IGF-2 activity, it happens when there is a failure of imprinting. And imprinting, as we all know, depends on methylation. And methylation is dependent on folic acid. So this epigenetic battle, it is in the hands of the pediatricians. And it is in the hands of all of us to stop the tussle. So we stop the tussle by giving good epigenetic counseling to other mothers. First of all, by knowing that there is a paternal IGF-2 and there is knowing a maternal IGF-2. And we want the gene to be imprinted. And imprinting is done only by methylation. And methylation occurs by folic acid. So I only want to give small 10 windows of opportunities. And my 10th window of opportunity is the food for thought for our president, Dr. Kinjodekar, who is working on gut-brain axis. May you all, with all pediatricians, find out whether we can have good imprinting and how our mothers can take folic acid before conception. So with these few words, I think I have already crossed the time. So I will not take on further slides, but I thank the audience, I thank the organizers and all the pediatricians for thanking me. Thank you very much. Thank you so much.
I'll request now.
Thank you.
Request Dr. Kedar Malvatkar,
Thank you
sir.
so much. Thank you.
I'll request Dr. Kedar Malvatkar, sir, to read the citation. Madam, please. Read the citation now.
I invite
And
our
also invite
National President, Honorable Dr. Ramesh Kumar sir, to be with us while we are honoring Madam. I also invite Rahul Kopikar, son of Dr. Gautam Kopikar, to be with us. Before handing over the citation and felicitating Madam, I would like to just read out the citation.
I invite Dr. R.D. Patil, sir, organizing chairperson of MAHAPEDICON, and Secretary Dr. Milind Bharati.
इंडियन अकॅडमी ऑफ पेडियाट्रिक्स नाशिक शाखेच्या वतीने 2022च्या वार्षिक अधिवेशनाप्रसंगी डॉक्टर गौतम कोपीकर सन्मानपत्र आपणास प्रदान करण्यास आम्हाला अत्यंत आनंद होत आहे. उर्दूमध्ये सय्यद हसमत सुहेल यांच्या 2 छान ओळी आहेत. मिटा दे अपनी हस्ती को अगर कुछ मर्तबा चाहिए कि दाना खाक के मिलकर गले गुलजार होता है. अर्थात आपल्याला फुलांनी बहरलेली बाग दिसते, पण त्यामागे एका बीजाने जमिनीत गाडून घेऊन कठोर तपश्चर्या केलेली असते. महोदया, आपण आपली ही असंख्य यशांनी बहरलेली बाग बघताना त्यामागे आपली कठोर तपश्चर्या, अत्यंत शिस्तप्रिय वृत्ती, प्रखर बुद्धिमत्ता आणि प्रचंड उत्साह अशा अनेक असामान्य गुणांचे बीजांकुर आपण वाढवले आहे. यात काहीच अतिशयोक्ती नाही. वैद्यकीय महाविद्यालयात प्रशासक, नंतर अधिष्ठाता, तदनंतर वैद्यकीय शिक्षण संशोधन विभागाचे संचालक, महाराष्ट्र आरोग्य विद्यापीठाच्या कुलगुरू आणि निवृत्तीनंतरही WHO, युनिसेफ, बिल गेट्स फाउंडेशन अशा अनेक उत्तुंग आंतरराष्ट्रीय संस्थेत आपण सल्लागार म्हणून कार्यरत आहात. या सर्व पदावर काम करताना आपण आपल्या विशिष्ट गुणांचा वापर करीत एक इतिहास निर्माण केला आहे. यात काहीच वाद नाही. आपल्या दूरदृष्टी आणि विकासाचा ध्यास या वृत्तीने आपल्या कारकिर्दीत आपण लातूर, कोल्हापूर, अकोला येथे वैद्यकीय महाविद्यालय सुरू करण्यात मोलाचा वाटा उचलला आहे. आपल्या न संपणाऱ्या असंख्य पुरस्काराच्या यादीत डॉक्टर BN पुरंदरे उत्कृष्ट सेवा पुरस्कार, डॉक्टर शिरोडकर उत्कृष्ट सेवा पुरस्कार, एड्स या आजारात संशोधन करून आंतरराष्ट्रीय पुरस्कार, आपल्या शैक्षणिक योगदानाकरता अत्यंत ख्यातीच्या केम्ब्रिज युनिव्हर्सिटीच्या शैक्षणिक पुरस्कार, हे काही निवडक महत्त्वाचे सन्मान आपणास प्राप्त झाले आहोत याचे आम्ही साक्षीदार आहोत. आपणास अमेरिकेत जॉन हॉपकिन्स इन्स्टिट्यूटचे, तसेच जर्मनी, लिबिया, रशिया, थायलंड, सिंगापूर या अनेक देशात व्हिजिटिंग प्रोफेसर म्हणून सन्मानाने निमंत्रित करण्यात आले आहे. आपण कुपोषण, हेमॅटॉलॉजी, जेनेटिक्स, HIV, एड्स, संसर्गजन्य आजार या अनेक विविधांगी विषयात केलेले संशोधन आजही आम्हाला दीपस्तंभासारखे मार्गदर्शन करते. एवढ्या उत्तुंग यशानंतर येणारा अहंकाराचा दर्प हा मानवी स्थायीभाव, पण या अहंकाराचा किंचितही लवलेश आपल्या बोलण्यात, वागण्यात आणि कृतीत दिसला नाही. इतरांना सातत्याने मदत करण्याची स्थायी वृत्ती, अत्यंत विनम्र स्वभाव व आपल्या नावाला साजेशी अत्यंत मृदुवाणी, यामुळे आपण केवळ विद्यार्थी आणि डॉक्टरांमध्येच नाही तर सर्व क्षेत्रात लोकप्रिय आणि आदर्श आहात. आपल्यातील हाडाचा शिक्षक, कुशल प्रशासक, आपल्यातील उत्कृष्ट वक्तृत्व शैली, कुठल्याही क्षेत्रात समरसपणे व समर्थ भावाने काम करण्याची वृत्ती, कठोर परिश्रम करण्याची तयारी, संशोधक वृत्ती आणि बाल आरोग्याकरिता आपण केलेले योगदान या सर्वाचा विचार करून अत्यंत सविनयपूर्वक हे सन्मानपत्र आपणास प्रदान करण्यास आम्हाला आनंद होत आहे.
I request Dr. Hemant Gangolia, sir, Maharashtra IAP President, to come on the dais while we are felicitating Mrudula Phadke, madam. Please, sir. Dr. Hemant Gangulia also will join the team. Thank you, sir. Thank you for understanding us. Side. शब्दांकन हे डॉक्टर शेखर दाभाडकरांनी केलेलं आहे. Thank you, sir. It's a lovely compilation. Really appreciate your words. Thank you, sir.
एक घरगुती पदार्पण सांगतो. काल सकाळी मॅडम ECD वर्कशॉपला निघाल्या. सकाळी 5 वाजता उठून स्वयंपाक करून स्वतःचा डबा बरोबर घेऊन आल्या. कॅन यू बीट इट? मगाशी बोलता बोलता म्हणाले काहीतरी दे बिकम ओबीज लाईक अवर्स. आम्ही म्हणजे ओबीज लाईक अवर्स म्हटलं. मॅडमनी असं म्हणणं म्हणजे वॉज अ वंडरफुल जोक.
His finishes are Dr. Kopikar oration. A big thanks to all the dignitaries who have come onto the stage, given their time. We are really lucky to witness this big oration. Thank you, ma'am. Now we will move on to the next program, that is inauguration of our MAHAPEDICON 2022. I hand over my mic to our two MOCs, Dr. Prachi Birari and Dr. Shalaka Bagul. Over to you, Prachi.
Thank you so much, Dr. Sheetal Pagar and Dr. Sheetal Mogal. Thank you so much.
Thank you, Sheetal.
Ready.
By Dr. Vijay Yavle. All the delegates, please pay attention. Finding a needle in a haystack. This session will start in Tapovan Hall, sharp at 12:30. At 12:30, sharp, we are starting the session, Finding a Needle in Haystack, the microbiological tests. Speakers for the same session would be Dr. Tanu Singhal, Dr. Sanjay Nathu, Dr. Camilla Rodriguez, Dr. Suhas Prabhu, and our very own Dr. Vijay Yavle would be moderating the session.
We are shortly starting with the inauguration program. I request everyone to please be seated.
I request all our delegates to be seated. We're soon starting with the inaugural function. Request to all our delegates to please get seated. Ladies, we can have that photo session later on. Please get seated. We have an excellent lush greenery outside the hall. Photos can be clicked there.
I request everyone to please be seated. We are soon starting with the inauguration program. I request everyone to please be seated. Once again, a very good afternoon, everyone. As we proceed towards the inauguration function, we welcome you all to the land of saints, social equality, fraternity, and wisdom, a state which is progressive and full of creativity.
We, Nasik IIPians, extend our warm greetings and welcome you all to this abode of cultural heritage and values, yet a positive and a modern city, Nasik. I request Dr. Amol Pawar, Honorary Secretary General, Maharashtra IIP 2022, to come on the dais.
Dear dignitaries and friends, please be seated. Please be seated. On the right-hand corner, dear delegates, be seated. Organizing team members, please be seated. Good morning, everyone. Dear all friends and delegates, now we'll start with the most awaited event, that is inauguration ceremony of this MAHAPEDICON 2022. As all dignitaries have arrived in the hall, so I request MOCs to conduct the proceedings now. Thank you.
Let us commence this inaugural ceremony by escorting our esteemed dignitaries to the dais. I request Dr. Shyam Chaudhary to escort Dr. Hemant Gangolia, President MAHA-IIP 2022.
Ladies and gentlemen, we are privileged and blessed to have the teacher of teachers, Dr. Mrudula Phadke, as our chief guest of honor for today's function. She has been the ex-dean of BJ Medical College, Pune, ex-director of medical education and research. She has also been the vice chancellor of MUHS and continues to be the chief advisor to UNICEF and WHO. May I please request Dr. Sharmila Kulkarni to please escort Dr. Mrudula Phadke, ma'am.
I request Dr. Sushil Parak to escort Dr. Ramesh Kumar Sir, Central IIP President 2022.
I now request Dr. Praful Patel to please escort our chief guest, Dr. Upendra Kinjwarekar Sir, President-elect CIIP 2023 on the stage.
I request Dr. Devi to escort Dr. G.V. Basavaraja Sir, President-elect Central IIP 2024.
I now request Dr. Ravindra Sonone to please escort Dr. Chetan Shah, Vice President, West Zone, CIIP 2022.
May I now request Dr. Sagar Sonone to escort Dr. Samir Dalwai, Treasurer, Central IIP 2022.
I now invite Dr. Ramakant Patil, Organizing Chairperson of MAHAPEDICON 2022, to please step up the dais.
I request Dr. Amol Pawar, Honorary Secretary General, Central Maharashtra IIP, to be there on the dais.
I also request Dr. Milind Bharadia, Chief Organizing Secretary and founder of Nashikon, to please come on stage. We are thankful to all the dignitaries on stage for sparing this valuable time and gracing this occasion.
To express our gratitude towards all the dignitaries, we will felicitate them by the hands of patrons of Nasik IIP. These patrons are the pillars of Nasik IIP who have worked relentlessly, and their blessings have taken Nasik IIP to a greater height. So I request the senior most pediatrician from Nashik, Dr. J.S. Patel Sir, to felicitate Dr. Hemant Gangolia Sir. Dr. Tanaji Kanade Sir will felicitate Ramesh Kumar Sir.
I now request Dr. Subhash Kashyap sir to please felicitate Dr. Upendra Kinjwarikar. I now request Dr. Sadhna Patil and Dr. Vaishali Bharadia to please come up on stage and felicitate Dr. Murdula Phadke ma'am.
Next on the list is Dr. Abhay Tirak sir, who is going to felicitate Dr. Basavaraja sir. May I call upon stage Dr. Abhay Tirak sir, who is going to felicitate Dr. Basavaraja sir.
May I now request Dr. Manaklal Chopra sir to please felicitate Dr. Chetan Shah sir.
I request
I request Milind Baradia and Amol Pawar sir to felicitate our President 2023, Dr. Upendra Kinjwarikar, please.
Dr. Praful Patel sir will felicitate Dr. Samir Dalwai. Ready, no? Candles.
Thank you, everyone. I would now request all the dignitaries to please be seated.
To make this morning a blessed one, we invoke Goddess Saraswati by kindling the lamp of knowledge and wisdom. For seeking the choicest blessings for the same, I request all our dignitaries on the dais to step forward for lamp lighting ceremony.
Thank you, everyone.
I now invite Dr. Milind Bharadia, Chief Organizing Secretary, MAHAPEDICON 2022 and Founder President of Nashikon, to deliver his welcome speech.
Hello. Respected dignitaries on the dais, Dr. Ramesh Kumar, respected Madam, Dr. Phadke Madam, Dr. Basavaraja, Dr. Hemant Gangulia, Dr. Upendra Kinzadekar, Dr. Chetan Shah, Dr. Samir Dalwai, Dr. Ramakant Patil, Dr. Amol Pawar, dignitaries sitting in the first row, Dr. Sharmila Kulkarni, Dr. Devi, Dr. Sham Chaudhary, Dr. Sushil Parak, and all my dear friends, we have came here from the different parts of Maharashtra. Friends, I welcome you all to this holy city and wine capital of India, Nasik. This is the first physical conference after the COVID. COVID, you all know, was the most uncertain humankind has experienced. We have taken a charge in the month of November. After the November, there was like, everything was worried whether the fourth wave will come, fifth wave will come. And there were so many uncertainties were there in the atmosphere. At the same time, we have got the news from MAHAPEDICON that MAHAPEDICON has got postponed. After this news, so many delegates who have registered early were asking for the refunds. And at the same time, the pharma companies were not ready to participate for more than 200 gatherings. Friends, I am telling you all this on such a background of the uncertainties. We have accepted this challenge to host MAHAPEDICON. It was a very difficult task, especially for the first 6 months. But we Nashik IAPians, with our background of our own startup, Nashikon, which we have started in 2015, we have experience to handle such type of the crisis. Yes, we handled it very well. Friends, at the MAHAPEDICON, it's a very good mixture of the science and the cultural program. At the science, going by the theme, let's view the magic with the logic. We have designed the science where the pediatricians who are practicing at the periphery can manage the patients with the limited resources. At the same time, we are discussing some advanced pediatrics also. Going by the experience of the COVID, we are going to discuss pediatric practice, Kal, Aaj aur Kal, the future of the pediatric practice. We are going to have a lengthy discussion on it. At the cultural program, first time in the history of MAHAPEDICON, we have invited our delegates from all over the Maharashtra to participate our cultural program, that is the Love Is Zindagi. Love Is Zindagi is inspired by by our Pula Deshpande, ज्यांनी असं म्हटलंय, जर तुम्हाला तुमचं आयुष्य जगायचं असेल तर तुमचा छंद जोपासा. Friends, I am very happy to share with you all that we have got a tremendous response for this Love Is Zindagi. And I am very sure this pattern is going to follow in the next various conferences which are going to come. Last and the most important, please forgive us for any shortcomings in the next 2 days. I wanted to share with you all, we all organizing team have worked very hard since last 1 year. There is not a single day in our life in the last 1 year we have not spoke about the MAHAPEDICON. Thank you. Jai IAP.
Thank you, sir. I would now request Dr. Ramakant Patil, sir, organizing chairperson of MAHAPEDICON and executive board member of CIIP, to please present a synopsis of MAHAPEDICON.
आजच्या कार्यक्रमाचे अध्यक्ष, महाराष्ट्र बालरोगतज्ञ संघटनेचे अध्यक्ष डॉक्टर हेमंत गंगोलिया, तैल बुद्धी आणि अमोघ वाणीचं वरदान लाभलेल्या आदरणीय पूजनीय महामहोपाध्याय, टीचर्स ऑफ टीचर डॉक्टर मृदुला फडके मॅडम, तैलबुद्धी म्हणजे काय, अजूनही सेलिब्रेशन इन्स्टंट फास्ट किमान 100 विद्यार्थी आता मॅडमना भेटलेत आणि प्रत्येकाचं विदिन फर्स्ट सेकंड शी कॉल्ड हिम विथ हिज पर्सनल नेम and the range is from 75 to 2010. This is what is celebration called at the age of 80 years. सन्माननीय व्यासपीठावरील आजी अध्यक्ष, राष्ट्रीय बालरोगतज्ञ संघटनेचे अध्यक्ष डॉक्टर रमेश कुमारजी, राष्ट्रीय बालरोगतज्ञ संघटनेचे अध्यक्ष 2023 अवर होम ग्रोन डॉक्टर उपेंद्र किंजवडेकर जी, our friend philosopher and teacher 2024 CIIP president Dr. Basavaraj ji, West Zone Vice President of CIIP Dr. Chetan Shah sir, and my idol for being a poster boy and a smart guy, and at the same time very excellent in academics, our national coordinator for ECD and treasurer of CIIP Dr. Samir Dalwai. एनर्जी म्हणजे काय हे ओतप्रोत भरून असलेले महाराष्ट्र बालरोगतज्ञ संघटनेचे महासचिव डॉक्टर अमोल पवार, आणि तरुण पिढीचा शब्द आहे स्वॅग. स्वॅग म्हणजे काय, माझे सहकारी आणि महापेडिकॉनचे चीफ सेक्रेटरी डॉक्टर मिलिंद बराडिया, गणरायाला वंदन करून श्री प्रभू रामचंद्राच्या पदस्पर्शाने पुनीत झालेल्या पुण्य नाशिक नगरीत आपलं सहर्ष स्वागत, सहर्ष स्वागत, सहर्ष स्वागत. मान्यवर माझे ज्येष्ठ गुरु सहकारी डॉक्टर JS पटेल सर, डॉक्टर बोधनकर सर, डॉक्टर पांढरीकर, आमचे नाशिक IIP प्रेसिडेंट डॉक्टर केदार, वसंत खळदकर आणि किती नावं घेऊ. पण महोदय, आपण जे म्हणतो की इंडिव्हिज्युअल कॅन विन अ गेम बट टीम विन्स चॅम्पियनशिप. And my team, Dr. Shyam Chaudhary, who has been the backbone of this conference. He has worked, then we 5 of us, me, Milind, Sushil Parak, who is co-chair, Praful Patel is co-chair, PN Devi, who is treasurer. But putting all 5 together, one man has worked for this conference, and he is Dr. Shyam Chaudhary. How to work silently, selflessly, without hogging a limelight. He has given me a lesson for my life. I owe a humility, a virtue which I will like to imbibe in my life to Dr. Shyam Chaudhary. Friends, after again 10 years, in 2009, Nashik IIP had given me an opportunity to contend for or give or bid for MAHAPEDICON conference, MAHAPEDICON at Pune. We did get MAHAPEDICON in 2011. Again, in the story repeated in 2009. Again, they gave me responsibility to bid for 2019 in Amravati. We could successfully take the bid for Nashik MAHAPEDICON in 2021, but due to oblivion of COVID, we had to postpone it. And the credit goes to Thane IIP, Dr. Parmanand Andankar and Dr. Ram Shezara, who has graciously given us a buy to take this MAHAPEDICON. Friends, many of the guests have to take flight to NCPID. I think this is an opportunity for me to talk and talk and talk, but I will not take your time more. But what is more important, गणितात कच्चे असला तरी चालेल, हिशोबात पक्के पाहिजे. मी गणितात पक्का आहे, हिशोबात नाही, पण माझी टीम हिशोबात सगळी पक्की असल्यामुळे ही महापेडिकॉन शक्य झालेली आहे. मिलिंदने तुम्हाला सांगितलं आमचं जिस्ट काय आहे, weave the magic with the logic. And this will be possible only because of MAHAPEDICON delegates. Their response is very important. We had 8 pre-conference workshop. I think it was it in itself a record. And all the workshops were having more than 40 delegates. That is itself also a record because everybody attended it till the last lecture. We have got 44 sessions. We have got panel discussions. We have got lectures, orations, and all those things. Friends, I offer and with the team, please enjoy hospitality of Nashik IIP, the scientific and academic feast and cultural program, which has been specially designed by my better half, Dr. Sadhana Patil, Dr. Vaishali Baradia, Dr. Milind, Dr. Tushar Godbole, et al. The able scientific committee who has guided by Dr. Anil Kasliwal sir and Sharmila and ably supported by Amol et al. And other peoples. For the brevity of time, I will not take names of all, but whatever you appreciate, it is the credit of my team. Whatever shortcomings are there, please, onus is on me. Thank you.
Thank you so much, sir. Now I invite Dr. Amol Pawar, Honorary Secretary General of MAHAPEDICON, to present his report.
थोडा वेळ लागणार आहे. सेक्रेटरी रिपोर्ट आहे, पूर्ण वर्षभराच्या ऍक्टिव्हिटीचा रिपोर्ट आहे आणि टीम तर्फे सादर करण्याची जी संधी आहे ती सेक्रेटरीला दिली जाते. त्याबद्दल मी आधीच दिलगिरी व्यक्त करतो.
चीफ गेस्टना जायचं आहे, एवढं लक्षात ठेवून आपला कार्यक्रम कमीत कमी कसा होईल बघावा अशी विनंती.
तुम्हाला मी खालून तेच सांगणार होतो की लवकर आवरा, पण आता ठीक आहे. मी
तुम्ही दोघे नाशिकचे की पुण्याचे?
अजेंडामध्ये प्रत्येकाला 2-2, 3-3 मिनिटं लिहिलेले आहेत, पण प्रत्येकाला 4-5 मिनिटं लागणार. तर सेक्रेटरी रिपोर्ट आहे 5-7 मिनिटं लागतील. महा अकॅडमी ऑफ पीडियाट्रिक्स या राज्यस्तरीय संघटनेच्या 32व्या राज्यस्तरीय अधिवेशनाकरता अर्थात महापेडिकॉन 2022च्या उद्घाटन समारंभाला आवर्जून उपस्थित असलेले आपले सर्वांचे लाडके राष्ट्रीय बालरोगतज्ञ संघटनेचे अध्यक्ष डॉक्टर रमेश कुमार सर, प्रेसिडेंट इलेक्ट डॉक्टर उपेंद्र किंजोडेकर सर, महाराष्ट्र संघटनेचे अध्यक्ष डॉक्टर हेमंत गंगोलिया सर, पूजनीय मृदुला फडके मॅडम, डॉक्टर चेतन शाह सर, आत्ताच जे 1 महिन्यापूर्वी इलेक्शन पार पडले त्यात आतापर्यंतच्या सेंट्रल आयपीच्या हिस्ट्रीमध्ये सर्वात हायेस्ट वोट्स मिळवून जिंकलेले 13,000 मतं घेऊन जिंकलेले माझे मोठे बंधू डॉक्टर जी.व्ही. बसवराजा सर, डॉक्टर समीर दलवाई सर, डॉक्टर रमाकांत पाटील सर, चीफ ऑर्गनायझिंग सेक्रेटरी डॉक्टर मिलिंद भराडिया सर, पहिल्या रो मधले असलेले सर्व माझे मेंटर्स डॉक्टर उदय बोधनकर सर, डॉक्टर जे.एस. पटेल सर, डॉक्टर सुभाष कश्यपे सर, डॉक्टर पांढरीकर सर, डॉक्टर खलटकर सर, डॉक्टर यशवंत पाटील सर आणि सर्व ऑर्गनायझिंग टीम मेंबर्स, सर्व डेलिगेट्स आणि महाराष्ट्राच्या कानाकोपऱ्यातून आलेले सर्व मित्र, बंधू आणि भगिनींनो, तुम्हा सर्वांना माझा टीम MAHAIP तर्फे सप्रेम नमस्कार. As per presidential action plan, our presidential action plan, when we took over from 1st January 2022, we as a MAHAIP team comprising of office bearers, 14 EB members, MAHAIP Women Committee members, and 2 editors, we implemented various programs throughout the year across Maharashtra state involving MAHAIPians from each and every district and city branches. Administrative wise, we conducted 2 EV meetings held at Nasik and Yavatmal. Activity wise, we organized midterm CME along with HPCLC screening camp at Yavatmal on 5th June of 2022. As per CIP guideline, CIP Presidential Action Plan, various modules were organized by various branches. The most important, there are 15 nurturing care ECD workshops were arranged across Maharashtra state. We translated We translated CIP's parental guidelines from English to Marathi, and recently we completed half century with the help of none other than Dr. Nani sir, Dr. Nani Vadekar sir from Kolhapur. ऑनलाईन ऍक्टिव्हिटी वाईज डॉक्टर एक विचारू, पालक शिक्षक विद्यार्थी कार्यशाळा, ह्या माणसाची कितीही शब्दात आपण त्यांचं प्रेज केलं तरी ते कमी पडतील असे माझे बाबा डॉक्टर शेखर दाभाडकर सर. Then we conducted 2 HINA modules online with the help of Dr. Avinash Bhosle and 1 physical yesterday also. Then we conducted MAHAPEDICON case challenge, then mentor mentees, subspecialty, then Tuesday takes with the help of MAHAPEDICON Women Committee headed by Dr. Renu Boralkar and Dr. Sonali Shirbate ma'am, then Mahakatta, then various online quizzes for pediatrician, general practitioner, nurses with the help of none other than Amitabh Bachchan of our MAHAPEDICON, Dr. Jay Bhandarkar sir. Then journal journey with team Raigad IIP, then medical legal CMEs with the help of Thane IIP with help of Dr. Ram Gopal Chejara sir. Then MAHAPEDICON nursing stations with the innovative team Palghar IIP under the leadership of Dr. Hemant Joshi. All these activities were very well attended by pediatricians, PG students, nurses, parents, school teachers, and students, students as well. We have released 2 issues of MAHAPEDICON bulletin, and third will be released today in next few minutes. We made MAHAPEDICON website with all necessary recent updates of academic activities and member details also. We celebrated all days and weeks as per CIP calendar. First time in the history of MAHAPEDICON, which you will all have noticed. All these activities are recorded and available as on MAHAPEDICON YouTube channel, and the gallery has now 456 videos. All these activities were promoted on various social medias, Facebook, YouTube, WhatsApp, Telegram as well. We had 155 subscribers at the start of this year, and at present we are at 3,730. Facebook friends were around 200 initially, and now the number has crossed 4,000, and the exact figure is 4,844. It is the single largest Facebook account across Central IIP. It was Kerala before, and now we have crossed Kerala in Facebook account also. On Telegram, we are little bit behind. There are 382 members. On WhatsApp, we have almost 3,000 members across various groups. And they are bearing me a lot because of my various forwards, these online activities. And we don't now stop here. In next month, MAHAPEDICON Sports Conference will be hosted by Jalgaon IIP under the leadership of Dr. Sanjay Bhaviskar sir. I appeal all enthusiastic sports lovers as well as all MAHAPEDICON members to actively participate in various sport events. Details are there in registration counter. Friends, these things were not possible for us with full backing of Central IIP headed by my dear friend Dr. Ramesh Kumar sir and his team. Each organization needs a dedicated team to run the show. I am blessed with my friend, elder brother, coolest person, respected Dr. Hemant Gangulia sir, and our team members. These team members are Dr. Jay Bhandarkar sir, convener, KBC, Amitabh Bachchan of this our MAHAPEDICON quiz. Then Dr. Vinayak Patil sir, vice president. Then Dr. Mohan Warke, my little brother, treasurer. Then the most helpful of all these, Dr. Neha Singh, my right hand, the editor, and Dr. Vishal Raut, my left hand, the digital wizard. Then Dr. Renu Boralkar, chairperson, Women Committee. Dr. Sonali Shirbate, co-chairperson, Women Committee. Dr. Mahesh Moite sir and team Raigad IIP. And all MAHAPEDICON EB members as well as all MAHAPEDICON Women Committee members. So friends, it was a complete teamwork. We all have shown to MAHAPEDICON members that teamwork can do wonders. Lastly, I thank my dear wife, Dr. Sulbha Pawar, my parents, my family and kids, for considering my absence in various family occasions and also allowing me to work for IAP. I thank all my mentors, friends from Nasik as well as from entire Maharashtra for bestowing faith and also supporting me at various junctures in last 6 years of my career in Nasik IAP, MAHAPEDICON and Central IAP as well. My humble request to all to continue your love and support in my future. Thank you one and all. Thank you.
Thank you, Amol sir. That was quite an elaborative annual report of MAHAPEDICON. I now request Dr. Samir Dalwai sir, who is the National Treasurer of CIP 2022, to please share a few words with the audience.
कालपासून अमोलने मला 3 वेळा सांगितलं 1 मिनिटाच्या वर बोलायचं नाही बरं का सर. Respected an institution of this country, Dr. Mrudula Phadke madam, whose presentation was equally mesmerizing and humbling. And the more we learn from her, the more we understand that the most important quality for a pediatrician or any doctor is humility. जहां मैं का अंत होता है वहां आपकी शुरुआत होती है. Where the pediatrician ends, the child begins, and that's the great lesson we have learned from you, madam. We will try our best to emulate that. Respected President of Indian Academy of Pediatrics, Dr. Ramesh Kumar sir, all of you have heard of Marco Polo or Ibn Battuta. Why have you heard of Marco Polo? Marco Polo was a man who traveled the maximum distance in the world. India has beaten that record in the name of Dr. Ramesh Kumar. In the last 1 year, this gentleman must have traveled the whole country. Ibn Battuta and Marco Polo traveled for their own pleasure, for their own knowledge. He has traveled to give happiness, support and encouragement to the last pediatrician or IAPN in each and every part of the country. Every Friday morning, he leaves his house, leaves Cochin on Friday night, and travels at least 2 or 3 states on Friday, Saturday, Sunday, sleeping at airports, using public toilets at the airports, only because he wants to cover each and every IAPN. Mahan work, sir. I just wanted to enumerate that. पायगुण असावा तर डॉक्टर उपेंद्र किंजळेकरांसारखा असावा. गेली कित्येक वर्ष स्वतः डॉक्टर दाभोळकर साहेब आहेत, अमरावतीचे डॉक्टर पांढरीकर साहेब आहेत. आम्ही किती मेहनत केली, किती प्रयत्न केला. महाराष्ट्र सरकारने MOU साईन करून शेवटी करायला लागल्या तेव्हा कोविड आला. साहेबांचा पायगुण असा आहे, गेल्या 2-3 महिन्यात त्याने एक संकल्प सुरू केला आणि एका 15 दिवसाच्या आत चीफ मिनिस्टर काय 3 महिन्यात चीफ मिनिस्टर काय बदलतात, डेप्युटी चीफ मिनिस्टर काय बदलतात, सरकार काय बदलते, नवीन सरकार काय येते आणि यांचा MOU 4 दिवसात साईन होतो. असा पायगुण असलेले आणि त्या पायगुणाचं कारण आहे नियत. अगर नियत साफ है तो पूरी दुनिया आपको कायनात आपके साथ होती है. असे डॉक्टर किंजवडेकर त्यांचा ते सांकेल प्रकल्पाच्या बद्दल. मायटोकॉन्ड्रिया ज्यांना बोललं जातं असे डॉक्टर बसवराज. एंडलेस बाउंडलेस एनर्जी ऑफ फ्रेंडशिप, सपोर्ट आणि भरारी देणारे आपले IAPचे पदाधिकारी, जनरल सेक्रेटरी हु हॅज राईटली बीन इलेक्टेड विथ द लार्जेस्ट मार्जिन. डॉक्टर चेतन शाह आमचे व्हाईस प्रेसिडेंट, जे सर्वात एक त्यांचा मी त्यांना आता सांगत होतो, पूर्ण महाराष्ट्रात जर एक अशी who is the person who is the most unpolitical and the most truly genuine person who really wishes to help and connect with each other at a very low profile असले आपले डॉक्टर चेतन शाह सर. डॉक्टर हेमंत गांगुली आणि डॉक्टर अमोल पवारांच्या बद्दल मला थोडक्यात एवढं सांगावे लागेल की मला 2 चॅनल एकत्र बघितल्यासारखे बाजूला लावल्यासारखे वाटतात. आणि ते 2 चॅनल म्हणजे WWF आणि आस्था चॅनल. आता त्यातलं कोण काय ते तुम्ही बेटर जाणता. मला परत यायचं आहे कार्यक्रमाला म्हणून मी जास्त काही बोलत नाही. त्याचबरोबर माझे लाडके बंधू ज्याने खूप माझ्याबद्दल म्हणजे काय म्हणायचं तर ते प्रेमच म्हणावं, दुसरं काही म्हणू शकत नाही असं माझ्याबद्दल नेहमी बोलतात. पण ज्यांना मी एक आधारस्तंभ मानतो, मला काही सर्वांनाच बोलायला काही प्रश्न असतील आपण नेहमी त्यांना डॉक्टर रमाकांत पाटील साहेब आणि त्याचबरोबर डॉक्टर मिलिंद भार्डियाजी, तुमचे खूप खूप आभार. तुम्ही या खूप कठीण परिस्थितीत एवढा मोठा कार्यक्रम आयोजित केलात, आम्हाला सर्वांना या भव्य दिव्य ताजमध्ये इन्व्हाइट करता काय, हा कोविड होता खूप खूप आभार तुमचे. आणि हार्टिएस्ट कॉंग्रॅच्युलेशन्स फ्रॉम ऑल ऑफ अस. Respected Dr. Uday Budankar sir, who is a master at inspiration. After all the things that one has gone through, his inspiration to be with all of us. Dr. Malvadkar ji for inviting us to your city, Nasik President Sahib. And if I have forgotten anybody, please forgive me, but all the respected Central IIP EB members, Maharashtra IIP EB members, all the office bearers of all the district and branches and city branches across Maharashtra. And I just wanted to only name all of them, because final thank you is to all the delegates who come from all over Maharashtra, who attend these conferences. And these are the conferences, मी आता माझ्या शेवटच्या विषयाकडे येतो, that fill the coffers of the IIP. The money that the treasury in IIP has is only and only because all the effort put in by all of you and these people, that IIP has a treasury, and I get the undeserved honor of being the treasurer. Thank you so much.
Thank you so much, Sameer sir. Now I request Dr. Chetan Shah, Vice President, West Zone Central IIP 2022, to share a few words with us.
Most respected Dr. Mudula Fadke Madam, Dr. Hemant Gangolia Sir, President Central IIP, Dr. Ramesh Kumar Sir, Upendra Kinjwadekar Sir, President-elect, Dr. Basavaraj, going to be President in 2024, my friend Dr. Samir Dalwai, Dr. Ramakant Patil, and Dr. Milin from Nasik itself, and of course the two mitochondria of Maharashtra, Amol Pawar. So he is a very lively person. At the outset, I thank all of you for this overwhelming respect that I got from all of you throughout the year. I take this opportunity to congratulate the organizers of the conference, Dr. Ramakant Bhai and Dr. Milin. I heard what difficulties they had in organizing this, and they've done to the best of their capacity, and I feel it's going to be one of the best conference that we are going to have. I take this opportunity to thank all of you from bottom of my heart, because I had a great time working with all of you. See, I feel like homecoming because I'm born in Maharashtra. My father, mother, both are from Maharashtra. So मला असं वाटतं की मी घरी परत आलो. And I have that bond from childhood onwards. So I always enjoy, and I feel, little bit at the same wavelength as all of you. And of course, I had a great time working with our EB members, Dr. Nani Vadekar, Dr. Sanjay, Dr. Bela Madam, and Dr. Kulkarni Pramod Bhai, and the fourth person, Ramakant Bhai himself, and one of the, I think, great future in IIP. I always encourage him because he comes in a very prepared in all the EB meetings, and he contributes a great. So this is a moment of extending my thanks to all of you, and I get this opportunity. And of course, I congratulate Maharashtra IIP also for coming out with so many programs and doing them successfully. I have attended few of the programs and they were really good. So I congratulate all of you. So at the end, this may be my, last address to so many people in IIP, because ये प्रेसिडेंट सर की पोस्ट मेरे जैसे के लिए नहीं है. वो बहुत हेवी पोस्ट है एकदम. So maybe this is my last opportunity, and I thank all of you from bottom of my heart.
Thank you, sir. It was great listening to you. Next, I invite our President-elect CIIP 2024, Dr. G.V. Basavaraja, sir, to briefly address the audience.
Good morning to all. Respected our teacher of teacher, Professor Mudula Fadke Madam, and our national president, Dr. R. Ramesh Kumar sir, and our president, incoming president, Dr. Upendra Kinjidekar, and our MAHAPEDICON president, Dr. Hemant Gangolia ji, and Dr. Amol Pawar ji. First of all, I take this opportunity to thank all MAHAPEDICON who have supported me overwhelmingly. So I came to thank you all. And coming to the Maharashtra, I was working with the team Maharashtra for the last 4 years continuously, with the blessings of Dr. Uday Bodankar sir, our past president. Dr. Vijay Avale sir was my president of when I entered in a central IAPP. The present team, which is led by Dr. Hemant Gangolia ji, and secretary, rightly said, is the mitochondria of not only Maharashtra, is going to be the mitochondria of India. Dr. Amol Pawar, and of course our president-elect, incoming president, Dr. Ramakant Patil sir. Really, they have steered this, and previously Dr. Jayant Pandrikar sir was there, Dr. Sadachar was there. At that time, Dr. Amol was a treasurer. I was watching the whole team. What they brought, the culture in Maharashtra is, how do we take the academics to the greater heights, infusing the energy and reaching to the unreached. That's a great credit goes to the last 3 to 4 years team, which is really an amazing thing. And coming to the Nasik MAHAPEDICON, the team of MAHAPEDICON, which is led by organizing chairperson, Dr. Ramakant Patil sir, and Milind is an energetic, and I interacted with him quite some time. Of course, our Nasik president, Dr. Kedar Malvetkar sir, and many people who are working behind the scene, rightly said by Dr. Ramakant, Dr. Sham, Dr. Sharmila madam, Dr. Reena madam, and many more who are really Prapul sir. All really, they have done a lot of work to get this act together. So for last 1 year, I'm attending a lot of conferences. This is one of the best conference in terms of all hospitality and the scientific content and the attendance. Really, it is a benchmark for the future conference to come in. Of course, Dr. Ram is here. He may be watching the things. So Ram, Parmanand, Sneha madam, I think they're going to conduct an excellent program in 2023. Fine. Of course, our it wouldn't have been possible without the guidance of our past vice president like Chandrasekhar Dabadkar, Kedar Malvetkar, Vasant Kalatkar, Yashwant Patil. They all put in an effort. So once again, thank you for inviting me. Really enjoyed the stay. Once again, from my bottom of heart, I thank you all. Of course, our vice president, Dr. Chetan Shah, and our national treasurer, Dr. Samir ji. So put it in a very right way. So the whole treasury can come because of you, nothing behind that. Thank you.
Now I request our today's guest of honor, Dr. Mrudula Phadke ma'am, to address our delegates. Ma'am is XD in BJ Medical College, Pune.
My greatest of good wishes, blessings, and stay well, stay healthy, and reach more and more heights. Thank you so much.
Thank you so much, ma'am. It's our privilege to have you here.
May I now request Dr. Upendra Kinjidekar sir, President-elect CIIP 2023, to please briefly touch the audience.
मला तेवढं ब्रीफली का ग? न चोरहार्यम न च राजहार्यम न भ्रातृभाज्यम न भारकारी व्यये कृते वर्धते एव नित्यम विद्याधनम सर्वधन प्रधानम. इतका सुंदर श्लोक आहे की ज्याची चोरी होऊ शकत नाही, ज्याच्यावर राजा टॅक्स लावू शकत नाही, भावाभावात भांडण झालं तरी ज्याचा बटवारा विभागणी होऊ शकत नाही, आणि जितका त्याचा व्यय कराल तितकं ते वाढत जातं. अशी एकच गोष्ट, असंच एकच धनसंपदा आहे, ती म्हणजे ज्ञान. आणि तो ज्ञानाचा यज्ञ गेली 50 वर्ष अविरतपणे चालू ठेवणाऱ्या डॉक्टर मृदुला फडके मॅडम, तुम्हा सर्वांना, तुम्हाला सर्वप्रथम नमस्कार. And so many teachers over here. So my dear respected President Dr. Ramesh Kumar, President-elect of next year, Dr. G.V. Basavaraj, I'm so fortunate to have, they would be on the left and right side with me in 23. And we share excellent bond much before even there's such IIP and elections and this post when they were not there. So such kind of bond helps us when we are building IIP in future. So rest assured, next couple of years, IIP is in very safe hands. There's not going to be any turmoil. Anytime you're not going to feel that what are we members of. So let us assure that. My dear President of Maharashtra IIP, Dr. Hemant Gangolia, and Secretary Dr. Amol Pawar, they have done phenomenal job in the last years. As it was said, Dr. Jayant Pandrikar set the ball rolling. ती 2 वर्ष इतकी कठीण होती आणि जयंत आणि सदाचार तुम्हाला खरंच पुन्हा एकदा टाळ्या. खूप छान काम केलं तुम्ही. डॉक्टर मिलिंद भराडिया, मगाशी कोणतरी त्याला स्वॅग वगैरे म्हणाला. अहो, तो अमोल कोल्हे आता थोडा म्हातारा व्हायला लागला. शिवाजी संभाजीच्या रोलसाठी दुसरा कोणीतरी तुम्हाला दिसतो का? मी खरंच सांगतो, मी अजिबात अतिशयोक्ती नाही. अमेझिंग काम, कामाचा उरका त्याचा पण. आणि तो पलीकडे बसलाय, तो माझा जुळा भाऊ म्हणतात बरेच जण, काय कळत नाही. ते पहिल्यांदा स्वाती भावे मॅडमने मला एकदा म्हणलं, अरे समीर तू मला ते रिप्लाय केलं नाही का? म्हटलं काय, कशाच ते ऑटिझम वगैरे काय? म्हटलं मॅडम हो करतो. दिस वॉज रिअली स्वाती भावे मॅडम डिड दॅट मिस्टेक. सो इमॅजिन. सो हे चालू आहे आणि सध्या गेली काय कित्येक दिवस आम्ही असे एकत्र शनिवार रविवार बाहेर फिरत असतो, समीर आणि मी. सो वी आर लाईक great friends always. Dr. Chetan Shah very rightly described by Sameer. आणि किती हंबल बघा हा माणूस, की आज डिक्लेअरही केलं. म्हणजे आता उद्या उभं राहायचं ठरवलं तरी तो म्हणाला महाराष्ट्रात कुठं फोन करू, तिथे तो बोललो मी उभं राहणार नाही. पण ही इज रिअली जेन्युइन, अ बिग ब्रदर. आणि सर आता इथे मी आधीच अधिकृतपणे, इवन दो आय एम नॉट इन चार्ज, डॉक्टर रमेश कुमार इज इन चेअर, की 23 ऑल्सो यू आर गोइंग टू गाईड अस as far as financial health of IIP is concerned. Rest assured, you will be there in the prime positions. And Dr. Ramakant Patil यांनी मगाशी सांगितलं, ऑर्गनायझिंग चेअरपर्सन या कॉन्फरन्सचे, हा माणूस पहिल्यांदा इबीत आला या वर्षी बरं का. पहिल्या इबीला थोडेसे लोक दबकत असतात. रेणू घाबरू नको, ठीक आहे. तर तसं न करता म्हणजे प्रत्येक वेळेला व्यवस्थित अभ्यास करून जी काही जबाबदारी दिली ती पूर्णपणे पार पाडून, इनफॅक्ट त्याला आता यावेळेला FBS एखादी काही वेगळ्या गोष्टींची जी जबाबदारी दिली गेली ती पूर्णपणे यथार्थपणे ते शिरावर पेलून त्याचा अभ्यास करून त्याचा रिपोर्ट परत देणं. सो इट वॉज अ ग्रेट थिंग. आणि इट वॉज नो वंडर की सेकंड टाइम ऑल्सो ही कुड कम बॅक विथ थम्पिंग मेजॉरिटी. सो रमाकांत, इट विल बी अन ॲसेट टू वर्क विथ यू इन 23 ऑल्सो. आणि अर्थात नवनिर्वाचित आमच्या टीमचे सदस्य रेणू बोराळकर आहे, जितू आहे का? अरे जितेंद्र गव्हाणे आहे, डॉक्टर प्रमोद कुलकर्णी, हा प्रमोद पण आहे, आणि अर्थात डॉक्टर गिरीश चरदे नागपूरचे. या सगळ्यांचं पुढच्या टीम मध्ये स्वागत. एकच गोष्ट मगाशी समीर म्हणाला तसं, आय टेक दिस ऑपॉर्च्युनिटी टॉक अबाउट द संकल्प संपूर्ण स्वास्थ्य, the program, flagship program. If you have seen last couple of years, IIP has changed focus. Madam, you would be happy to know that. So it is not only remaining an academic body only for academicians, doing programs and modules only for our members, but we are shifting our focus towards community. So we had parental guidelines, we had ECD module. What is it? It is to empower the parents. त्याच्यात pediatricianचा काही रोल नाहीये. So we are empowering the parents to take better care. So we and parents together take care of the child. So that was all about ECD module. Dr. Ramesh Kumar went one step ahead. UFI-MR, madam just talked about, the complete oration was on that UFI-MR, and he chose some aspirational districts, not Kerala and Maharashtra, where the work is not much required. In fact, he comes from Kerala, which has the best health indicators in the country. But still, he had that idea to go somewhere which is, UP, then the Jammu and Kashmir, Odisha. So these are the districts he chose for, and then the committed efforts. So big round of applause to Dr. Ramesh Kumar for doing that. And then we went one step ahead. आणि मला यावर्षी वाटलं की शाळांमधल्या मुलांबद्दल काहीतरी करावं, आणि म्हणून हे संकल्प संपूर्ण स्वास्थ्य हा कार्यक्रम मी नियोजित केला विथ NCDP, आपल्या डॉक्टर रेखा हरीश मॅडम, डॉक्टर दीपक पांडे आणि खूप टीम मेंबर सगळ्यांचं अर्थात सहकार्य आहे. तो आपले 2 की फोकस एरियाज असणार आहेत. लेट मी टेक दिस ऑपॉर्च्युनिटी टू टॉक टू यू. 3 ते 9 आणि 10 ते 18 वयोगटातल्या मुलांसाठी आपण न्यूट्रिशन, एक्सरसाइज, स्क्रीन टाइम, स्लीप आणि वरच्या वयातल्या मुलांसाठी मेंटल हेल्थ आणि सबस्टन्स अब्यूज आणि ऑफकोर्स टेकिंग केअर ऑफ एन्व्हायरमेंट, हे 5 की फोकस्ड एरियाज असतील. आपण एकदाच शाळेत जाणार आहोत. 45 मिनिटाचा प्रोग्राम असेल फॉर 3 to 9 year old अँड फॉर 10 to 18 year old इट विल बी अ 2 hour प्रोग्राम. टीचर्सना आपण अटेंड करायला लावणार आहोत आणि एकदा आपण गेल्यानंतर प्रत्येक वेळेला तुम्ही टीचर्स त्या शाळेत जायची गरज नाही. तर ते टीचर्स तो प्रोग्राम पुढे त्या शाळेत चालू ठेवतील. जवळच जवळ तुमच्या जॉग्राफिकल लोकेशनच्या ज्या शाळा असतील तिथेच कमिट करा. आणि नक्की मी तुम्हाला सगळ्यांना ही नम्र विनंती करतो की प्लीज या योजनेमध्ये मोठ्या संख्येने आपण सहभागी होऊया. आता समीरने सांगितलं त्याच्याबद्दल थोडंसं सांगतो की हा प्रोग्राम तयार होता, पण आपले नवनिर्वाचित सदस्य प्लीज डॉक्टर गिरीश, विल यू प्लीज गेट अप. A big round of applause to this man. He had some connections with BJP higher authorities. आणि देवेंद्र फडणवीसांची त्यांनी सहजपणे अपॉइंटमेंट, अर्थात आम्ही गेलो वेगळ्या कामाला तिकडनं एकनाथ शिंदेचं बाजूलाच, तो सहज म्हणाला खरी अपॉइंटमेंट नव्हती, सर वर्षावर पण जाऊया ना. वर्षावर गेलो आणि लेट मी शेअर दिस एक्झाम्पल. आपल्याला कळत नाही कसं काम केलं जातं. एकनाथ शिंदे सर वॉज सिटिंग देअर. वी हॅड टू वेट फॉर 3 hours outside. Myself and Dr. Pakul Parekh also was with me. And we just went and he was sitting. He said 2 minutes, 2 मिनिटात सांगा काय म्हणायचंय. And I could tell him in 2 minutes. ते म्हणाले बसा बसा 1 मिनिट, इंटरेस्टिंग वाटतंय काहीतरी. थांबले तिथे, मला म्हणाले इथे बसा. पीएला म्हणाले दीपक केसरकरांना फोन कर रे. शालेय शिक्षण मंत्री केसरकरांना फोन केला. नुसतं तेवढंच नाही, केसरकर तुम्हाला एक डॉक्टरांना तिथे पाठवतोय आता मंत्रालयात, ताबडतोब मला त्याच्यावर कार्यवाही व्हायला पाहिजे. तेवढ्यावर थांबले नाहीत. लेट मी टेल यू. त्यांनी मला विचारलं डॉक्टर साहेब तुमच्या गाडीचा नंबर काय, म्हणजे तुम्हाला मंत्रालयात एंट्री कशी मिळेल. म्हणजे व्हॉट काइंड ऑफ डिटेल थिंकिंग ही ऑलरेडी हॅड. आणि मोठा शेरा त्याच्यावर मारला, ताबडतोब कार्यवाही करावी. व्हॉट मोर वेन एकनाथ शिंदे माननीय मुख्यमंत्री असं म्हणतात आणि तो कागद घेऊन मी तिथे केसरकरांकडे जातो काय, त्यांनी तो कागद बघितला. हे सगळं गेले गुरुवारची गोष्ट. आणि त्यांनी वर बघितलं कॅलेंडरकडे आणि मला म्हणाले 14 नोव्हेंबर बाल दिन चालू करूया. अहो फक्त 3 दिवस आहेत सर. मी म्हटलं जस्ट काश्मीरहून मी आलो होतो, आमची ईबी मीटिंग होती. सो विच फिनिश्ड, वी केम ऑन ट्यूजडे. हे सगळं बुधवार गुरुवारी झालं. शनिवार रविवार सुट्टी, तरी 2 दिवसात MOUचे थोडे एक्सचेंजेस करून फायनली सोमवारी तो लगेच साई नाही झाला. समीर, डॉक्टर बेला, डॉक्टर जीतू सगळेजण बरोबर आले होते. सो इट वॉज ग्रेट. आणि समीर म्हणाला ते अगदी खरं आहे की आपली नियत साफ असेल तर सगळ्या गोष्टी बरोबर येतात. पण आता हा चान्स मिळालाय, आता हा सोडूया नको. वी ऑलवेज लुक फॉर ऑपॉर्च्युनिटी की मला एक कॉमन IPN म्हणून कम्युनिटीसाठी मी काय करू शकतो. हा प्रत्येकाच्या येतो की ना मनात विचार की मी माझ्या समोरच्या पेशंटला चांगलं बघतो. आय गेट बेटर ट्रेंड इन अस्थमा, बेटर ट्रेंड इन इंटेन्सिव्ह केअर, बेटर ट्रेंड इन NRP. बट धिस गिव्हन अस ऑपॉर्च्युनिटी. त्यांनी GR काढला की तुम्हाला शाळेत जाणं आपल्याला जे इझी होणार आहे. तो प्लीज सगळेजण, 65,000 शाळा आहेत. मला म्हणाले एकनाथ शिंदे साहेब, सर माहिती आहेत ना आपल्या महाराष्ट्रात शाळा, एवढा मोठा व्याप आहे ह्याचा. So let's hope that we get this opportunity and all of us use it in the best possible way and we set an example to other states की ह्याने सुरुवात करूया की ज्याद्वारे आपलं एकच 18व्या वर्षी जेव्हा आपली मुलं ऍडल्ट होतात तेव्हा एक सुदृढ आणि सशक्त महाराष्ट्र आधी, आणि अर्थात महाराष्ट्र प्रत्येक गोष्टीत अग्रेसर आहे देशात, ते रोल मॉडेल आपण भारतासमोर उभं करूया. आणि या IIPच्या अमरेलाखाली आपण सगळेजण एकत्र येऊन या कार्यक्रमात हातभार लावूया ही सगळ्यांना नम्र विनंती करतो. पुन्हा एकदा या महापेडिकॉनसाठी खूप खूप हार्दिक शुभेच्छा. एकच सॉरी टू से की आम्हाला आता लगेच 1:30 वाजता इकडनं बाहेर पडायचं. शिर्डीला NCPID आहे, त्यामुळे इकडनं शिर्डीहून पुढचा प्रवास आहे, त्यामुळे थोडंसं लवकर जायला लागू शकेल. पुन्हा एकदा शुभेच्छा. थँक्यू.
Thank you, sir. It's always a pleasure listening to you.
I now request Dr. Ramesh Kumar, sir, President Central IIP 2022, to share a few words of wisdom.
The vibrancy of Upen is still in the auditorium, I believe. Upendra, we are yet to see him for a year. Wonderful coming up. It's going to be great, Upendra. Most respect. I mean, the tone set by Samir in when a lighter vein is still in the auditorium, and I feel it's not a formal function; it's an informal function where you are all connecting with the speakers as well as you are all enjoying together and appreciating the dignitaries. Most respected, Madam Mudula Phatke, ma'am, we are really honored by your presence here and the oration, and in fact, it was a great learning experience. Right from you made it very different, and the intrauterine battle between the XX and XY itself. That is something which was so enamoring for all of us. The president of MAHAPEDICON, Dr. Hemant Gagolia, I mean, it was a wonderful year for the MAHAPEDICON for 2022. And Dr. Amol Pawar, I must take this opportunity. this Amol power is not the mitochondria. I mean, it must be the ATP of IAP. We should go a step ahead because it comes on the energy comes in bundles, not mitochondria is a one. So the Amol and the team has been such an amazing team. We have gone to the report there. I mean, Maha EAP has been such a good one in numbers. But last few years, we have seen it growing visibly, active, physically, academically, and I mean more as a family, and you are getting that the knack of getting into the scheme of things. So congratulations, Amol, because you have been your webinars, you are active, and I mean, as I echo Basavarai's sentiments, you are going to be the man for IAP in the next decade. So congratulations, and Hemant Gagolia, sir, you have been leading from the front. My dear friend, my beloved friend Upendra, once again, I'll come back to you. You are, I mean, the son of the soil, and it has always been quite interesting to travel with Upendra and Samir together. And the twins there, they make such a great bench. When I was a Secretary General in 2018-19, I had a treasurer as well as the Joint Secretary, Upendra and Samir with me, and that was a great time. Anytime you look forward to them, they will be really at your help, and the troubleshooting, everything was done. And of course, Samir with his oratorial skills, he can match anything and beat anybody on that point. And he is the brand ambassador for IAP. That is Dr. Samir, you have been our spokesperson. You lead IAP in all visible forums, and you speak for the children, and you speak for IAP. And we have been so happy that you are at the helm, and you are going to be at the real helm maybe 2-3 years down the line. I am certainly sure about it. That the years are not that far. And well, of course, my friend Dr. Basavaraj, the man who has been leading IAP for the troubled times of COVID. I'm with Dr. Piyush Gupta and Dr. Basavaraj, Dr. Bakul Parekh. Dr. Basavaraj has done an amazing job, and thank you, Dr. Basav. And my dear friend, Dr. Chetan Bhai, as Vendra said, Chetan Bhai is an asset for IAP on financial grounds. And of course, Chetan Bhai is now, we have got another financial wizard there, Dr. Ramakant Patil. Ramakant Patil ji, because last year executive board, both of them were there, and I never had any issues. We had any financial issues coming up, Upendra will say Chetan Bhai and Ramakant, you put them on the honors on them, and that is settled. The next EB meeting, it is settled, and Ramakant has been, I heard your speech today, and somebody was talking to him, you are 3 years back, and now is 1.5. I'm not sure who told me just now. So it's so heartening to listen to you, and my best wishes to the year coming up in your tenure as the president. And I was wondering, I saw this R.D. Patil as the president-elect. I asked just now only, Dr. Gagaulia, who is R.D. Patil? I know him only as Ramakant only mostly. So then he said, I was looking at where is R.D. Patil, where is R.D. Patil? Then he only told me, sir, this is only Ramakant Patil. So that was quite interesting, and I was really surprised. Again, your secretary, Dr. Milind, and first time only I'm meeting him. I don't remember how you meeting him, but this vibrancy and energy is infectious, I believe. The way the smiles and the unique beard and everything that makes you the tag man for IAP and Nasik. And I think people once who have seen you will never forget you for your looks as well as for your activities. And on the stage, I think all the dignitaries are there. And well, of course, Uday Bodanga sir, always the youngest president IAP ever had, and he's still the youngest president, physically, mentally, as well as in all terms and all thoughts. You see the president's group every day morning, at least some 10-20 messages are there, Bodanga sir. Maybe some forwarded, maybe some his own thoughts, maybe his new thoughts. And many times we may agree, may not agree, but it is always there. And the sir's thoughts are there. Dr. Jayesh sir, Dr. Jayesh Patil, and my Dr. Kedar Malvarkar ji, he was my vice president in 2018. And now he's the president of the Nasik IAP, sir. It's so good that coming to Nasik, the city of grapes, and we had been here for the MAHAPEDICON too. So it's always we've been quite heartening talking to you and enjoying with you over the time. And Vasant Kaladkar as well as Dr. Yashwant Patil. And my EB members here, I must take, because it's always official when you come to the EB members. My EB members, Dr. Pramod Kulkarni is there, Dr. Sanjay Deshmukh is there, Dr. Nani Vadekar. Nani Vadekar ji, we are so happy to see that the parental guidelines have been translated under your listing, as well as Dr. Where is our Baba? Baba is here. So, Dabadkar ji, Dabadkar, you used to tell. That time he used to make our executive board so vibrant. So, Dabadkar ji is here. And the new, of course, the new EB, Dr. Jitendra is there, as well as Renu is out here, Pramod Kulkarni again coming up, and Girish, so happy for you that you are taking the lead. And well, of course, we are blessed with people who are the connects. Because IAP has, if we explore, we have got enough numbers who have got a good connectivity. We are not actually utilizing that connectivity. We need to explore. We have 39,000 members. If you go ahead in our community, if you use the political will which we can generate, the goodwill which we can generate to our connectivity, it is amazing. And that we need to do it. It is nothing wrong in that. It is nothing bad in that. We need to go ahead further. And well, of course, my friends from Thane, Dr. Ram Gopal is there, Dr. Sneha, the year ahead, your MAHAPEDICON is there. And I still remember, I did many of you may are not sure, I did my MD in Sholapur only, Dr. BM Medical College. And I went for my first Pedicon in Maharashtra in 1992 or 93, I think, Nagpur. I am not sure. Dr. Deshmukh was there that time, our HOD, and we were compelled to go. 3-4 of us should register and go. And that was my first journey. And that was my first Pedicon. And I never believed I should be in this dais on the stage as a president. Afterwards, some 30 years down the line. So that was 1992, maybe to 30 years now. So it's a God-given opportunity, and I am so thankful to my friends in IAP and across the country for me. I'll take only 1 or 2 minutes more, friends. The Central IAP is going quite good, quite strong now. Academics has been our fort. Well, we are exploring everything and whatever we can do on every front. Academics, we have done enough, and we have been doing NCECD, NDEP, some 15 modules this year. Of course, that has been running and we keep running. Again, we are looking at the other aspect, advocacy front also. We have been doing quite well. Child rights, we had a connectivity there, and Mr. Sailas Satyarthi was there again in MP Pedicon. Kailash Satyarthi was all praised for IAP activities. So the way we have been doing child rights, whichever way we can do. And again, for the charity front, we had that Indian Academy of Pediatrics has taken some certain districts. We have started our charity work in Bairabingi in UP on this Children's Day. We have taken 200 families in select districts. We are giving something called a superhero kit to these families. One school bag with IAP, everything is written, and one small kit which contains certain messages. Each item in the kit contains certain messages. None of these items are given by Government of India or any other agency. For example, it contains a nail cutter. Nail cutter contains a message for the hand hygiene. Soap is there. One mosquito repellent is there. He give a steel mesh is there to contamination food of water. So certain messages are there, and we had this function there in Berabangi, UP, and 200 families in 20 districts is being taken. We have been talking about charity, and we never have been doing charity. I mean, executive, we never have been involved the community as direct beneficiaries anytime. We always talk about charity. How charity? We are improving the capacity building of our members and indirectly doing charity to the community. No, we are making ourselves bigger or our office better. So that has been a question. IAP is basically registered as a charity, the charity trust, the registrar of charities. So we have been looking. This year, we have started with the honest support of our executive board by Upendra, Samira, and everybody. We have started going in the charity, and we have spent around 40 lakhs for 50 lakhs we are spending on charity this year, where the downtrodden, the underprivileged families are the direct beneficiaries. Nobody is there. Central ISP is spending directly on them. And again, the under 5 mortality, as Upendra said, we are going to the aspirational districts, the high burden districts. Our efforts get diluted across the country. There is no point in spending this under 5 mortality. The IAP championing the cause in Kerala. The under 5 mortality is hardly 8 there. But we have got districts in Budaun in Uttar Pradesh where the under 5 mortality is 88. You add 1 8 to the other 8. 8 in one side of the country, 88 in another side of the country. I have come from Cochin. The under 5 mortality is 8 there. But I have been going to Budaun, I have been going to UP, Madhya Pradesh, where the under 5 mortality is more than 60, 70. So that is the target where IAP is putting. So we need not dilute our efforts across the country. We need to be focused. The focused implementation of IAP's force. These districts, we have selected champions of IAP. And these champions have been going to the districts. With the help of the district administration, we have selected families there, where IAP's efforts are there. We have been focusing and catalyzing the efforts of Government of India on efforts like Anemia Mukt Bharat, on efforts like diarrhea control, the infectious disease control, the malaria, encephalitis control. We are not doing ourselves. We are catalyzing the activities. I am sure with the year 2025, we will be achieving the under 5 mortality 25 by the year 2025, well ahead of the sustainable development goals. With Indian Academy of Pediatrics, we are definitely going with the government. I believe all of you have to contribute. All of you have to contribute in every district, every branch, and we are taking focused implementation. So that's great from coming from IAP. Once again, my congratulations to MAHAPEDICON. Once again, Amol and Hemant Gagolia for making it so visible every day, so much of activities. You have been so good, DS, and wish you the very best. And I'm so happy coming to MAHAPEDICON after 1992, coming over to this 2022 as the president, greeting you and wishing you, and wish you all the best. Wish you the best of the post-COVID time. And let's hope the great years are ahead. Upendraj, Basavaraj, all are here. All executive board members are there. I wish them all the best. Once again, all my Nasik friends, hats off to you for having such a great event, including Dr. Kedar and all the friends for arranging such a great meeting here. And it has been amazing coming here. Your affection and care is unimaginable. Thank you very much, IAP.
Thank you, sir. We truly thank all the dignitaries on stage for sharing their valuable words with us.
We'll now proceed to unveiling of various publications and souvenir at the hands of all the dignitaries on the dais. I request Dr. Sadachar Uzramkar and Dr. Sushil Parekh to come on the dais. I request all the dignitaries to do the honors of unveiling MAHAPEDICON souvenir.
Dr. Sushil Parekh. Sushil Parekh. Praful, you come on the dais.
Dr. Sushil Parekh, sir, please come on the dais.
Dr. Shyam Chaudhary.
Next, may I call upon stage Dr. Neha Singh, Dr. Vishal Raut, and Dr. Chandorkar, sir, with their MAHA-IAP bulletin to be unveiled. May I now request Dr. Narendra Nanavarikar to please come on stage and unveil the Marathi edition 2022 of Parental Guidelines. The English edition was released in 2022 and now they are releasing the Marathi edition.
Dr. Davadkar, you have got great contribution into this. Please come on the dais to unveil this. Anna, you then photo.
I now request Dr. Narendra Nani Vedekar with his parenting guidelines, the Marathi edition 2022.
Now I invite Dr. Chetan Shah to come on the dais. I request all the dignitaries to unveil antibiotic awareness poster.
हिना वर्कशॉप सोबत वारकेचं हिना वर्कशॉप सोबत दिलं ना award.
I appeal, see, we are beginning with the celebration of Antimicrobial Awareness Week from today. 18 to 24 is celebrated all over the world. You can put this photograph and put it on your media profile or anywhere where you can WhatsApp DP, everywhere in OPD, you can put it this big number and use this to spread this important message. Thank you.
And MAHAPE is releasing a flyer on this today itself at 4 p.m.
May I now request
Thank you.
Dr. Avinash Bhosle and Dr. Kashmiri Barbade to please come up with the HINA workshop souvenir.
I request the dignitaries to be seated on the dais. We'll now have the most awaited and important award ceremony. Please join your hands and give a thunderous applause to these hardworking branches. I request Dr. Amol Pawar, Honorary Secretary, Maharashtra IP 2022, to conduct the award ceremony.
पुढचे एक 15-20 मिनिटं लागतील. जी वर्षभराची मेहनत आहे, प्रत्येक डिस्ट्रिक्ट ब्रांचेसचे जे ऑफिस बेअरर्स आहे त्यांना खरंच सॅल्यूट की पूर्ण वर्षभरात ते ब्रँचला जेव्हा पोस्ट घेतात प्रेसिडेंट, सेक्रेटरी, ट्रेझरर, त्यावेळी त्यांना फॅमिली टाईम, इतर बऱ्याचशा गोष्टींना कॉम्प्रोमाइज करावं लागतं आणि ह्या सर्व कॉम्प्रोमाइज करताना ते वेगवेगळे IIP डेज, वीक्स सेलिब्रेट करत असतात. तर एक 15-20 मिनिटं लागतील, लवकर आवरण्याचा प्रयत्न करतो. We'll start the most awaited this prize distribution ceremony as we want to acknowledge and reward all the district branches office bearers for their contribution in various MAHAPE activities and CIP activities. With the hands of all the dignitaries,
We'll request, 1 minute. We'll request all IP branches to be as quick as possible, as fast as forward to come forward, as their name is announced on the PPT. Please be fast.
I will call upon all the office bearers of various branches category wise. So first category is the members branch with less than 50 members. So the first prize goes to IIP Yavatmal. So I request office bearers, Dr. Ajay Keshwani, Dr. Swapnil Mankar, Dr. Zubid Khan. They won the best branch award in this category. Then in ORS week, they are also winner. In breastfeeding week, they are first runner-up. Next branch is Palghar and then Hingoli. So the Palghar IIP own base branch, first runner-up in base branch category. Then in ORS, they are first runner-up. In breastfeeding, they are winner. Next branch is Hingoli. Be ready. नहीं रोड हेलो चालू कर दो ना. Next is IIP Hingoli branch. So I request Dr. Venkatesh Damkondwar sir, President, Secretary Dr. Nitesh Chaudhary, Treasurer Dr. Manish Mupkalwar. They won they are second runner-up in base branch. They won the appreciation award in ORS and also in breastfeeding. They won the appreciation award. Next is Wardha.
Dr. Snehal Nakre, Dr. Damkondwar, from all Hingoli.
Anybody is there? Hingoli, from Hingoli? From Hingoli. Come, sir, soon, fast. Ale. Ale. Hingoli. Next is Wardha. After Hingoli, Wardha is there. Thank you, madam. Next is Wardha. So, Dr. Rajendra Borkar, Dr. Amol Lohkare, Dr. Abhijit Khanke, please come on dais. They won the appreciation award in base branch and ORS, and they are second runner-up in breastfeeding. Then IIP Usmanabad branch. Then Jalgaon. Thank you, sir. Thank you. Usmanabad, anybody is there? Usmanabad. Wardha nahi kun. Wardha. Next is Jalgaon. Usmanabad. Usmanabad. Dr. Abhay Patil, Dr. Makrand Barate, Dr. Abhay Kumar Golkar. Usmanabad.
अरे उस्मानाबाद द्या ना. वर्धाच होता. उस्मानाबाद. हाँ येस.
Next branch is IIP Jalgaon. Come on, dais, sir. Dr. President, Dr. Sanjay Baliskar, sir. Secretary, Dr. Vishwesh Khadke. Treasurer, Dr. Gaurav Patil. They won the appreciation award in all three categories.
जलगांव. 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 जलगांव कौन है? जलगांव. जलगांव.
Next is IIP Dhule. They won the appreciation award in all categories. Dr. Vilas Wadile, Dr. Avinash Sendane, Dr. Dadabhai Patil.
Dear friends, Dr. Ramesh here. I was supposed to take the next lecture due to anemia, but now that we have to leave just now by 1:30 to Shirdi and for NCPAD, I beg your apologies. we have to leave right now. So continue with your program. Thank you so much for the love and affection. Thank you.
Thank you for gracing my talk. IIP Dhule. Then next is IIP Malegaon. They won the appreciation award in all three categories. President Dr. Prashant Wagh, Secretary Dr. Pushkar Aire, Treasurer Dr. Virendra Patil. Malegaon, IIP Malegaon.
Anyone from Malegaon?
Yes, they are coming, sir.
Malegaon,
They are
be
coming,
fast.
they are coming. Then next is IIP Sangamner. Then next is IIP Sangamner. President Dr. Jayashree Jadhav ma'am, HOD, Pravara Medical College, my medical college. Then the Secretary Dr. Amit Awad, my friend, and Treasurer Dr. Sandeep Hon.
Malegaon.
Deepak sir.
Sir, तुम्ही द्या ऐकून.
थांब याच्यानंतर. बॅकग्राऊंड पण बदललं. पुढचे रेडी आहेत. Then next is IIP Nandurbar after Sangamner. IIP Sangamner now.
Anyone from Sangamner?
IIP Sangamner, Dr. Amit Kakad is there. Then next is IIP Nandurbar. President Dr. Jayan Shah, Secretary Dr. Nishat Rangrej, Treasurer Dr. Shamal Jain. Then now next, anybody from Nandurbar? Dr. Nishant. Then next, anybody from Nandurbar, last call. The next category is branch with 51 to 150 members. So the first prize goes to IIP Solapur branch, headed by President Dr. Manisha Chavan, Secretary Dr. Rajeshri Meru, and Treasurer Dr. Atul Janwar. They own
Solapur.
they are winner in best branch, they are winner in ORS, and they are first runner-up in breastfeeding also. IIP Solapur. Pratibha Patil madam is also there. She is a zonal member of Women Committee. Next is IIP Kolhapur. Be ready. IIP Kolhapur. So, President Dr. Kimaya Shah, Secretary Dr. Shilpa Hirgude, Treasurer Dr. Ajay Shinde, Nani sir is there from Kolhapur, so he can join. So they won the best, they are first runner-up in base branch. They won the appreciation in ORS, and they are the winner in breastfeeding. Next is IIP Amravati. Pandrikar sir, will please ready. IIP Kolhapur. Next is IIP Amravati branch. So, President Dr. Naresh Taide, Secretary, my dear friend Dr. Sushil Rajput, Treasurer Ruturaj Deshmukh. They are the second runner-up in Breast branch. They won the second runner-up in ORS, and they won the appreciation award in breastfeeding. IIP Amravati.
Hey, 1 minute, Amol. Dr. Vinayak Patil. Dr.
Vinayak Patil.
Vinayak Patil.
I request Dr. Vinayak Patil sir to come on dais during award ceremony.
Next is IIP Raigad. Next is IIP Raigad. President Dr. Pramod Wankhede, Secretary Dr. Dhananjay Singh Rajput, and Treasurer Dr. Sagar Khedu. They won the Appreciation Award in Best Branch. They are the first runner-up in ORS. They won appreciation in breastfeeding. Next is IIP Bid. Next is IIP Bid. President Dr. Subhash Joshi, Secretary my dear friend Dr. Sachin Jetlia, Treasurer Dr. Rahul Raut, सासरवाडीचे लोक,
बर्थडे बॉय डॉक्टर परमानंद आंदनकर इथे आलेले आहेत, तर सगळ्यांनी त्यांना बर्थडे विश करायचंय. हॅपी बर्थडे परमा.
Happy birthday Permanent sir. Past TB member of Central IIP. IIP Bid on the appreciation. Yes. They won the appreciation award in Best Branch. They won the appreciation in ORS and they were second runner-up in breastfeeding. Next category, be ready. The most awaited awards between 151 to 300 member category. Now the awards in 151 to 300 member category, and the first award goes to Navi Mumbai team, headed by Dr. Jitendra Gawane sir, President Dr. Jitendra Gawane, Secretary Dr. Satish Shahane sir, Treasurer Dr. Mangaya madam. They are base branch winner in base branch, then winner in ORS and winner in breastfeeding, and also they are first runner up in IIP Charity Day. 4 awards. Next team is IIP Thane. Be ready. IIP Thane, be ready. Next is IIP Thane team, headed by Dr. Sneha Kokate madam, Secretary Dr. Amruta Baviskar, Treasurer Dr. Grivita Raikar, and ably supported by Dr. Ramgopal Chezara, past president. They are the first runner-up in base branch. They are the first runner-up in ORS, and they won the appreciation in breastfeeding. Next team is IIP Aurangabad. Be ready. I request Dr. Uday Bodhankar sir and Dr. J.S. Patel sir to be on dais during the awards. I request Dr. Jayant Pandrikar sir also to present on dais during the awards. Next is IIP Aurangabad, headed by President Dr. Rajeshri Ratnaparkhe, Secretary Dr. Sandhya Kondpalle madam, and Treasurer Dr. Varsha Vaidya. They are the second runner-up in base branch. They are the second runner-up in ORS, and they are the second runner-up in breastfeeding also. So so applause to IIP Aurangabad also. Then next is IIP Nashik. Next is IIP Nashik, headed by President Dr. Kedar Malvatkar sir, Secretary Dr. Rishikesh Kute, Treasurer Dr. Nitin Mekarkar. They won the appreciation in base branch and ORS, and they are the first runner-up in breastfeeding also. My home team. No, yeah.
आवाज कुणाचा आवाज कुणाचा
Nashik IIP won the appreciation award in base branch and ORS, and they are the first runner-up in breastfeeding. So the next category is more than 300 members. The next awards are for branch more than 300 members category. We'll celebrate this in banquet also, please guys. We have less time. So the IIP Mumbai won the award in all categories. So President I request Dr. President Dr. Rajesh Chokhani sir, Secretary Dr. Nehal Shah, Treasurer Dr. Amruta Shirodkar madam. They own all the awards. So today's main winners are IIP Mumbai. So they are winner in base branch, they are winner in ORS, they are winner in breastfeeding, and they are winner in IIP Charity Day also. आवाज So the next branch is IIP Pune. Be ready. आता ते इकडे गेलंय ना ते मग ते नंतर वाचतो ना ते हा मग बाय Next branch is IIP Pune, headed by President Dr. Parag Gaikwad, Secretary my dear friend Dr. Deepak Mule, Treasurer Dr. Manoj Jhalte. And they are the first runner-up in base branch. They won the first runner-up in ORS, and they are the first runner-up in breastfeeding also. Next is IIP Pune. President Dr. Parag Gaikwad, Dr. Deepak Mule, Dr. Manoj Jhalte, And all the stalwarts are from Pune, Dr. EB member, Maharashtra IP, Sanjay Natu sir, then Pramod Kulkarni sir, Lalit Rawal sir.
Branch Pune, under able leadership of Dr. Sanjay Nathu.
IP Nagpur, be ready. Dr. Jain Joshi, please come on dais. Next is IP Nagpur. Congratulations, Dr. Shilpa Yadav madam, my guide. She will be next president for Pune. Next is IP Nagpur. President Dr. Rajkumar Kiretkar, Secretary Dr. Pritesh Khatwar, Treasurer Dr. Mahesh Turale. They are the second runner-up in base branch. They are the second runner-up in ORS, and they are the second runner-up in breastfeeding also. All stalwarts, Yashwant Patil sir, Bodhankar sir, Anil Raut sir, Rajkumar sir. Thank you. I request Dr. Snehal Nagre to receive the IIP Hingoli branch award. I request delegates to be seated. There are more awards for some special contributors also. So be seated after your awards for only 5 minutes. So thank you for cherishing the cherishing and appreciation for these awards for the year of 2022. Thank you one and all. So now we want to acknowledge and thank the special contributors. काय पाहिजे आता? हिंगोलीचं कुठे? हा, हिंगोली. हिंगोली है ना? Now we want to acknowledge and thank our special contributors. Dr. Shekhar Dabadkar sir, my Baba. Dr. Shekhar Dabadkar sir. Okay. Dr. Shekhar Dabadkar sir. I request Andorkar sir to receive the award of Dr. Shekhar Dabadkar sir. Dr. Shekhar Dabadkar sir, come on dais to receive your. Next is Dr. Narendra Naniwadekar sir. Dr. Shekhar Dabadkar sir, please can I help kara? झालं आता 4-5 जण पाहिजे. डॉक्टर एक विचारू, हा महा अकॅडमी ऑफ पिडियाट्रिकचा पालक शिक्षक यांच्याकरता सुरू केलेला शैक्षणिक व सामाजिक उपक्रम या माणसाने वर्षभर वेगवेगळ्या विषयांना स्पर्श करत दर रविवारी अत्यंत नियमितपणे चालू ठेवून कमालीचा यशस्वी केल्याबद्दल महाराष्ट्र आयपी तर्फे त्यांचे आभार. नेक्स्ट अवॉर्ड इज फॉर डॉक्टर नरेंद्र नानेवडेकर. पॅरेंटिंग गाईडलाईन्स आपण जे वाचतोय ते सर्व काही शक्य झालं नरेंद्र नानेवडेकर सरांबद्दल मुळे. हो गया. अरे ठीक है ना, अभी वो नवी मुंबई जाते Dr. Naniwadekar sir, then Dr. Jay Bhandarkar sir, the master of MAHAPEDICON, the convener of MAHAPEDICON. Then next is Dr. Vinayak Patil sir. Dr. Vinayak Patil sir. Dr. Vinayak Patil sir. Then Dr. Neha Singh, be ready. Dr. Neha Singh, then Dr. Renu Boralkar, be ready. चलो चलो जल्दी. Dr. Renu Boralkar, be ready. Then Dr. Renu Boralkar, she is the chairperson of MAHAPEDICON Women Committee and the next year EB also. She won the elections with good margins in recently concluded elections. Then Dr. Shonali Shirbate, be ready. Dr. Sonali Shirbate. Then Dr. Jayant Pandrikar sir, IPP, and now then Dr. Jayant Pandrikar sir.
अजून कोण राहिले? अरे वा.
Dr. Vishal Raut, Dr. Pramod Kulkarni, be ready. Dr. Girish Charde sir, be ready. Number hai na. Dr. Vishal Raut, come on guys. The digital wizard who will be promoted to Central IIP as a digital wizard also, as Dr. Upendra sir has told yesterday. पूरा फ्लायर्स वगैरह सब वही पूरा सिर्फ करेक्शन कर लेता मेरे से बस. Then Dr. Pramod Kulkarni. Then next is Dr. Girish Charde, who has recently won the EB elections. We want to acknowledge the new EB members also. Then Dr. Vasant Khalatkar sir. I think he has gone. Dr. Vasant Khalatkar, Dr. Uday Bodhankar sir will receive this award as Dr. Vasant might have gone to catch his flight at Shirdi. Yeah, sir. Hey, special contributors. आणि EB वाले चला झालं 2 मिनिटं. Then Dr. Jitendra Gawane sir, my elder brother from Navi Mumbai. आमचे सटाण्याचे जावई आता जस्ट ते EB मेंबर म्हणून नॉमिनेट झालेले आहेत वरती सेंट्रल IIP ला. Dr. Sanjay Deshmukh sir, whether is here? Dr. Sanjay Deshmukh. I request Dr. Yashwant Patil sir to receive the award of Dr. Sanjay Deshmukh. Dr. Yashwant Patil sir is election commissioner from West Zone, Central IIP. Then the last is Dr. Ramakant Patil, who won the elections with the biggest margin in consecutive 2 years. Last year he got 2,390 votes, and this year he has crossed 2,400. My mentor. Thank you for bearing with us. I declare this award is over now. Thank you. Thank you, one and all. I request all the dignitaries to be seated for 5 minutes.
I request all the dignitaries to be seated. And I also request the delegates to be seated.
Hello.
I kindly request once again all the delegates to be seated. I now invite Honorable President of MAHAPEDICON and President of today's function, Dr. Hemant Gangolia, sir, to deliver his presidential speech.
Hello. If everybody is there in the hall. Namaste and good afternoon to everyone, whoever is present in the hall. All names are being said, all adjectives of the guest being said. I do the city all of that. And I endorse as MAHAPEDICON president, the adjectives said all about the whoever the present on the dais in absentia or in the presence. भाषण करताना मला असं वाटलं की कुठल्या भाषामध्ये बोलावं, कारण 2 चॅनलची भाषा आहे, आस्था किंवा WWF. आता प्रश्न पडलाय कुठल्या शब्दात माझं भाषण करावं. एनिवे, जी भाषा मला माहित आहे ती मी सांगतो. तरी मला उल्लेख करावासा वाटतं, our youngest IAP president Dr. Bodankar sir and our Ladke Baba Dr. Dabadkar sir, my IPP Dr. Jayant Pandarikar sir, who just set the ball rolling. Yesterday, past IIP president Dr. Bodankar sir said, because there was a question of 2 channels. So yesterday, what Bodankar sir said, Hemant Pawar. He devised this name yesterday, and I was surprised to see that. So that is a unique combination. Hemant Pawar. Of course, Pawar may be having the gun of Sharad Pawar. The gun of Hemant, I do not know. Amol, you have stolen my 2 words, right and left. The year started when I used to say, my right hand is Amol and my left hand is Jay Bhandarkar. As the year progressed,
I request Jay sir to be on dais and Dr. Pandrikar sir and Dr. Dabadkar sir to be on dais. Please, please.
as the year progressed, anyway, as the year progressed, the right hand and left hand of Amol became Neha and Vishal. Hearty congratulations to Vishal and Neha. Hearty congratulations to IAP Nashik branch under the lead presidentship of Dr. Kedar Malvatkar and fine organizing team, Dr. Ramakant Patil sir, Dr. Milind Baradia, to organize such a wonderful conference at a wonderful venue with excellent scientific deliberations. Hats off to you, sir. All the organizing team from IAP Nashik. And whatever the claps are present in the hall should go to them. To say what the year was, because I will take short time, what the year was, first of all, I should really thank my IP Raigad branch who gave me this opportunity to come to this post. And equally, I would like to thank Dabadkar sir. Dabadkar sir, तुझे खूप खूप धन्यवाद आणि आभारी आहो. आणि IP रायगडची टीम ज्यांनी मला ही संधी दिली ऍज अ महाआयपी प्रेसिडेंट टू वर्क करायला पूर्ण वर्षभर. आणि खरोखरच बऱ्याच काही गोष्टी शिकलोय IP रायगड करून. आणि मला काही नावं नमूद करावीशी वाटतात जे महत्वाचे आहेत. जेव्हा आम्ही हे प्रोग्राम रोल आउट केलं, सुरुवातीला 2 महिने थोडे मला कष्ट झाले, पण इट वेंट इनटू द टोटली इन ऑटो मोड. म्हणजे काही करावं लागायचं नाही. इट वॉज टोटल इन ऑटो मोड थिंग. So to start with, it was Friday releases. All pictures from Mumbai released on Friday. So there were Friday releases, starting with MAHAIP Mentor Mentis, thanks to Nilima Bhandarkar and Mahesh Moite sir and the team of that. It was MAHAIP subspecialty and the 2 faces of Mass Social Connect, Dr. Samir and Dr. Jay, were the instrumental for that. MAHAIP Case Challenge, again, which was started in the MAHAPEDICON Paschim Rang, and that it fly to Alibag, physical one, and then that it came to this year, thanks to Dr. Mahesh Moite, Dr. Sunita Ingre Madam, Dr. Shilpa from Raigad, to keep the high level secrecy up to the last moment. Thank you very much. Mahakatta under the leadership of Dr. Amol, anything. Tuesday takes 100%, Dr. Renu and Dr. Sonali. Heartfelt thanks to you and all 10, I will not take the name, but all 10 zonal committee members across the Maharashtra state to do the wonderful. Tuesday takes मध्ये मी माझं डोकं कधीच घातलं नाही. I am very frank and honest with you. Just any query, I used to say, otherwise Tuesday takes totally yours. Then IIP Thane, I don't know, they have gone, but IIP Thane, medical legal hearing from Ram Gopal Sajera, excellent thing. जेवढे एपिसोड होते, they were the viva of us because we never knew all the questions. All questions came to instantaneous, spontaneous. Then with this, that was with IIP Thane. Then we came with the IIP Raigad with Journal Journey. Journal Journey we started on a doctor day. It went across the year with in association with MAHAIP. Then we had with IIP Palghar, MAHAIP Nursing Station. Then I would like to thank Dr. Naresh Taide and Dr. Sandeep Karle for whole month of celebration of thalassemia and sickle. Excellent work was done in that time. And then lots of week celebrations were there throughout the year. I would like to express my deep, sincere, heartfelt thanks to all MAHAIP office bearers, executive board members, MAHAIP One Committee, chairman, co-chairman, all zonal committee members. I am really indebted to them. And I'm very, very proud to say or respect all MAHAIPs who took the initiative part and just it went on auto mode. Many people used to ask how you're managing. Really to say it went totally on auto mode. Everyone learned there what they want to do, what time they want to do, and everything was kept rolling and rolling and rolling. And these things all went away finally. And I would like to thank each and every MAHAIPian to come to the social media. We really hit the YouTube channel, all the episodes which was started in Dr. Jayant Pandrikar's, I think so, yes, left. That is also on the MAHAIP YouTube channel, yes, sir. And this year's all episodes related to whatever I said, the statistics well known to Dr. Samir. Kitna videos hai? So 470 videos. Kitna subscriber hai? So he is the statistician man, 3,000. Same thing about Facebook, same thing about Telegram, we are lagging. The Telegram, we started thinking that all 5,000 members of MAHAPE should come on Telegram. Lots of efforts was put by Dr. Amol, Dr. Neha, but we could not. We managed to only collect 370 to 380 members itself. Probably next year it will get.
थोडं दम धरा बाबा.
ते डायनामाइट आहे. It will explode. ते सर्व शेवटी ना concluding remarks होते. तर this was all done in the presence of academics, then social media, and lastly to come to the, we have to, as a pediatrician, we should have a social connect. And the social connect, we were missing this all years. And probably name was not taken, but I was accompanying when the MOU was signed by our Upendra Kinzavdekar and Jitendra, then Bela, and then Nehal, and then Samir. Everybody was there. Excellent initiative made and excellent initiative started. One thing I do not know whether Amol mentioned or not, because the ECD module, 15 of them we completed in Maharashtra. And so in the West Zone, it's almost 25. And probably West Zone completed all the whatever the ECD module was suggested. Though extra kiye aise. That was social connect, and the social connect, the most important thing. आणि जसं सर्व म्हणतात माझे पण बाबाच आहेत. डॉक्टर एक विचारू का? Excellent program. I started with communicating myself with the first person to talk was Dr. Samir Dalwai sir. Then came to Dr. Dabadkar, because I have to make the coordination proper between these two people. Then only I knew that things will roll down. So Samir and Dabadkar sir. And then I went across to Suchit Tamboli, because I should take the names of the Suchit Tamboli and another one minute. This thing started with these three people, first of all, and with myself. And then we get into Dr. Jayant Pandharikar, because I wanted to make all things sure. You should work. Otherwise, it's starting and nothing in place. If it doesn't work, then there is no use of discussing all the things. So all programs, I first make sure that you should run for the year. Then only I went to it. So these three people were the initial instrumental. And then I went to Dr. Jayant Pandharikar, Dr. Sadachar, and I'm forgetting Abhinay Daravade was there, and Sandeep Kaude was there, Dr. Sandeep Kaude, and then later it got added with Dr. Nilima Bhandarkar is now doing fine, excellent work, and Dr. Shubha Khirwadkar, sir, my काय नाव आहे सांगा, Shubha Khirwadkar, आणि कुठलं नाव आहे अजून कुठलं? These people are doing fine, but फ्लायर बनवणे, फोटो टाकणे, विषय लिहिणे, सोमवारी ते चालू होतात. Yes, Dr. Dabadkar sir, you have taken excellent pains and खूप काम केले. So really, I am indebted to you, sir, and so that is wonderful things. To conclude, actually I wanted to say a lot, but I will conclude with just two points. Any association runs on its name, not on any individual. When individual comes in between, association deteriorates, gets disrupted. So I think so we should take care of the association, rather individuals. And I think so that is the most important thing. And I always believe teamwork. कधी जसं दिसतं तसं नसतं, तसं नसतं तसं दिसतं, अशा गोष्टी होत राहतात. पण मला असं म्हणायचंय, it is a teamwork which with the association, which makes all glamour and all whatever the feathers in the cap of the association, it is only the teamwork which will do the wonders. And probably I experienced that in this year, because मला काही करावं लागलं नाही गेल्या 7-8 महिने. Everything खाली फोन करायचं, everything was got done. Thank you once again all MAHAPians for that wonderful work in this year. And lastly, yesterday, Dr. Ramakant Patil and your team, you started well. Dr. Milinची दाढी खूप आवडली मला. आणि आम्ही 1-2 दिवस शेअर नाही केलं तर एकदम बेकार दिसतोय. पण मिलिंद, यू एक्सलंट सीन. आणि डॉक्टर रमाकांत काल बऱ्याच वर्कशॉप मध्ये बोलताना मी बोललो होतो काल की ये जो वर्कशॉप ये जो कॉन्फ्रेंस शुरू हुई है ना, ये तो वर्कशॉप इधर ट्रेलर है. और आज सुबह मूवी चालू हुई है पूरी पिक्चर अभी बाकी है और ये 2 दिन में आने वाले में आप 100% श्योर हूं मैं कि सब लोग एंजॉय करेंगे। सब बढ़िया कॉन्फ्रेंस होगा और बढ़िया मेन्यू है और तहे दिल से सभी का धन्यवाद। और सभी ने मुझे बहुत अच्छा मदद की है, इसलिए बहुत 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 धन्यवाद। खूब आभारी आप सभी लोगों का। थैंक यू वेरी मच। जय आईपी, जय महाराष्ट्र।
Thank you so much, Hemant sir. It was great listening to you. Let us now move on to presidential medallion handover. Let us now move on to presidential medallion handover. I request Dr. Hemant Gangulia, President Maharashtra IIP 2022, to hand over President Medallion to incoming President, our very own Dr. Ramakant Patil sir, in the presence of all the dignitaries on the dais.
So it gives me immense pleasure to hand over the presidential medallion to our incoming MAHAPEDICON president, academically very strong, and I really love his English, and sometimes I feel I should take the tuition class from Dr. Ramakant sir, because though I can speak English, but I am weak in English. Sometimes I have to Google search so many words that you say, Dr. Ramakant sir, and then I understand what you want to say. So I want to just hand over this presidential, sir, mic.
Yeah. So give a big round of applause, Dr. Hemant to Dr. Ramakant. Yeah. Yeah. See, I'm really grateful to Hemant for being accidental president and accidental guest. After 3 presidents left, Hemant said, Amol said, you come. So I'm really grateful to you. Yeah, please, madam.
Sir, that was a word I used when I gave my acceptance speech as an accidental president of my IP.
Anyway, thanks.
So you should not take my word.
Yeah. Asa gaya. Photo gaya asa. Yeah. Madam, yeah. You can pass EBM. Wow. Madam, you can come. Bella, madam,
Madam, hat lava.
Please. Yes.
So on behalf of Central IIP,
Congratulations, Ramakant Patil sir. I may now request the incoming president of MAHAPEDICON 20.
on behalf of Central Indian Academy of Pediatrics, CIP, I would just like to mention one word for Dr. Hemant Gangulia. Maharashtra is our biggest branch in India, in IIP. And to manage that so well is a very, very difficult task because 5 regions आहेत आपले, वेगवेगळी माणसं, cultures change across regions, and he has been able to be the unifying factor आणि सर्वांना एकत्र घेऊन ह्युमिलिटी दाखवून कसं काम करायचं त्याचे प्रतीक आहेत. आम्हाला ECD चे लॉन्च करत असताना 40 वर्कशॉप वेस्ट झोन मध्ये करायचे होते, तेव्हा त्याच्यात द फर्स्ट पर्सन वी थॉट हू कॅन कोऑर्डिनेट दिस एंटायर झोन and everybody in this zone, and to assist Dr. Chetan Bhai Shah, we only thought of Dr. Hemant Gangulia ji, and he accepted that, and he has excelled in that branch as well as all the other things. So a big round of applause for this humility, his hard work, and we wish you all the best. Thank you so much. On behalf of CIP, we would also like to welcome the incoming president of this
very important branch, my dear friend and brother, Dr. Ramakant Patil. उपेंद्रजीने सांगितलंच आहे. Thank you. उपेंद्रजीने सांगितलंच आहे that in this first EB, we were so impressed by his accuracy, his attention to detail, आणि बारकाईने प्रत्येक गोष्ट बघायची, कॉन्स्टिट्यूशन असो की अकाउंट्स असो, आणि त्याने एवढा मोठा एक इश्यू जो होता तो त्याने सॉल्व्ह केलेला आहे. मला वैयक्तिक फक्त एकच सांगायचं आहे, this man is extremely strong person, very, very grounded, and most impressed I am by the fact, I think he's the best dressed pediatrician in Maharashtra. You see him anytime, he's perfectly, immaculately dressed. एखादं जॅकेट खूप इंटरेस्टिंग घालणं वेगळी गोष्ट असते, एखादी फ्लॅशी टाय घालणं मी जशी घातल्याचं इझी असतं, पण टॉप टू बॉटम बुटा सकट, आणि असं म्हणतात की माणसाचं खरं ड्रेसिंग सेन्स बघायचं तर बूट बघावे, मोस्ट परफेक्ट. आणि बाबांचं म्हणणं आहे कदाचित क्रेडिट ते मॅडमचं असेल. सो एक्स्ट्रीमली प्राऊड and happy to have you as part of CIP. Thank you so much. Thank you very much.
आणि ड्रेसिंगच म्हणायचं ना, कॉन्फरन्स झाली सर, नंतर मग आम्ही निघायचंय ना, तेव्हा बघितलं, अरे रमाकांत सर डिफरंटली ड्रेस when departing from the venue.
सर आइए ना.
आप बोलो.
आता माइक.
Now I'll request Dr. Ramakant Patil sir to give his acceptance speech.
खूप छान सजेशन आहे. किती वेळ आहे? Now our president sir has said adhere to his lecture of adhere to anemia. Our Maharashtra state president and president of this function and conference, Dr. Gangulia sir, youngest president and still the youngest president, representing all the three presidents of CIP, Dr. Ramesh Kumar sir, Kendavadekar sir, and Dr. Basavaraj. Our my friend, philosopher, idol, Dr. Samir Dalwai, Chetan Shah sir, who is having a very calm demeanor, composure, and financial wizard, and putting his say in very gentle manner, and he has been a very transparent man. As I said, I want to be like Dr. Shyam Chaudhary. He is the second person like whom I want to be, a calm demeanor and well composed person. MAHAIP Sachiv Dr. Amol Pawar, newly elected executive board member with whom I will have a pleasure and honor to be working with them in CIP 2033. My friend, Dr. Jitendra Gavane. Everybody must have witnessed the spree and flurry of wonderful online lectures and even offline lectures by NMAP. This was the first district branch which has started its YouTube portal, and all the lectures and panel discussions and everything are available. I thank Dr. Jitendra Gawane for having this visionary action. My other colleague will be Dr. Renu Poralkar madam, who will be the first time EB. Dr. Girish Charde, a resourceful man, and he will be an asset to CIP. My senior colleague, Jyesht Sahakari, Dr. Pramod Kulkarni, probably he is attending to other delegates because we are busy over here. And he is again a friend, philosopher, and very studied man, MRCPCH from London College. Friends, we had an excellent year after the oblivion of 2 years last year. What different programs were there? Mentor mentees, Tuesday take, case challenge, Dr. Ek Vicharuka. Wonderful, wonderful, wonderful. I congratulate team MAHAIP, Dr. ably led by Dr. Hemant Gangulia and ably supported by Dr. Amol Pawar, and wonderful the team. I will not take the names for the sake of brevity, but it was wonderful. What I do want to say in this, is that all these excellent programs in next year will be continued. And I take due permission from Dr. Hemant Gangulia sir, because these are all his brainchild, to allow us to continue these programs. What I learned in CIAP, we have got all these 12A certification and different things. But income tax says yours is not a 12A certified, which can be justified, and we didn't get a 36A. So whatever our income will be taxable. So already social norms has been taken place on virtually, but we have to work, increase our share of social work, and in the footsteps of CIAP, we will be trying to follow and increase our social work and share of it. Friends, we are working basically what I thought. CIAP, MAHAPEDICON, this is organization of pediatricians. And what my personal view is that we should be working for our members first. And if we work for our members, naturally we are working for the child health. Now the time has come that we are being strangled from many sides. Gone are the days when like family physicians have been extinct. Now coming 10 years, 15 years down the line, small hospitals, individual hospitals with individual face of a doctors will be extinct. And water might have flown below the bridge, and at that time, the introspection and being sorry will not going to help. If many of us are not in a private practice or having small hospitals, but we have to comply at least 22 to 25 compliances. And these 22 to 25 compliances make us run pillar to post. We are at the same time clerk, manager, doctor, and whatnot and whatnot. And what I feel that under able leadership of Dr. Upendra Kinzavdekar, Dr. Basavaraj, and Dr. Samir Dalwai, we would like to represent through CIAP a different compliances. Like there is already a letter of CPCB, that is Central Pollution Control Board, that if a corporation or municipal area has got central effluent treatment plant or sewage treatment plant, we don't need any MPCB authorization. These people unnecessarily make us run pillar to post. CA certification is required, and as whatever your value of your project, and then they charge the fees. This has to be stopped somewhere, and it will be done only if we are united, even if we have to do some Sakhri Uposhan and whatnot and whatnot. So in my presidential tenure, I will like to work in addition to whatever has been done and social work. I will like to work particularly for pediatricians, a small hospital pediatrician, their problems, hospitals. This I will like to work, and I need your support of all, and particularly dignitaries who can guide us on this, and support of all the delegates to conduct this work for private practitioners. Friends, we will unveil our programs of presidential plan in due course of time. I again thank my host branch, Nashik IAP, and then President Dr. Ravindra Sonone in 2021. Under his able guidance, Nashik IAP has chosen me to be presidential candidate for president-elect. आणि आपण माय माऊलीचं कधी इनफ पुरेसं धन्यवाद देऊ शकत नाही, तसं मी होस्ट ब्रँचचं धन्यवाद देऊन उतराई होऊ शकत नाही. And जसं गंगोलिया सर म्हटले की institution is always larger, I say it cannot be compared. Nobody can be larger than institute. Even if we have witnessed one man institute, they also believe institute is always larger. I thank my host branch, and also thank the president in 2019, Dr. Pankaj Gazre, to depute me, Dr. Sanjay Aher, for bringing this MAHAPEDICON in Amravati to Nashik, and in 2021, Dr. Ravindra Sonone, and Nashik IIP under his able leadership to choose me the candidate for presidential elect in 2022 for Maharashtra State Branch and president in 2023. Thank you so much. Thank you for your patient listening.
Thank you so much, sir. Coming to the concluding part of the ceremony, I now invite Chief Organizing Secretary of MAHAPEDICON, Dr. Milind Bharadia, and Honorary Secretary General of MAHA-IAP, Dr. Amol Pawar, to propose the vote of thanks.
I thank Dr. Hemant Gangulia, Dr. Samir Dalwai, Dr. Uday Bodhankar sir, Dr. Pandrikar sir, Dr. Jitendra Gawane, and everyone on the respected dignitaries on the dais for attending this program. And the most important, the large number of the audience was present throughout the inauguration and prize distribution ceremony. Friends, I can't end my vote of thanks without mentioning 2-3 more names. One is Dr. Sadhna Patil and the other one is Dr. Vaishali Bharadia. They have tolerated us in last 1 year. They have handled all our family responsibility, professional responsibilities very well. Thank you, Sadhna madam, and thank you, Vaishali. And the most important, the program has been conducted very well by Dr. Prachi Birari and Dr. Shalaka. Thank you. Now we will all stand for the national anthem. Lauto ka? To bol do.
शुभ आशीष मांगे जय दाता जय भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Once again, happy birthday to Dr. Parmanand sir. आज रात्रीचं बँक्वेट त्यांनी स्पॉन्सर केलेलं आहे.
On this note, we hand over our MOC to Dr. Reena Rathi and Dr. Neha Mukhi for further proceedings.
Good afternoon, everyone. I, Dr. Reena Rathi, along with my colleague, Dr. Neha Mukhi, will be with you for the couple of hours and take you to the journey full of knowledge and wisdom. May I invite chairperson for this neonatal session, Dr. Shama Kulkarni from Nasik, Dr. Prabha Khaire from Aurangabad, Dr. Chopra sir from Nasik, May I invite chairperson for the session, Dr. Chopra, sir, from Nasik. Dr. Prabha Khaire, ma'am, from Aurangabad. Dr. Shama Kulkarni from Nasik. Along with our first speaker of this neonatology session, Dr. Sandeep Patil from Nanded on the stage. Although it deals with the smallest form of human life, neonatology assumes so much importance that our good proportion of our pediatric colleagues have shifted our practice from just neonatology. The advancement of this branch of pediatrics has made huge strides to save those little ones who had difficulty in their first breathing. For further light on this, I invite Dr. Sandeep Patil, sir, who is going to talk on prognostication of asphyxiated newborn. Over to you, sir. Thank you.
Good afternoon to one and all. First of all, I want to say, I want to express my sincere thanks towards IIP Nashik, as they have allowed me to stand in front of you. So today I am going to talk on prognostication of asphyxiated newborn. So before going into this topic, I will tell a small story to you. There was a call from obstetrician. Resident goes for attending a delivery. Baby delivers who is limp. The baby needed ventilation. Baby was intubated. And after intubation, when the resident was busy in fixing the endotracheal tube, anesthetist बाहर आता है, anesthetist पूछता है, मैडम पूछ रही है, बेबी कैसा है? तो at that time, the resident tells कि हार्ट तो नहीं था, हार्ट शुरू हो गया है और ब्रीदिंग में दे रहा हूँ, बैग एंड ट्यूब वेंटिलेशन से ब्रीदिंग इंटरमिटेंटली आया है. वो एनास्थेटिस्ट अंदर जाके बोल देता है ऑपरेटिंग ऑब्स्टेटिशियन को, और उसके बाद व्हेन जो रेसिडेंट बेबी को शिफ्ट करने के लिए ऑब्स्टेटिशियन को बोलने जाता है, मैम दिस बेबी नीड शिफ्टिंग, आई एम गोइंग टू शिफ्ट दिस चाइल्ड एंड आई विल अपडेट यू. तो वो ऑब्स्टेटिशियन तुरंत बोलती है, सर इसका बेबी कैसा रहेगा, ये बचेगा कि नहीं, ये बच्चे का न्यूरोलॉजिकल कैसा रहेगा? तो वो जो पुअर पीडियाट्रिशियन रेसिडेंट है वो बेचारा बोलता है, मैम पहले मैं इसको शिफ्ट करता हूँ, उसके बाद मैं बताता हूँ. So this topic I kept in front of you to make you sensitize, make you strong to prognosticate in labor room to your NICU, because you are answerable from obstetric unit to your NICU for long-term prognosis of the child. So the two questions which the obstetrician simply asked कि क्या होगा ये बेबी का, So basically they wanted to know what is the outcome of the child in the form of mortality and what is the neurological outcome. So I will put in front of you this topic in these following heads. So we are going to discuss how is the prognostication of this child goes. So just look into the Indian data. So Indian data, see this Indian data suggests for extramural babies, birth asphyxia incidence is 18.1% and neonatal seizures is 16% out of them. While intramural births where in-house deliveries happen, so the birth asphyxia rate is very less as compared to extramural deaths, extramural born babies. So Western data is similar to intramural data, and most of the neonatal encephalopathy, whatever baby you see, so first you have to label it as it can be, or if you find a baby with HI encephalopathy, you should find out whether it is secondary to HI or not. So after that, just I want to sensitize you people about what is called as perinatal asphyxia. So ACOG and AAP has combiningly put these 4 criteria to diagnose it as a birth asphyxia. ऐसा नहीं है कि बस baby रोया नहीं है तो it is birth asphyxia, because there is a lot of medical legal issues are there in US as well as in overseas. So for that, these 4 criteria to be fulfilled to diagnose it as a birth asphyxia. Clear? So now I am going to take you directly from birth asphyxia definition to the prognostic markers, which is my intention today to tell you about that. So there are different markers which I had put in front of you. And here I'm going to tell each marker what is the importance of that. And basically here needs a lot of statistical terms are going to come in, like negative predictive value, positive predictive value, confidence interval, odds ratio, and that. So basically I'm going to tell you at the point when they come in. I will try to simplify it basically so that to understand what, how they are going to help you out. First of all, we'll see Apgar score. Apgar score usually calculate at 1 minute, 5 minutes, and every 5 minutes till the Apgar score reaches to the 7 or more than that. So whenever you want to define it, or when you want to use Apgar score, basically for prognosticating the child, you are more concentrated for Apgar score. If it is less than 5, basically you should consider that prognostication will be very bad. So very poor prognosis will be there at 5 minutes or more than that Apgar score. You are not going to prognosticate depending on 1 minute Apgar score. 5 minute, 10 minute Apgar score, or beyond that Apgar score carries importance. So 5 minute Apgar score you should tell. If it is less than 2, the poor prognosis in the form of death or disabilities around 80%. Like that you can consider, or you can confidently consider for saying that. So Apgar score at 1 minute does not have importance. 5 minute Apgar or beyond that Apgar less than 2 carries importance. This point you have to keep in mind. Disadvantage of this scoring for prognostication is that it is a retrospective scoring. Whenever you are resuscitating, you are not going to calculate the Apgar score. You have to remember it and then you have to write. So there might be some memory bias in that. So basically this retrospective scoring is there, which is going to be useful for to some extent. As I told you, you are responsible for answering to the obstetrician from obstetric unit to your NICU for long-term prognosis. For obstetrician, it can be useful. Second thing, when you are clinically examining the child, 2 scoring systems are going to helpful for you. One is Cernat and Cernat, and another is Thompson scoring. Simple Cernat and Cernat is not, this is the scoring system that we use for asphyxia categorization as HI stage 1, 2, and 3. Here, just to simplify, this is a busy slide. I will just tell severe encephalopathy will be divided into Cernat score by these different parameters, where stuporous, comatose, no activity, decerebrate posturing, flaccidity is there, heart rate variability is very poor, apnea is there, dilated pupils are there. There is a poor prognosis in the form that baby is going to have disability or death. So by clinically examining the child, you can consider by doing that. Thompson did similarly. These are the different parameters which are available. Score marks given as 0, 1, 2, and 3. Maximum score available is 22. Lowest score is 0. You have to calculate this while examining the child. This is a clinical scoring. So you can tell in after stabilization of the child within 6 hours of baby's birth. So when you see that if the score goes towards towards 22, then poorer is the prognosis. The score goes towards 0, it is a very good prognosis. Score 0, baby can be normal. Simple scoring system, Thompson scoring system is there, where clinically you can examine and tell to the parents while doing counseling. Another part which hemodynamic instability, a lot of papers had been published for that. See, the basis for hemodynamic instability is that birth asphyxia leads to cause fall in blood pressure, delayed CFT or tachycardia. That is hemodynamic instability is there, or need of inotrope depending on, because asphyxia leads to brainstem damage as well as myocardial hypoxia or decreased myocardial contractility. That leads to parasympathetic stimulation and leads to bradycardia and hypotension. So if there is a need of inotrope, then prognosis of the baby goes poorer and poorer. If initial 24 to 72 hours, if there is a need of inotrope, it's okay. But need of inotropes beyond 72 hours is there, definitely it carries poor prognosis to the tune of 33 to 77% of poorer neurological outcome as well as mortality. Now, another parameter is lab parameter, that is pH, PCO2 and lactate. Before going into depth of that, just I will sensitize you why these are important. Because as pH falls, means acidotic pH will not lead your inotropes, internal inotropes, or your means like internal hydrocortisone is there, adrenaline is there, they are not going to act in acidosis. So as pH lowers, prognosis is going to be better because brain is less perfused, means blood pressure of the baby is going to fall. PCO2, when it falls, it causes cerebral vasoconstriction, which leads to cerebral hypoxia, and baby is going to have PVL-like changes, that is periventricular leukomalacia. So PCO2 is very important. Third parameter is lactate. Lactate, similarly, it lowers pH and doesn't allow the internal inotropes to act, so blood pressure falls. So whenever there is a pH fall, which is should be, see, there is a practice of doing. See, if you see the definition also, within 1 hour of birth of the child, to define birth aspect, you should have ABG in your hand, cord ABG. If pH is within 1 hour is low, PCO2 is low, then there is poor neurodevelopmental outcome. And lactate, it needs more time for normalization. If it is taking more than 24 hours of normalization, it carries poor prognosis. If you see the evidence, the odds of having cerebral palsy is 2.3% with confidence interval of 1.3 to 4.2%. The odds ratio is simply, you can tell that the babies with birth asphyxia with pH low have 2.3 times odds, means 2.3 times having cerebral palsy more as compared to those who are having with normal pH. This is an oversimplification of odds ratio. Confidence interval should be as narrow as possible to have a significance of that. Second, more advanced things will go about AEEG. AEEG, this is the picture which shows that it carry amplitude integrated EEG. How it is done? It is a very simple technique. If you are using hypothermia, you should have a AEEG technique in your unit. So we as a pediatrician can read AEEG very simply. Just you have to concentrate for 1 minute. So these green things are normal on AEEG. See, you can see this tracing. In this tracing, see this lower margin is more than this 5 microvolt and upper margin is more than 10 microvolt. So here is a 10 and upper margin is that. So all tracings are more than 5 microvolt in lower margin, and upper tracing, if they are going more than 10 microvolt, it is a normal AEEG. Second green thing is continuous normal voltage is there, or discontinuous normal voltage is there. There is a difference. See, this continuous normal voltage means lower margin is always more than 5 microvolt and upper margin is always more than 10 microvolt, then it carries a normal importance. But in between, if it is having abnormality, baby can have moderate abnormality in future. These all red pink things suggest it is a abnormal AEG. So what abnormal AEG is? Very simple to understand. You see the bottom line. This is a flat trace. The trace is very flat. Electrocerebral silence is there. If you find such kind of AEG, then there is a definitely lot of abnormalities are going to happen in the child. Second, see this trace. See, this is a low voltage AEG. See, the lower margin is below 5 microvolt, you can see. So in the normal, it is more than 5 microvolt. And upper margin also is not going beyond 10 microvolt. So it is a low amplitude AEEG, which also carries poor prognosis. And burst separation pattern is, you will have burst followed by suppression. So here you are finding that where upper margin is more than 10 microvolt, but lower margin goes less than 5 microvolt. So such kind of tracings, if you are getting, so you can blindly say you need not have to have neurologist to say that this AEG is going to be abnormal or normal. So see, I made it very simple to understand how AEG is to be read. So now how AEG is useful? So differently, less than 6 hours of life, if you are using, it is having basically high prognostic value if you are using less than 6 hours to less than 24 hours. But more than 72 hours, it does not have. At the end of this lecture, I will tell which investigation to be done at what time. So another is time of recovery in AEEG is prolonged, then definitely poor prognosis is there. So different values are there. See, at 72 hours of life, if time to recovery means baby's AEG is abnormal, then 90% of the babies are going to be mentally retarded, having cerebral palsy, or mortality risk will be very high. And time to establish sleep-wake cycle is long, then baby is going to have poorer prognosis. So these patterns on AEG like burst suppression, low voltage, and isoelectric tracing, that is electrocerebral silence, is going to have poorer prognosis. Simply this you have to keep in mind. Leave about the percentage. If you remember these three parameters on AEG, you can prognosticate the child. Then EEG wise also same. If there are low voltage, flat traces, and burst suppression patterns are there, there is a poor neurodevelopmental outcome is going to be there. So just don't see the report abnormal EEG or normal EEG. Just see few lines above that also, which suggests that there is a low voltage, flat tracing, burst suppression pattern. So if such words are there, definitely it is going to have poorer prognosis. Another thing is seizure burden in that. Seizure burden is calculated hour-wise also, how much maximum seizure burden is there and how much in hour seizure baby is going to have in last 24 hours. So like that, depending on also we can have that. So if you see neonatal seizures as such, अपने यहाँ क्या बोला जाता है, baby को fits आए तो baby is going to have cerebral palsy. It is not like that. See, कुछ ये line इसलिए मैंने डाला है, seizures and abnormal outcome have a controversial outcome in the form, because those babies who are having seizures can be neurodevelopmentally normal also. So whenever you are getting neonatal seizures, at that time, try to combine that with acidosis, burst suppression pattern on day 4 and MRI findings. If abnormal, then only these seizures are going to have abnormal outcome. So this study, basically this was a meta-analysis, and this is showing that total seizure burden and highest maximum seizure burden of more than 13 minutes in hour is there. Then basically it is going to have abnormal neurodevelopmental outcome. So other than that, only seizures are present, so p-value is not significant here. So basically what you have to consider is, if maximum seizure burden is high or TSB is going to be more, then there are more chances of poor neurodevelopmental outcome. Here also shown in this. So now going into evoked potentials. So different evoked potential, this picture is for calculating SSEP, that is somatosensory electric potentials. Then this is visual evoked potential and BERA, as we routinely do. So how they are important? So out of these three, only SSEP and visual evoked potential have some prognostic value, where they have more NPV values. Means negative predictive value is 93% with SSEP. That means that if baby's SSEP is going to be normal, baby is not going to have abnormal neurodevelopmental outcome. So to rule out cerebral palsy in future, this investigation is going to be more. BERA doesn't have any importance. It needs more and more studies for prognostication of a child for neurodevelopmental outcome. So NIRS is basically emerging kind of technique where you are going to find out the cerebral metabolism. Here like that, simply you have to apply the electrodes over the baby's brain, and you are going to find out how is the oxygen consumption on NIRS. So regional cerebral oxygen consumption, as it is elevated, that gives a poorer prognosis, means it also carries more negative predictive value, means it can confidently rule out that cerebral palsy is not going to happen if it is a normal regional cerebral oxygen saturation. Just I will sensitize about HRV, also you will come to know about prognostication. HRV is heart rate variability. Heart rate variability, you have to calculate the QRS complexes on ECG and how much they are changing from bit to bit. If heart rate variability is poor, then baby is going to have poorer neurological outcome. So this is little bit theoretical. Basically, I'm trying to make you more sensitize you all people basically for how you will be able to answer. So this MRI, basically, you should know at what time it to be done. MRI, if you are doing, you first advise diffusion weighted images or MR spectroscopy facilities are available. So in diffusion weighted images, you will find such kind of images. So this is watershed area where the two major arteries, that is anterior cerebral artery and middle cerebral artery, where they supply at the end part of that, you are going to have such kind of watershed infarcts. And if there is a hypoxia, near total or complete hypoxia is there, or cerebral vascular occlusion is there, then such kind of basal ganglia will be affected. So MR spectroscopy wise, it carries lot of importance if you are doing diffusion weighted images and MR spectroscopy in early part of hypoxia. So this is last but one slide I am going to put in front of you. See, as I have shown these MRI pictures in front of you, so Barkovic has made a few stagings for that. So they have given these scores, and these scores are given for basal ganglia abnormality, watershed area, and basal ganglia divided by watershed area. Depending on that, if you find more basal ganglia involvement, you are going to have motor and cognitive developmental defect in future. So you should be able to read about DWI, that if motor and cognitive defects are going to happen, basal ganglia are affected, and only watershed area are affected, then cognitive defects are going to happen, and that you have to monitor in the child. Now, this is the last slide where I want to tell you that we are, as a pediatricians, always confused at what time these investigations are to be done, because I have put around 10 to 15 investigations or prognostic markers in front of you. So out of that, when I have to do that? So on this y-axis, I had put that day 1 to day 5 and from day 7 to day 14. On this side, I had tried to put when or what investigation is to be done. So see, on day 1, when you want to prognosticate, the investigation useful will be neurosonography, MR spectroscopy. DWI is not going to be useful at that time. AEEG to some time. EEG is also not useful. So at day 1, you can do these 3 investigations. In addition to your ABGAR and clinical examination, they combiningly have a good prognostic value. Clear? So when you want to prognosticate, try to order these investigations, do clinical scoring. Which 2 scorings we have done? Cernat, and second is Thompson scoring. So these scoring systems you should do, and combine that with these 3 investigations, you can prognosticate with very good narrow confidence interval and very, very good predictive value. Second day of life, you can do MRS. Neurosonography does not have any role at that time, because neurosonography only tells about cerebral edema. Any intrauterine means PVL, cystic malformations or structural malformations are there, they will be there of no use after that. Second thing, day 2 of life, diffusion weighted images PIP in. At that time, you can do that, and AEG carries importance, and EEG also PIPs in that. After day 5, means when day 7 to day 14 is there, basically you should advise your conventional MRI. T1 and T2 weighted images you should advise, and you have to look into that. So whenever you are advising investigation, you should know at what time you have to advise. So I'm taking only 1 minute, ma'am. It's okay. So this is day 7 to day 14, T1 and T2 weighted images, and then you have to advise according to that. So simply you have to keep in mind that only single prognostic marker is of no use. You should be able to know at what time, what investigation to be advised, and combine that. Means day 1 of life, if you want to tell to your obstetrician, जो पहले हमने सवाल बोला था आपको कि उसको अगर एडवाइस आपको करना है, तो try to get one sonography done. Another thing to be done is MR spectroscopy and AEEG in combination with ABGAR score and clinical examination finding by Cernat and Thompson, and then prognosticate to the obstetrician. If you want to prognosticate after day 2 of life, then combine your clinical examination with other parameters like EEG, and you can consider for diffusion weighted images also. MR spectroscopy is not available at all centers, but diffusion weighted images they are easily available where MRI facilities are available. That's all I want to say. So I want to tell you the simple thing that not a single prognostic marker is not going to be of any use. You have to combine that, and time of doing investigation is very important. That's all. Thank you.
Thank you.
Any
Thank
question
you
is
very
there?
much, sir.
You can ask me.
Question answer we will take at the end of the session.
Okay. Thank you.
Sorry,
Sorry, this was very theoretical topic, but I tried to make it very
No,
simple.
sir, it's a very excellent guidelines that you have given. Stage, sir. I request our chairperson, felicitate our speaker. Thank you, sir. I further invite Dr. Sandeep Kadam from Pune on the topic, all is not sepsis. As an unwritten rule, as soon as a baby is brought to an ICU, the second thing that goes into the baby's veins after the intracat is an antibiotic. This is more of an habit than need. But we should keep in mind that every sickness in the newborn should not be labeled as sepsis unless proven otherwise. Dr. Sandeep Kadam will clarify further on this. Over to you, sir.
Respected chairperson, seniors, dear friends, after a delicious lunch, our blood supply to gut increases and brain decreases, and we are in the stage of postprandial hyperglycemia with stage of lethargy or drowsiness going into stupor. So what I'm going to discuss in the next 15 minutes is that neonatal sepsis, not always, which means that we are going to see some conditions which may look like infection but may not be infection. When I opened my third MBBS book of Davidson's Book of Medicine, the first page writes, when you diagnose rare things, you are rarely correct. So I think the first message that I want all of you to go out of this hall is, please don't forget to diagnose infection. My teacher told me, if I see 10 patients who look like infections, 8 out of 10 or 9 out of 10, you are going to be correct. So I think common sense tells me that if I put sepsis as my first diagnosis, I'm going to be 8 or 9 times correct, and only 1 or 2 times I may be wrong. So the purpose of today's presentation is to look at that neonatal infections or sepsis is the commonest diagnosis that we all sitting in this hall give to our newborns who get discharged from the NICU. We label different terminologies, maybe probable, suspected, culture negative, culture positive, so on and so forth. But that is the commonest diagnosis. Neonatal infection often have some signs and symptoms which mimic other conditions. And often we always overtreat and not undertreat, because we all realize that undertreatment is risky, time is precious, and if I don't give antibiotics in time, or if I don't give correct antibiotic, I may have a fear of losing that baby and the baby may die. So the purpose of today's presentation is to look at some of the clinical clues and assessment which will help us rationalize the medical management of neonatal infections. I'll start with my first case. This is a male child, term baby, born to a mother who was primigravida. It was a normal delivery. Weight of the baby is 3.2 kilos, cried immediately after birth. On day 3, this baby had increasing distress. Baby was not feeding well. The baby was transferred to NICU. There was no history of any mother having leak or maternal fever. This was a non-consanguineous marriage. There was no previous neonatal death or any abortion. So basically, this baby came to us on day 3 with increasing distress and not feeding well. On examination, this baby had tachycardia, heart rate of 170, respiratory rate was 64, capillary filling time was 4 seconds, peripheral pulses were not well felt, there were minimal lower intercostal retractions. The first and second sound was normal, there was tachycardia, upper limb blood pressure was 52 by 28, lower limb was 50 by 26, liver was 3 centimeters below the right costal margin, spleen was not palpable. There were minimal crepitation with air entry which was bilaterally equal. So basically the problem was that this baby presented on day 3 with shock and there was hepatomegaly. So any diagnosis? What do you think? Correct. This was the X-ray, which we could see that there was good lung expansion, there was no pneumonia. The heart size was normal. So the echo showed that this was a duct-dependent left-sided obstructive lesion. This was an hypoplastic left heart syndrome. The shock basically presented because the PDA started closing. Obviously, the prognosis was poor, and we offered treatment, but obviously surgery and all the long-term prognosis was not that great, so the care was withdrawn. So coming to cardiac disorders, which look like infections. So whenever you see a baby who has tachypnea without much distress, there is tachycardia, there may be tachypnea. Second sound is often single. There may be cyanosis or shock, which presents on day 2, day 3. Now remember one thing, that if you see a baby with severe shock on day 3, it may be a left-sided obstructive lesion. And if you see a cyanosis on day 3, it may be a right-sided obstructive lesion like a pulmonary atresia where the PDA is closing. Often the babies may have failed SpO2 test. Please remember that it is very important to palpate or examine the femoral artery pulses on day 3, day 4 of life, because lower limb pulses may be absent, and you may be able to diagnose a coarctation of aorta when the PDA closes. On X-ray, often you may see a cardiomegaly, you may see unusual size of shape of heart. Sometimes we often see a egg on side like a TGA, or you may see a coherent subway like a Fallot's physiology. But some of these tests obviously give us a clue that this could be a cardiac disease. Second child, 18 days newborn, referred from peripheral hospital for respiratory distress, fever, and abdominal distension. On evaluation, this baby was febrile. The baby had respiratory rate of 48. The baby had heart rate of 164. CRT was 3 seconds. Air entry was equal. There were no crepitations. Again, the first and second sound was normal. There was no murmur, but the baby had big liver and big spleen. So any clues or any diagnosis that you would like to give? This was a baby with big liver, big spleen, abdominal distension, high fever, respiratory distress. On evaluation, this baby had a hemoglobin of 6.4. Total counts was 3,200. Platelets were 23,000. There were no blast cells, no atypical cells. CRP was very high. Blood culture was negative. CSF and urine culture were negative. X-ray was normal. Ultrasound showed big liver, big spleen, no abscess, no free fluid. Skeletal examination on X-rays were normal. So basically, what are we looking at? We are looking at a baby who has got probably a late onset infection. That is what I would make a diagnosis of. But this baby definitely has anemia, leukopenia, and thrombocytopenia. So any diagnosis here in this case? Congenital infections. So obviously, that is a possibility that we would see. But here, mind well, what is the thing that I have not told you is the weight of the child. This was a term baby, and the weight was 3.1 kilo. So if it is a full term, appropriately grown baby, probably intrauterine infection may not be a possibility, wherein if I have a baby who is a small baby, IUGR baby, obviously I would think first in terms of a congenital intrauterine infection. 2D-co was normal, serum ferritin was high, triglycerides were high, there was pancytopenia. So raised ferritin, pancytopenia, very high CRP, high triglycerides. Bone marrow showed HLH. So this baby had probably a hemophagocytosis, which may be primary or secondary to some infections. IVIG was given, baby responded well to steroids. So probably a secondary HLH, which is quite well known. And we keep on giving antibiotics after antibiotics, and these babies may not improve. So please look for HLH as a possibility in pancytopenia or bicytopenia with high fevers and high CRPs. Coming to third case, day 6 newborn, term baby presented with high fever. There was no history of any leak or chorioamnionitis in the mother. Heart rate was 180, respiratory rate is 60, CRT is 3 to 4 seconds, weak pulses, no murmur. There was no difference in blood pressure in the upper and lower limbs. The pre and postductal saturations were 95%. There was no liver, there was no spleen. So again, I would like to make a diagnosis of sepsis. How many of you think it is not sepsis? Just because I'm talking about sepsis, not always. This baby had oliguria on day 8, generalized swelling, seizures on day 8, sodium was 122, potassium was 5.6, There was involvement of the kidneys, involvement of the lung, involvement of the neurological examination. So obviously 2 or more organ involvement tells me that this is multi-organ dysfunction. TLC was normal. There was low absolute neutrophil count. There was low platelets, high CRP. Echo was normal. COVID antibodies were positive. So this was multisystem inflammatory syndrome post-COVID. Again, the treatment would be IVIG. If the coronaries are abnormal, obviously we would give IVIG. If the coronaries are normal, when we give a trial of steroids or IVIG. So please remember that post-COVID MISC, you may have fever, multi-organ dysfunction, laboratory evidence of inflammation. You have to have a COVID antibodies which are positive, but most important, this is a diagnosis of exclusion. There is no other plausible explanation which you could give in terms of explaining this condition. So you have to rule out sepsis. So post-COVID MISC is a diagnosis of exclusion. The fourth case is a term baby weighing 3.4 kilo, male child, brought with history of excessive crying, weight loss on day 14 of life. On admission, the weight was 2.2 kilo. There was a weight loss of 1200 grams. This baby had sodium of 117, potassium of 8.2, renal functions were normal, sepsis workup was normal, cultures were negative, CSF was normal. So what am I looking at? This is the case of excessive weight loss, hyponatremia, hyperkalemia. Not thriving well. Adrenal dysfunction? Yes. The 17 OHP was very high. So this is the classical congenital adrenal hyperplasia. Baby would respond to steroids and was managed with fluorocortisone and hydrocortisone. Baby did well, and there was significant weight gain postnatally. So once in a while, if I see a baby with excessive weight loss, not thriving well, I need to look at CH as a possibility. You may get ambiguous genitalia, you may get abnormal genitalia, hyperpigmentation, but please remember that once in a while, all these genital manifestations may take a while, but hyponatremia, hyperkalemia, I would keep CH as a strong possibility. Coming to the next case. Term baby, 2.1 kilo, female child born with normal delivery, presented with fast breathing, lethargy, and not feeding well on day 3 of life. Baby had tachypnea, mild stupor, CNS examination was normal. Other systems, the respiratory, cardiac, and abdominal system was normal. X-ray was normal. Hemogram was normal. Septic workup done was negative. Blood gas done showed a pH of 7.1, PCO2 of 22, PO2 89, bicarb of 6, base excess of -20. The kidney functions were normal. Electrolytes were normal. So what am I looking at? Sugar was slightly low. Metabolic disorder. Ammonia was 500. So this is a case of severe metabolic acidosis, high ammonia, and the baby presented with tachypnea. That this tachypnea was more of metabolic acidosis, because of metabolic acidosis, that the baby was breathing fast. And this was a metabolic acidosis with hyperammonemia. The diagnosis turned out to be a propionic acidemia. So when do I suspect inborn error of metabolism? Whenever you see a baby where there is a history of consanguinity, previous losses, previous abortions or neonatal deaths, often metabolic disorders present like infections. They typically present after day 3, day 4 of life, not on day 1, day 2. Very rarely I would see a non-ketotic hyperglycemia presenting on day 1 of life, or a pyridoxine dependency presenting with seizures on day 1 of life. But by and large, metabolic encephalopathies would present on day 3, day 4 because they need to have a substrate. The sepsis workup may be normal. Often there is metabolic acidosis which is persistent, may or may not be associated with hypoglycemia and or hyperammonemia. Most of the protein metabolic disorders typically present with hyperammonemia and hypoglycemia, but you may have carbohydrate and fat metabolic disorders. So please remember that metabolic disorders are not uncommon, and if you suspect, I think we need to do a screening and we may find a metabolic encephalopathy. The next case is a term baby, 3.5 kilos, born to a mother who was priming. It was a normal delivery. This mother had prolonged labor. Baby did not cry immediately after birth. Baby required bag and mask ventilation, ventilated for 24 hours in view of poor respiratory drive. Baby developed poor perfusion, tachycardia, reduced urine output, required bolus. There was no murmur. Air entry was bilaterally equal. Peripheral pulses were not felt despite 2 boluses. The baby was initiated on dobutamine, dopamine, given adrenaline. Baby looked pale. What am I looking at? Myocarditis, a possibility, very good. Intracranial hemorrhage, very good. So this baby, problems in this baby was anemia, there was shock, there was asphyxia, query, sepsis. Hemogram and CRP were normal, blood culture was negative. Ultrasound showed cerebral edema, echo showed mildly reduced cardiac function, but there was hyponatremia, hypoglycemia, and hyperkalemia on day 3. That was what we saw in this baby. Any diagnosis? Now, these are all the patients that we have seen in our NICU. There is no hypothetical case. They were all managed. Excellent. So, ultrasound showed a big adrenal hemorrhage, started on hydrocortisone. The baby was weaned off inotropes and baby was extubated. So basically, shock was due to adrenal hemorrhage and cortisol deficiency. So please remember, if you see a baby which is full term and there is shock and is not responding to your vasopressors, so catecholamine resistant shock, I think we need to think of adrenal dysfunction or adrenal insufficiency. And one of the differential diagnosis may be a adrenal hemorrhage, especially in a breech presentation. If it is a breech extraction, adrenal hemorrhage is quite well known. This is a term newborn presented with blackish lesions on the scrotum on day 12 of life, was referred from outside to us. There was no history of any fever, seizures, or distress. The vital signs were stable. This baby, I will show you the photo, but there was ichthyoma gangrenosa which was present on the scrotum, and subsequently more and more lesions started coming on the abdomen and scalp. When I saw the baby on admission, my first diagnosis was Pseudomonas infection with ichthyoma gangrenosum. And I'm sure you could see these lesions which were present on the scrotum and the perineal region, and then subsequently on the abdomen. And you could see that the left hand was showing blackish discoloration. And there were multiple such scabs which were coming on the body despite giving meropenem, despite giving ceftazidime. The baby just could not respond, and the complications went on increasing. The scrotal lesions were so bad that the surgeon advised, I will do a escharotomy, but provided you first do a colostomy. So we ended up doing a colostomy because there was fecal contamination of the perineum, and that's why we did a colostomy. After colostomy, there was a lesion around the colostomy, then on the scalp. So I was just fumbled. What is it that I'm looking at? This was looking typically like a Pseudomonas infection, but the baby is not responding. So hemogram showed inflammation, CRP was 112, cultures were negative, CSF was normal, urine culture was normal, baby received colistin, meropenem, ceftazidime, there was just no response. INR was normal, PTTK was normal, D-dimer was 5,300, COVID antibodies were negative. Yes. So this was thrombophilia. This was a protein C deficiency, which was genetically proven. And unfortunately, this was a primary mother. And luckily, we could pick up a protein C deficiency, which would help them diagnose in the subsequent pregnancy. So this is a autosomal recessive hereditary thrombophilia presenting with conditions which looks like ichthyoma gangrenosum and multiple gangrenes. So to summarize, I just have 2 or 3 slides, and then I'm happy to answer some of the questions if you have. What did I learn from my neonatology journey? Sick or unwell baby, my first diagnosis has to be infection, 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 and infection. So please don't forget to diagnose neonatal sepsis. That is my commonest condition that I'm going to look in my day-to-day practice, which I don't want to see. If you have a clinically relevant risk factor, obviously my diagnosis will be very easy. If you have a mother with fever, chorioamnionitis, obviously early onset sepsis is present, quite explainable condition. But if you don't have a risk factor and you need to spend some time with the mother to ask the relevant history, and if you don't have a relevant risk factor in the mother and the baby presents like early onset infection, then look for birth asphyxia, look for metabolic or a cardiac disorder. Day 3, if a baby crashes or collapses, it has to be either a cardiac disease or it has to be a metabolic disorder. Unexplained tachypnea and tachypnea without distress. Please look for a inborn error of metabolism, or a cardiac disease with a left to right shunt. It could be PDA, it could be ASD, it could be a VSD. So tachypnea without distress, it has to be a cardiac disease, or it could be a metabolic disorder because of severe metabolic acidemia. Sometimes you may get unexplained tachypnea because of AV malformations, and once in a while, it could be a vein of Galen malformation presenting only with tachypnea and cardiac failure. You may not get hydrocephalus. If you see a febrile newborn baby and there is pancytopenia or bicytopenia, think of HLH or MISC. MISC, why? Because we have started seeing post-COVID effects and we have started seeing a lot of multi-system inflammatory conditions. So think of these possibilities, and I'm sure with all of us seeing so many clinical conditions, we would be able to pick up these conditions routinely. My carry-home message for today, for all of us and practice pointers is that neonatal sepsis is the commonest clinical diagnosis. We should be aware of common presentations. At the same time, there are some of the uncommon presentations which always present once in a while with neonatal infections, but not always. So the bottom line that I would like to remember today is, if you have a baby with probable, suspected, or culture negative infection, and there is no response to treatment, you are changing antibiotics 1, 2, 3, 4. I think it's time to review your diagnosis. Repeated sepsis screen, if you have done and they are still negative, think of an alternative diagnosis. And most important, interpret your laboratory test only in the shadow of your clinical picture and not otherwise. Don't interpret your clinical sign laboratory test. Often the parents tell me, आम्ही तुमच्या WhatsApp report पाठवले जरा आम्हाला diagnosis सांगा. पण आधी आम्हाला patient बघू द्या, मग आम्ही diagnosis सांगतो. I think it's the other way around. We all need to see our patients clinically, take a good history, and then interpret our investigations in the light of our clinical history and clinical science. I think if we start doing these, maybe the burden of laboratory tests, investigations, and finances for our patient care definitely will come down. I think the carry-home message is that think of sepsis, but once in a while, if you have a baby who doesn't respond to your antibiotics, please review your diagnosis and make an alternative diagnosis that it may not always be neonatal sepsis. Thank you very much.
Thank you. Thank you very much, sir, for the excellent session and giving us a way how to approach the sick neonate. May I request our chairperson to felicitate our speaker? A small announcement, lunch will be served only up to 3:30 p.m. So those who have not taken the lunch, please proceed to the food court. Thank you. Thank you, sir. For the next topic on surfactant, I invite Dr. Sanjay Ahe from Nasik. Surfactant is like a magic foam, the amrita of life, the surveyor given to us by God. Surfactant deficiency in preterm is an established fact. Dr. Sanjay Ahe, sir, will tell more about this. Over to you, sir. Thank you.
So good afternoon, everyone, respected chairpersons, all my seniors and colleagues. I welcome you all to City of Nasik for this wonderful academic feast, which is going to happen over next 3 days. So welcome you all. Can you show my first slide, please? So I'm going to discuss the updates or what is new in surfactant. So what I'm going to cover in next 15 minutes. So what is the role of prophylactic surfactant versus selective surfactant? Then what is the evidence about ensure, that is intubate, give surfactant and extubate? Then whether to give surfactant early or delayed surfactant? Then is there any role of synthetic surfactant against natural surfactant? Then there are various novel surfactant administration techniques like LISA, MIST, MISA, SALSA, and so many. Then is there any role of multiple doses of surfactant in premature infants? And any role of surfactant after 72 hours of life? Then there are new studies coming with intratracheal steroids along with surfactant. So you can add intratracheal steroids with surfactant and then give that surfactant. And what are the non-RDS uses of surfactant? So let's start with a clinical case. We had this baby on day of Diwali, 29 weeks, one of the triplet baby, born on 24th of October by LACS, 1100 gram baby, cried after birth, his Apgars were good. But he started grunting, so he was immediately started on bubble CPAP. But his respiratory distress was increasing, so non-invasive SIPPV was given to this baby on non-invasive machine. At 2 hours of age, baby was still oxygen dependent and requiring more than 30% oxygen. So decision was taken to give surfactant by a LISA technique using a rigid surfactant catheter. And you can see this technique of using this surfactant by LISA technique. So this is the same baby who was born at 29 weeks, weighing 1100 gram. He was maintaining saturations, but he was needing more than 30% oxygen. And you can see the baby had a significant distress. So this baby is on non-invasive ventilation. And we decided to give this surfactant while he was breathing without intubating this baby. So I used this LISA catheter using surfactant. So the beauty of this technique is you don't have to intubate the baby. The baby is breathing and you can visualize the vocal cords with the laryngoscope. As soon as you see the vocal cords, you put this surfactant catheter. You can use simple feeding tube also. Then you insert that tube up to certain mark below the vocal cords. Remove the laryngoscope and you can give surfactant while baby is moving, breathing. So you just give bolus surfactant to this baby. So you don't have to intubate. It is very less invasive as far as the baby's airway is concerned. So it will not cause any trauma to the baby's airways. And you are not using positive pressure ventilation while giving surfactant. So that will avoid barotrauma and volutrauma to this baby. So after giving surfactant, you just remove the catheter and continue whatever non-invasive ventilation you are doing, and baby goes back on his CPAP or non-invasive ventilatory support. So after this, you can see baby's mouth, if there is any excessive surfactant has gone out of the airway, but you can see all the surfactant has gone into the baby's airway, and baby is back on his non-invasive ventilatory support, and he was doing well. So he remained on non-invasive support for 2 more days. Then he was on high flow nasal cannula. He was tolerating feed and he's ready to go home tomorrow. So that's the beauty of giving surfactant by non-invasive technique. So we all know surfactant replacement therapy has been one of the major breakthrough in care of preterm neonates with RDS in this 20th century. So what is the role of surfactant? We all know that when we were not using bubble CPAP as a primary mode of ventilation, there is decreased need of ventilatory support, decreased risk of pulmonary air leaks, mortality, and combined outcome of death or BPD at 28 weeks without increasing adverse neurodevelopmental outcomes. But however, there remain number of questions related to how surfactant should be used with non-invasive respiratory support like CPAP and the availability of newer techniques of administering surfactant. So what is the role of, is there any role of prophylactic surfactant? So what do you mean by prophylactic? So strategy of providing exogenous surfactant at birth to infant at risk of RDS with the aim of preventing severe RDS. And what is selective use? It is a strategy of providing surfactant to an infant with established RDS. So the old trials before routine use of delivery room CPAP showed significant benefit of prophylactic surfactant in reducing morbidity and mortality and air leak in this preterm infant. But with recent use of delivery room CPAP, the recent clinical trials mandating routine use of CPAP has shown increased mortality, increased bronchopulmonary dysplasia at 28 days and 36 weeks post-menstrual, and BPD or death at 36 weeks with use of prophylactic surfactant. So instead of causing benefit to the baby, it is causing harm to the baby if you are giving prophylactic surfactant. In 3 recent trials where rates of antenatal steroids were high, more than 50%, prophylactic surfactant was associated with significant increase in BPD or death at 20 and BPD or death at 28 days. And there was increased mortality, BPD and BPD at 28 weeks. So level 1A evidence, that means you should stop using prophylactic surfactant. You should not give prophylactic surfactant. So these are the NNF guidelines published in December 2021. So prophylactic surfactant should not be administered to preterm neonates below 28 weeks. Preterm neonates with RDS should be stabilized first on CPAP, and if indicated, selective surfactant should be given. It is a strong recommendation. The clinicians may consider delivery room surfactant in subgroup of neonates below 28 weeks who get intubated in the delivery room for severe RDS. How surfactant to be used in preterm infants on non-invasive respiratory support? So there are different trials of using CPAP and then selective surfactant versus prophylactic surfactant. Again, prophylactic surfactant was causing more harm than putting the baby on CPAP and then giving surfactant if indicated. Then this technique ensure intubate, give surfactant and extubate. It reduces the need for mechanical ventilation and it decreases the incidence of BPD or air leaks. So compared with delayed selective surfactant and ongoing mechanical ventilation, we have this study. So this was a meta-analysis which shows that the risk was less if the baby was given Ensure after putting the baby on CPAP. So this study suggests that use of nasal CPAP shortly after birth as primary mode of respiratory support is an acceptable alternative to elective intubation and administration of prophylactic surfactant. When to give surfactant? Early versus late. Early means below 2 hours and delayed means after 2 hours. So there was a meta-analysis of 6 randomized trials where early surfactant was associated with significant decrease in mortality, BPD or death, reduction in pneumothorax with no increase in the risk of pulmonary hemorrhage or severe intraventricular hemorrhage. So NNF guidelines says early Ensure within 2 hours may be used for preterm infants below 34 weeks with established RDS who satisfy the criteria for surfactant administration. I'm going to discuss which baby has to give a surfactant. Then what about natural versus synthetic surfactants? So there are 3 types of synthetic surfactant: first generation, second generation, and third generation. So this phase 2 trial of third generation synthetic surfactant that includes DPPC and analogs of surfactant protein B and protein C. The name of the surfactant is CHF5633. Is currently underway. It is a multi-center RCT which compares this synthetic surfactant with poractant alpha. First generation and second generation surfactants are out of market. Nobody is using those surfactants nowadays. Then there are novel surfactant administration techniques which we saw just now as a LISA technique. But surfactant can be given with various other ways, like pharyngeal instillation. You just put the surfactant in the baby's pharynx, and some of the surfactant baby will aspirate, some will go to the baby's esophagus. Then another way is surfactant administration through laryngeal or subglottic airway. It is also called as SALSA. Then various thin catheter surfactant administration techniques like minimally invasive surfactant treatment that is called as MIST. Then surfactant without endotracheal tube, that is SURE. Then minimally invasive surfactant administration MISA, and less invasive surfactant administration technique LISA. And surfactant can be given by nebulization. So there are various newer techniques coming up where we can cause less harm to the baby. Then we already saw this technique of Hobart method, which I demonstrated. Then another way is using simple infant feeding tube. This is called as a take care method, where we can use our feeding tube, maybe 6 or 5 number feeding tube can be used. but you might have to use Magin forceps to put the baby through the vocal cords. So this is a simple technique we can use, and we can keep these catheters in the freezers to make them little bit hard, and then we can insert those catheters. And Dr. Sandeep Kadam has done a study in KEM hospital about this. So this is another way of giving surfactant. So here we have compared various methods of non-invasive surfactant administration. So pharyngeal instillation, the amount of swallowed and inhaled surfactant is unknown, and it will increase prophylactic surfactant. Then supraglottic airway, again, cannot be performed in infants below 1250 grams, and it is not currently recommended unless it is a research study. Then various thin catheters, nasogastric tube, vascular catheters, manufactured catheters can be used. But here you need skills, you need laryngoscopy, Magid forceps. There can be surfactant reflex, so lots of wastage of surfactant. And sometimes you have to sedate these babies to keep them calm. Then surfactant by aerosol or nebulizer technique, mainly studied in cases with mild RDS. Then long duration of application, not enough data in infants below 27 weeks. So less invasive surfactant administration or LISA techniques are superior to ensure strategy in preterm neonates below 34 weeks gestation with RDS. So we have this meta-analysis which compares LISA with Ensure, and here they have shown that it was better to give surfactant with LISA, and all these parameters like BPD, mortality, and requirement of mechanical ventilation, pneumothorax was less with LISA. This was a very recent article published in September 2022, where they have compared this newer technique, they call it as a surfactant without endotracheal intubation versus surfactant with intubation. And this was a meta-analysis. So 21 research articles were eligible for this study, and there were around 20,000 participants, and data showed significant reduction in composite outcome of stage 2 NEC, BPD, and onset of hemodynamically significant PDA when treated with surfactant without intubation. There was trend towards lower rates of pneumothorax when surfactant was given without intubation, and there were no difference in the outcome of death or rate of other neonatal morbidities. So overall, this SURE was identified as a better substitute for intubation and then giving surfactant in neonates. So LISA may be preferred or ENSURE for surfactant administration in preterm neonates below 34-week gestation, but the recommendation is weak because the person who is doing this LISA must be properly trained to use Magin forceps while using the feeding tube. And in settings where dedicated LISA catheter is used, they should be trained regarding the depth of insertion. And sometimes we have to use pre-medication, but it is debated. What about outcome of these babies who received surfactant by LISA below 27 weeks? And this was very recent. Early November, this study was published in JAMA. And total 6,542 infants were analyzed who were below 27 weeks. 38.7% of these babies received surfactant by LISA. And 53.6% of these babies did not require mechanical ventilation in first 72 hours. So it is very significant. So considering 27 weeker and not requiring ventilation in 50% of the babies is amazing. So there was a reduced risk of all-cause death, BPD and BPD or death. So all p-values you can see, they are below 0.001. Then supraglottic airway, again, I have mentioned that it should be used in only research scenarios. Then what about the dose of surfactant? How much surfactant should be given? So this is a comparison of porcine surfactant, 200 milligram versus bovine surfactant, 100 milligram per kg in below 34 weeks. And again, you can see the dose more than 200 milligram per kg was superior to bovine surfactant at 100 milligram per kg. What about multiple dose of surfactant? So multiple doses of surfactant should be used in treatment of preterm neonates who satisfy predetermined criteria for additional surfactant dose. Then what are the indications of repeat dose of surfactant? So here we can have two categories, high threshold and low threshold. The high threshold means we can accept MAP of more than 7 and FiO2 more than 40 versus FiO2 of more than 30%. And in higher threshold, that is MAP requirement of more than 7 versus and FiO2 of more than 40% may be used for repeat dose of surfactant in preterm neonates below 34 weeks. So if any baby who has received first dose of surfactant and still he is needing more than 40% oxygen after 8 hours, you can consider second dose of surfactant. What about surfactant after 72 hours? So surfactant is not recommended beyond 72 hours, irrespective of prior surfactant therapy. So there is no role after third day in these babies. So don't give surfactant after 72 hours. We have this evidence which shows that there is more complications if you are, or there is no advantage of giving surfactant. Then this is very recent trial where they have studied intratracheal administration of steroids, that is budesonide with surfactant in very low birth weight infants to prevent bronchopulmonary dysphagia. So this was in Turkish Journal of Pediatrics. So surfactants were instilled into trachea via an endotracheal tube using a orogastric tube, the same thing what we do routinely. The treatment group was given mixture of budesonide, 0.25 milligram per kg, along with 105 milligram per kg of Infaserp. They used that type of surfactant. And the control group received only surfactant. So early intratracheal administration of budesonide with surfactant in preterm infants with severe RDS might decrease BPD and mortality without disturbing surfactant function, but we'll need more studies in this criteria. Then what about surfactant use in other conditions other than RDS or other than this HMD? So there are various conditions where surfactant has been tried, like meconeme aspiration, pulmonary hemorrhage, pulmonary edema, oligohydramnios, pneumonia, various types of aspiration like blood aspiration, amniotic fluid aspiration, bile acid pneumonia, and so many things. But only meconeme aspiration syndrome, pulmonary hemorrhage, pulmonary edema, there are some evidence to use surfactant. So what about surfactant in meconeme aspiration? So one systematic review included 3 small trials of surfactant diluted with saline and used for lavage in term and late preterm infants. No difference in mortality, need for ECMO, development of pneumothorax, duration of ventilation or length of stay was demonstrated. However, surfactant lavage was associated with reducing the combined outcomes of death or need for ECMO. What about neonatal pneumonia? For neonatal pneumonia, some clinicians may choose to administer surfactant based on similar principles, but there is no evidence to date to support the practice. Then in pulmonary hemorrhage, 2 small observational studies have suggested that administration of surfactant after pulmonary hemorrhage may improve infant oxygenation index. So there are 7 take-home points from this talk. In neonatal care settings where CPAP is routinely used, and the rate of antenatal corticosteroid is more than 50%, prophylactic surfactant is no longer recommended. It is a grade A or very strong recommendation. Non-invasive respiratory support, example CPAP, should be provided to preterm infants with RDS from birth, and early surfactant should be provided for newborns with increasing severity of RDS. Then infants with RDS whose oxygen requirement exceeds 40% should receive surfactant replacement therapy. Then intubated infants with RDS should receive surfactant before interfacility transport. So you have to give surfactant and then stabilize the baby and then transfer the baby to higher center. Then repeated dosing of surfactant should be provided to infants only when there is evidence of ongoing moderate to severe RDS. For spontaneously breathing infants on CPAP with RDS, non-invasive methods of surfactant administration such as LISA or MIST are preferable, or INSURE technique. Surfactant replacement for infants with meconeme aspiration or pulmonary hemorrhage may be considered at clinician's discretion. So thank you very much for patient listening.
Thank you. Thank you very much, sir, for your excellent talk. I will invite our chairpersons to felicitate our speaker. I also invite Atul Dasputre, sir, to felicitate our chairpersons. Dashputre sir, please.
Thank you, Dr. Sanjay Ahir, for providing valuable information. May I invite Chairperson Dr. Rajendra Patil and Dr. Sagar Wag on the stage. May I welcome Dr. Samir Dalwai from Mumbai to present his next talk, that is red flags in behavioral pediatrics. Often in our day-to-day scenario, in our busy OPD, we find something odd in few children. While entering the OPD, I find the toddler sitting unusually quiet and just staring at nothing. I get suspicious. When another kid during examination picks up my stationery, throws it around, bites his mother, or creates a nuisance, I get alert. And then comes the mother who complains that a young one doesn't respond when called, or she has to repeat multiple times and yet fails to understand. I get worried. In clinical terms, there are few absolute indicators, often referred to as red flags, that identify the behavioral or developmental markers, suggesting the need for further evaluation. A wait and watch approach is not to be considered here, because early intervention is the key for the great possible outcome for children with such issues. Sir, the stage is all yours.
I welcome Dr. Samir Dalwai, sir. Actually, sir needs no introduction.
Just
He's
a
the
small announcement.
national and international speaker. Yeah, I will write one announcement, please. I have just received the Dr. Jayashree Nadkarnia Oration Award in Madhya Pradesh on 12th of November. I congratulate, sir. Thank you.
I just a small announcement. The people sitting on the right side on the chairs, can you just shift on the left side? So there will be the space for the persons who are coming. It's getting difficult to go on that side while lecture is going on.
Yeah. Dr. Samir Darvi is a developmental and behavioral pediatrician, recipient of the Government of India Rajiv Gandhi Mano Seva Award for the services of children in 2012. He is a national treasurer of IEP and editor-chief of IEP Handbook of Developmental Pediatrics. As all of you know, the children, they need some economical or social support or behavioral support. If you don't observe their milestone, they may land up into some psychological trauma to the parents. So I welcome Dr. Samir Dalvi, sir.
Thank you. Shall we begin? Thank you, Namaskar, and thank you to the chairpersons and the organizing committee for giving me an opportunity to meet all of you on this topic, red flags in behavioral pediatrics. And straight away, I would like to make the point that when we talk about red flags, it's important to remember, for example, if we take hyperactivity, when does it become a red flag? At the same time, what is very important is how to arrive at a clinical diagnosis. There's no point just remaining restricted to what's a red flag or what is not. My request is very humbly to the people in this row, at least the last part, Dr. Bashir and everybody, if you can shift there, then people will be able to sit here. Just a humble request, if you don't mind. In the last 2-3 rows in this column. Thank you so much. So the whole idea is not just to identify red flags because they're there in all the textbooks, but more importantly, how do you arrive at a clinical diagnosis? The disclaimer is that none of this talk is based on any textbooks or any theory or any papers. This is all from the clinical experience in the last 20 years that I've had working with a team of wonderful people at New Horizons Child Development Center. And the numbers that we've had the fortune to see and help in the last so many years is huge. I mean, this data from 2009 to 2019 itself, around 7,000 children and adding. So what I'm going to take the permission of the honorable chairpersons is to go down on the audience because we'd like to have an interactive session. Is that okay? All right, so great. So let's talk, and I'll move down here, and I have a mic with me, and I'll see that we can all of us work together. Hello. Right. So this is the first case. I'll come across. Please feel free to get up and or don't get up, sit and just say what comes to your mind. 4-year-old child comes to your OPD for cough, cold, fever. Mother says, Doctor Saab, आज के आज टेस्ट कर लो. जे अँटीबायोटिक द्यायचे असतील ते द्या. डॉक्टर येवले काय म्हणतात ते जाऊ दे. मला ताबडतोब अँटीबायोटिक पाहिजे कारण शाळेतून ऑलरेडी खूप कंप्लेंट्स येत आहेत. I don't want to run into any more trouble with the school. What do you think is the kind of complaints this mother is getting? Samir.
So if complaints are coming, means something might be hyperactive, not attending, and destructive, something like that way.
That is because Samir Sadavarte is a very dear friend of Samir Dalwai, and if he thinks Samir Dalwai anywhere, he thinks it's only hyperactive. So the point message is everything is not developmental or behavioral.
She has come with the other complaint. This is just a presenting complaint, respiratory. But
Excellent.
inside-
So this is basically a child who just has cough, cold, fever, and maybe that is why she's missing attending school regularly, and that's what the parents or the teacher is worried about. So what important lesson is don't brand something as developmental or behavior when you start with. Go through the history. When you ask the mother in detail, the school counselor told her that the child is not doing well. He is rather hyperactive. मस्ती खूप करतो, बसत नाही, पण शिकतो मात्र. पोएम वगैरे, ABCD वगैरे, 1, 2, 3, 4 हे सर्व उत्कृष्ट करतो. Like other children, no problem. He does not seem to have a learning difficulty. आता काही शब्द मी ठळक किंवा इटॅलिक्स मध्ये किंवा इन्व्हर्टेड कॉमर्स मध्ये टाकलेत कारण हे आता सवय झालेली आहे. अरे हा की नाही हायपर ऍक्टिव्ह आहे, हा की नाही नॉटी आहे, याला लर्निंग डिफिकल्टी वाटते. त्याचं लर्निंग आणि डिफिकल्टीचा काय अर्थ आहे ते कोणालाच माहित नसतं. आपण काउन्सिलर झाले, टीचर झालेत, हे शब्द आपण वापरायला लागलोत. म्हणून ह्याच्यात पुढे काय करायचं? व्हॉट विल बी युअर ऍडवाइस? रिफर द चाइल्ड टू अन ऑक्युपेशनल थेरपिस्ट. आजकाल शाळेतच आहे ते, म्हणजे शाळेची फी भरा, मग थेरपिस्टची पण फी भरा, मग काउन्सिलरची पण भरा. और refer the child to a psychologist, or refer the child to a developmental pediatrician, or start some medications. What do you think you will do? Come on, everybody can speak. None. Very good. Will counsel. Further detail investigation or questions are needed. Don't do this, because this is what I say. Forget about hyperactivity. All of us in schools and parents have become hyper about any activity. Let's not get hyper ourselves. So what do you advise? Other point. अरे कुछ नहीं है, सब बच्चे ऐसे, लड़के ऐसे होते हैं. उसके फादर को पूछो, वो भी ऐसा ही रहा होगा, मैं भी ऐसा था. तो अभी ये क्या सब बोल रहे हैं आप? ये डाउटफुल होता है कि कुछ मत करो, because I don't want to take any responsibility. Who has time for these hyperactive children? I have 20 children waiting for vaccination after this. Or आजकल all ज्ञान is not needed. After some years, there won't be conferences like this. There'll be only Instagram discussions. Because people look at Instagram and come up with all knowledge. Osho ka kuch ye, some psychologists share something, and they feel, okay, we can share this. The point is, it's very important what Dr. Pramod Kulkarni said. That is what I call hypo about activity. कुछ नहीं करने का. देखेंगे, hopefully by the next time the child comes, he will not have any complaints, or mother may forget or she'll understand not to ask me some questions. ऐसे होता है ना? असं होतं ना? का? कारण आपल्याला कोणी क्लिनिकल अप्रोच शिकवत नाही. आपण या मोठ्या मोठ्या टर्म्स मध्ये कन्फ्युज होतो, आणि आता काय तिथून येते शाळेतून. काउन्सिलर स्टड युअर चाइल्ड इज लर्निंग डिसेबिलिटी. तुम्हाला काय वाटतं? मला काय वाटतं? मला सर्दी खोकल्याच्या बद्दल विचारा. म्हणून हायपो अबाउट ऍक्टिव्हिटी. बट वी हॅव टू बी लाईक शर्लॉक होम्स, and we are curious. Sir said, so we'll ask for details. Now, when the mom speaks to the counselor, you tell her, go to the school, ask the counselor in detail. She says the child is hyper for the first half an hour, then settles down. Remember, this is a child who's not doing badly in studies. Hyperactive hai, jam shararti hai, but is hyper for the first half an hour, then settles down. Now what do you say? Now what do you think? First time half an hour ke liye hyper hai, then he has settled down. Bola. You can be hyper. Bola. Yes, but from here, what do you think? Sorry? He could be hypoglycemia, fine. And otherwise, he is anxious about school. Why in 2022 would children be anxious about school? 4-year-old child. He has missed school for the first 2 years because of COVID. And this is a child who is getting separation anxiety. At what age we heard separation anxiety in our medical college when we read? 9 months, 10 months, 11 months. These children are getting it 3 years, 4 years. And खरं separation आत्ता व्हायला लागले. So it's possible that he has separation anxiety. Or The child has been hyper since day one. त्या कोविडच्या अगोदर पण 15 दिवस शाळा चाललेली तेव्हा पण हायपर ऍक्टिव्ह होता आणि आला तर throughout the 3 hours he is hyper active in the class till he is taken out in the play area. Now what do you think? Sorry? Least interest in the school. Maybe the classroom is very boring. Maybe the classroom is very boring. Once he goes out to the play area, he is happy. So maybe this is a child who doesn't get much opportunity to play in the house. Maybe he is just looking for an opportunity to play. So when he gets a chance to play, his hyperactivity goes down. He is not hyperactive on the playground. He is hyperactive only in the classroom. This is all the data we have so far. This is a child who is hyperactive in the classroom for 3 hours. Bus. We don't know what's happening. We know that in the playground, he calms down. Then, like sir said, what about home? Is he calm at home? But before we go that, look at the other option. He goes berserk in the playground. He's worst when he goes out. Is this anxiety? Unlikely. Is this a child who has separation anxiety? Unlikely. This is a child who has hyperactivity in multiple theaters. Everywhere he is hyperactive, giving him a chance to relieve his anxiety or play or give him an open area. He doesn't have to feel confined. Even in an open area, he still feels hyperactive. So what do? Now the action shifts from the child's school to the child's home. You ask the mother more details of home. Mother says, "Mera bachcha, mera lal." दाभोळकर साहेब बोलतात मला भेटल्यावर, मेरे लाल, मेरे काले, मेरे पीले, मेरे नीले. तसं तुम्हाला बोलतात की नाही भेटल्यावर? नाही बोलत. ठीक आहे, आम्हाला बोलतात. If the mother says, "My child is amazing at home, he has no problem," it is the school counselor who is all the problem. She creates trouble. तिला माझ्या मुलावर सूळ काढायचं, whatever. What do you think? Same child, hyperactive at classroom 3 hours, goes outside, is berserk in the play area. But mother says, "Nahi, che. Gharat." What do you think? Assess the details of his hyperactivity at home. A mother is saying, "He's not hyperactive." Authoritative parenting. आता आपण parenting. The point is, ask one question. Really? Does everybody really love him? Because in India you have this. For the mother, 10 खून भी किया बच्चे ने तो मेरा लाल सबसे अच्छा. So be very sure whether you are getting the history. So the history has to shift beyond the home into the neighborhood or the relatives. And this is what the neighbors say. The child is very active. Everybody does love him. Now what do you think? That's the neighbors also love him. Everybody likes him. Complaint is only from the school. This is where you have to really wonder, is he really hyper in the school? Now see, your attention goes back from the school, from the home, back to the school. What is happening in the school that this child is behaving so difficult in the school? Who is he going with? Is he going in a school bus? The child may be a gifted child, but more important than that, is the child having any sexual abuse, physical abuse? What's happening in the school bus? What's happening at daycare? What's happening when the parents leave the child in the creche? Is there something happening in the school? It could be that the first day the teacher shouted at the child, "What are you doing this?" Once the child got shouted, he Gautam Buddha principle. Grown up as Prince Siddharth. Suddenly एक बार बाहर जाके जिंदगी की reality पता चल गई, दिल टूट गया. Child has never been scolded in the house. Granny, grandfather, everybody loving. Is this the case that suddenly somebody scolded him in the school? So you have to check what's happening in the school. Then ask the mother more details. What about the friends? This is a different case. The friends are afraid of him. The peers are afraid of him. Nobody wants to call him to their birthday party because last time he went to a birthday party, all the children were playing. He went and cut the cake and ate it also before the birthday child could cut the cake. Now this is very important because now you have a child who's hyper at multiple places. Then you need to know, is this an issue of parenting at home, like somebody said? Is this the kind of parenting at home that everything is allowed, permissive parenting, do what you like? And hence when he goes in other situation, he thinks, "मैं तो नवाब हूँ, घर में नवाब हूँ, तो बाहर भी तो नवाबी करूँगा." And people don't accept you being a nawab everywhere else. But this is what the child may be carrying. The point is, don't jump to a diagnosis of ADHD. That's what I'm trying to say for the last 10 minutes. What is happening at home is very, very important. And I'm sure all of us have had many such lectures on parenting, so I won't go into it. The point is, my child complained that is hyperactive. Oh, you have ADHD, go to a therapist. Ye nahi karna hai aaj se. Going ahead, some scenarios I will describe. Mother says that the counselor told her child is hyperactive, but otherwise doing well. learns what is being taught, he does not have any learning disability. What are our thoughts? This is what we have to look for. First, the home environment, we have discussed that. Just make a mental list because you know all of it. मी नवीन काही सांगत नाहीये. हे सर्व माहित आहे, फक्त don't miss this. Home environment, is there any abuse and neglect at home? Is there any recurrent illness? The child who has recurrent illness will be irritable when he goes to a school. Does he have any issues related to malnutrition? We saw this morning wonderful talk by madam. More important, what are the details? This is what Dr. Kulkarni sir was saying. Details. And when you ask the details, begin with details of the child at home. The big misconception is my बच्चा घर में शैतान है, स्कूल में जाके परिंदा बनना चाहिए, फरिश्ता बनना चाहिए. Cannot happen. That's the biggest mistake parents do. Hey, डॉक्टर मेरे बच्चे को हाइपर एक्टिव क्लास में नहीं ले रहे, आप थेरेपी दे दो ताकि क्लास में बैठे. अभी थेरेपी दे दो के लिए शैडो टीचर, मतलब वो बच्चे को बिठा के पकड़ के रखेगी. ये थेरेपी हो गया, पेरेंट छूट गए. ऐसा हो रहा है कि नहीं हो रहा है? हो रहा है कि नहीं हो रहा है? They just don't want a complaint from the school. Shadow teacher is sitting, another 10,000 rupees being paid. पकड़ के रखो बच्चे को. अरे 10,000 रुपया क्यों खर्चा करता है? बांध के रखो ना बच्चे को. रस्सी से बांधो, 20 रुपए में रस्सी आता है. Because that's all the shadow teacher is contributing intellectually to that child. She's not helping the child. You are the one who's missing the chance to help the child because you are not going through this list. What can we do? Schedule. Most important, what time does your child wake up every day? What does your child do after that? Is he into a schedule? नहीं. मन में जैसा है करता है. जब मन में लगे उठता है, जब मन में लगे खाना खाता है, जब नहीं खाना नहीं खाएगा, दादी बाद में लाके खाना देगी. If the child is used to this kind of behavior, how do you expect him to regulate his behavior in the school? So don't call it ADHD. This is what I call it. Call it a poor self-regulation. Call it a poor self-regulation because in your mind, ADHD treatment refer to XYZ. What it should be, self-regulation. Can I teach the parents how to help the child to self-regulate? The action shifts to you. The power comes to you. Don't give up your power by referring to others. Adequate sleep, does he get or no? You all know the advantages. Does he get enough outdoor play? Very important about responsive parenting. We did so many ECD workshops, so you are aware about it. But if the child is not better with this with you in 3 months, then it may happen that you are missing something. At least after 3 months, then think that you may refer this child. So is this clear? Simple scenario. Scenario 2. School counselor told her child is not doing well in school. He does not seem to learn what other children are learning. Why have I added this other line? What other children are learning? Why is that important to ask? Sorry. One is comparison. The second thing is if the school itself is terrible, none of the children will be learning. That happens in some parts of India. You may not have a teacher, you may not have a classroom. So very important to note this. Are other children in the classroom doing well? Because if everybody is doing badly, maybe the teacher is bad, maybe the school is bad, maybe the syllabus is bad. Nowadays there is a fad. SSC medium not fashionable. SSC जमत नहीं है, IGCSE चालू करायचं शाळेत त्यांना. हो रहा है कि नहीं? SSC नहीं जम रहा है, कैम्ब्रिज बोर्ड चालू करना है. क्यों? पेरेंट्स कैम्ब्रिज मांग रहे हैं, डबल फी देने के लिए तैयार है. वही टीचर, वही कपड़े, वही बुक्स, वही क्लासरूम, कल से कैम्ब्रिज चालू हो गया. Be aware about all this. So he seems to have a learning difficulty. What are your thoughts? If you have a child like this, now what will you do? He does seem, he is suffering. He is not picking up academically. अब क्या बोलेंगे? ADHD, learning disability, dyslexia, dysgraphia, कुछ तो बोलो, autism. Very good. Some said vision and hearing. Yes, sir. Does he learn at home? You have to, fantastic. You are the developmental pediatrician. Fantastic. Does he learn at home? Don't outsource this into a scholastic performance. IIP is doing all this poor scholastic performance. School में क्या हो रहा है? अरे स्कूल गया तेल लगाने. घर में क्या कर रहा है वो कौन पूछ रहा है? Poor house performance या IIP program होना चाहिए. Is the child behaving normally at home? And are you parents behaving normally with the child at home? Do you have time for the child? Do you sit with the child? Do you have a schedule for the child? Or are you coming and going arbitrarily and expecting the child to be a military disciplined general at home? Then he will never be good at school. Remember this, ask about history at home, what the parents are doing, and hence inquire and examine, go ahead, don't just give a diagnosis. So again, go back to the child's birth history and developmental history at home. If you have the birth history, all of us know what is uneventful birth history. But if it is eventful, ask for all these things. Did this child, was he preterm? Was he low birth weight? Did he have a stormy NICU course? Or did he have seizures? Was he ventilated? Birth asphyxia? Now the picture will, so far whatever we discussed, and now with all this history, the picture will change. For example, developmental milestones, either they are age appropriate or they are delayed. For example, global delay, specific delay, only speech is delayed, only behavior is abnormal. Motor development is all normal. Or deviancy, he has repetitive behaviors, echolalia, वही बात repeat करता है, गोल गोल घूमते रहता है. Correct? Correct? So if you take this developmental history and birth history, you will have the following scenarios. 1. Scenario 3, the child has history of only speech delay. Scenario 4, the child has history of come on, wake up, of global developmental delay. There are two possibilities for this child, either only speech delay or only global developmental delay. Is this fine? What was the complaint? Child is hyperactive, does not learn in the school, like we discussed. Now, how will you go ahead? Find out whether he has only speech delay or global delay. If he has speech delay, this is one scenario, 3A. अरे वो तो बात करता है, सबके नाम बोलता है, नर्सरी राइम बोलता है, नंबर्स बोलता है, he repeats names of people. हाँ, but if you आप बोलेंगे जाओ बाहर के रूम से टेबल उठा के अंदर लेके आओ, 4-year-old child should be able to follow 2 steps, 3-step commands in the family language. He's not able to follow those commands. He has poor eye contact and does not respond to name call as expected for his age. Now what do you say? Come on, what do you say? He has a problem with social behavior. Very good. What else? He has a problem with communication. I'm so happy none of you said autism. I love all of you. I'm happy you didn't say autism. What is very important is to look at the clinical picture. Don't go to the diagnosis. What we have seen is, again, inquire, examine. First, like the gentleman said, what is your name, sir? Vision and hearing. For every child, you have to test this. And the second is simple test, which we have come up with. Check SRK sign. What is SRK sign? At least the ladies can tell me. Shah Rukh Khan sign. This is it. What is this? When Shah Rukh Khan does, what happens? What is the response? Kajol comes, running and hugs him. Why? Why? Is he offering some money? So what does she see? She understands that he's putting out his arms, asking her to come and hug her. And she understands that this is an emotional kind of a call from somebody I love. And she understands that, and she knows the right response is to go running and hug him. Kader Khan won't go and hug him if he does like this, right? So what it means is, normal children, when they grow up, they understand this, that if mother puts out her hands, if daddy does like this, the child will come running and jump into the father's hands. Or if the mother does like this and calls out the name, the child will come running and hug the mother. Simple sign of normal development. We fail to watch it. Because if this is absent, then you are worried about more than a communication problem, doctor. You are worried about a social interaction problem. The child is not connecting socially. The difficult American word for it is poor reciprocal socialization. ये सुन के फी तो मैं ले सकता हूँ, लेकिन कुछ फायदा नहीं, किसी को समझ नहीं पाएगा. Poor social reciprocity, सब छोड़ दो. Child does not reciprocate to the love that somebody shows by putting out their arms or calling the child out. He does not understand this. This is a problem. So suspect autism, counsel the parents to spend more and more time with the child and put the child away from screens so that the child can understand parents' behavior. Of course, you may need to refer to a developmental pediatrician, but as a pediatrician, monitor. Don't refer and forget about it. Going ahead quickly, these are the red flags. By 6 months, 10 months, 12 months, we all know about this. And then a simple thing just to make it fun. Please, exam going students, don't write it in your papers. Nobody has described this, is just my imagination. Just to make a topic very, very simple, and so that you can understand it, that the child who does not respond to these non-verbal gestures of communication has some problem. So going ahead, this is scenario 3B, again, speech delay. But look at this case. He doesn't speak anything, neither repetitive nor otherwise. He does not follow commands, but he has a good eye contact. However, he does not respond to the name call. He has a good eye contact and he's very, very good with gestures and pointing. He's able to do this. Fantastic, fantastic. So the point here again is, you've got the story. Don't miss vision in this child. This is a child who is a patra for only specifically speech therapy and hearing aid. None of the others are. Only this child. And hence, to differentiate the child who started from having a poor hyperactivity at school, poor learning at school, see where we have come. Now you have become an expert at judging whether he has autism or whether he has a hearing impairment. Going ahead, suppose this is a child not of speech delay but of global developmental delay, motor delay, everything. What will you do? In this child, what's very important is to look at whether he has dysmorphology. Examine the child. Going ahead, whether he has neurological deficit. I'm rushing a little bit because we are getting out of time. This child, when you examine him neurologically, scenario 4A, He's unable to keep up with the peers. In a group, he gets lost. He doesn't know what to do. The other children जेवढे smart आहेत ना, तेवढा हा माझा मुलगा smart नाही आहे. त्याला नेहमी सांगावं लागेल. त्याला 1:1 करावं लागतं. पण त्याला घेऊन बसले ना मी, की तो बिचारा शिकतो. शिकल्यावर विसरत नाही. पण त्याला नेहमी बार उसे सिखाना पड़ता है one-on-one. And nothing significant on medical history. As if all of this is political history. This was medical history. Please don't have these differentiations in your mind, because what we call as developmental medical history could be eventful perinatal history, history of seizures. Dr. Sanjay was just now telling you about all those things about surfactant and all that. We know all of this, that he has history of seizures, dysmorphic features, that is 4B. In 4A, if he has nothing significant perinatally or obvious genetically, what will you think? What do you think? No specific developmental history, nothing obvious on the genetic syndromology. What do you think? Again, let's not go to a diagnosis. This is a child who is functioning slowly. He is not very bad in one area and very good in the other area. He is equally bad in everything. But, when you give him attention, time, and one-on-one, he is doing better. So this is a child you will suspect. Again, don't jump to intelligence. First check vision. First check hearing. Even a child with hearing impairment can present like this. Poor adaptive skills. He cannot adapt. त्याला घरी शिकवलं पण प्रश्न शाळेत जरा वेगळा विचारला. He is not able to adapt to it. Suspect global developmental delay and likely intellectual disability. Counsel parents की हो त्याला वेळ जास्त लागेल. धावू नका. The years are going ahead. Don't be in a rush to put him directly in class 1 after COVID. These children will need more attention to develop them as per goals, step by step, one-on-one. You don't need to do any evaluations except if you want a genetic diagnosis for the next pregnancy. And this is the case that I wanted to say. And you are the one who has to monitor it. Because this is a child we have at New Horizons. Same story, 8-year-old child. Went to multiple pediatricians, multiple developmental pediatricians, multiple schools, multiple special educators, occupational therapists, speech therapists. सबने हाथ साफ कर लिए. Turned out to be a simple case of fragile X, because that is the history that he had. That was scenario 4B. 4A. Now, if you look at 4B, in addition, this child has what my previous friend speaker described, all those difficult things in the NICU. In this case, what is important is, again and again, vision and hearing. This is the child you will need an EEG. This is a child you will need an MRI. This is a child you need genetic evaluations. This is a child you need many medications. And you will have to refer him to pediatric neurologist also, because this is not only developmental. You also need to look at the neurological problem. And of course, everything common to all of us is you keep monitoring the child, whether he's doing better or no. Finally, no history of developmental delay. Last case, scenario 5. No significant birth history, no developmental delay, no dysmorphology, examination largely normal. But what are his complaints? Look at what his complaints are. Some confusion with right and left sides, what is above, what is below. Some confusion with spoken language, ask ko aks bolega. Some confusion with learning alphabets, B ko D, P ko Q, PAT ko TAP. One second. Simple Indian English words में बोलो. किधर गड़बड़ लग रहा है? IQ ठीक लग रहा है? IQ ठीक है? IQ ठीक है. Does he seem to have a significant perinatal disorder? Significant genetic disorder? Nothing. Does he have a significant problem with communication? No. Does he seem slow? No. His only problem is purely reading and writing and academics. Purely, he has no problem with socialization. He's positive. He's very good. Shah Rukh Khan says he goes and hugs everybody. He has no problem on responding to the call. He doesn't need one-on-one all the time. He only and only has a problem with reading, writing, and maths. Then after that, we'll start using words like dyslexia. So going ahead, what we need to do is, again, vision and hearing, no investigations, no MRI. There are people who have made fortunes doing MRI out of learning disability children. What we need to do is refer to it for evaluations, especially after 7 years of age, not at 4 years. But what they need is not remedial education. They need an entire program which includes behavior modification, which includes academics, which includes emotional concerns. So child has a reading problem, sent to remedial educator. Speech problem, sent to speech therapist. Behavioral problem, sent to occupational therapist. For God's sake, stop doing that. And we are doing it because we don't know how to approach these children clinically. Now we know. It's not very difficult at all. You just need to keep this approach. So you need to monitor, counsel the parents. So what will we do? If you have hyperactivity, the approach is this simple. Take a simple history. You know all this. Go through the history of the first 1,000 days. Dr. Phadke madam spoke about it. Look at the perinatal part, and very important, look at the school. But when you look at the school, within the school also, the mnemonic can be ABC, academics, behavior, communication, but never ever make the mistake of asking about academics first. Always ask about behavior first. If the child's behavior is not good, he will not have good communication and academics. If behavior is good, ask about communication. If communication is not good, academics will not be good. But if behavior is good, like in case 5, communication also good, like in case 5, only the problem with academics, then it's a different diagnosis. And hence, it's very important to have the same approach that Dr. Y.K. Amdekar sir tells us. Don't look at diagnosis, don't look at references, look at each and every symptom in detail. Unfortunately, nobody has taught us developmental pediatrics like this. We have been taught about fever, how to approach fever. Abhi wo point program kuch hai, ye program hai, wo program hai, fever, cough, wheezing. Everybody tells us clinically jaundice, but nobody tells us. And very important, how do the symptoms look in combination with each other? It's very, very important. And I hope today all of us have got that confidence that it's not difficult. This is very easy for all of us. I'm sure we all do it. What's very important is what shall we do? IAP is running the ECD program. It's available in your city. Please, I'm sure you must have done it. All of you know what we talked whole day, nutrition. And that's the whole thing. Let's weave the magic with the logic. All logicals, decrease the screen time. If required, send for a developmental screening. Look at the safety and security of the child. But if you refer to anybody, if you refer, मजाल है कि कोई diagnosis के बगैर आपके पास भेज दे. If you refer to somebody and they also refer to an OT or a speech therapy then they are a policeman. They are not a doctor. They are traffic police. You go right, वो असरानी सर का dialogue है शोले में, आधे right में जाओ, आधे left में जाओ, बाकी मेरे पीछे आओ. If the doctor you're referring to does not have a diagnosis, don't get fooled. I had a lovely discussion with Deshpande man in the morning. Don't get fooled. If they are saying refer to OT for what? What is the OT is going to do in 3 months and what will be the outcome in the child in 3 months? Tell me, then I will refer. Or tell me your working diagnosis. So this is what we need to demand from them. Take home messages, finally, do not be in a hurry to live. Do not be, all these children are getting messed for 15 years now, 10 years. All of you have been attending these IAP talks on developmental pediatrics, on ADHD, on LD, autism. काही फरक पडतो का? फरक एकालाच पडलाय. More number of children are being referred for therapy. Therapistच्या जीवनात खूप फरक आहे. सायकोलॉजिस्टच्या जीवनात खूप फरक आहे. कदाचित पेरेंट्सच्या पण जीवनात फरक असेल, कारण तो घरी आल्यावर ते मूल सेन्सरी काहीतरी करत असतं. म्हणजे शेप सॉर्टिंग, कलर मॅचिंग, काहीतरी स्विंगिंग करतात. त्याला टार्झन बनवायचं आहे का? त्याला शेप सॉर्टिंग, आता म्हणे बाळ तर कसं आहे तुमचं, एकदम इम्प्रूव्हमेंट आहे डॉक्टर का? तो शेप सॉर्टिंग करतो आणि पझल मॅच करतो. म्हटलं तुम्ही सकाळपासून शेप सॉर्टिंग किती वेळा केलं ताई? नाही केलं. मी पण नाही केलं. तुमचे शेजारी करतात का? तुमचे आई बाबा शेप सॉर्टिंग करतात? तुमचे काकी पझल मॅचिंग करते का रोज सकाळी उठल्यावर? हे करायचं काय? तुम्ही गेलात डॉक्टरांकडे, कारण माझं मूल बोलत नाही, तो लक्ष देत नाही, माझ्याकडे येत नाही, आणि ते म्हणतात हो, म्हणून त्याला बॅलन्सिंग शिकवते, शिडीवर उतरायचं कसं शिकवते, एका पायावर बॅलन्सिंग शिकवते आणि स्विंग्सवर बसवते. त्याला टार्झन बनवायचं होतं का झालं तुम्हाला? काय चाललंय काय? तुमच्या मुलाला प्रॉब्लेम काय आणि तुम्ही ट्रीटमेंट काय देताय आणि त्याच्यावर सांगताय की ह्याच्या ट्रीटमेंट इम्प्रूव्हमेंट आहे, अजून एक 7-8 वर्ष किमान लागतील तुम्हाला. म्हणजे एका पायावर नाही, पाय न ठेवताना बॅलन्स करण्यासाठी लागतील कदाचित. हेच होत आहे. आणि म्हणून जेव्हा आपण म्हणतो की XYZ डिसऑर्डर हॅज नो cannot improve, does not become normal, please make sure that nobody is building a business out of it. Don't make a business out of it. It's not going to get normal, but keep coming to me for 10 years, then we will see. That's not true. So we need to understand, don't use these labels, don't fall for these marketing gimmicks. These are all marketing gimmicks. Go to Instagram, go to Facebook, you will see, oh, your child has learning difficulty. Oh, your child is a slow learner. Oh, your child is not doing well in school. Oh, he has a speech problem. Come to our speech program. There is no blessed speech program. It's all about development and it's all holistic. So don't mistake symptoms for the disorder. So this is Crigler-Najjar and jaundice are two different things. Okay, don't ask me about Crigler-Najjar more. I won't know, but I know that it's a proper specific disorder. It's not the same as jaundice. So hyperactivity is not a diagnosis. I have seen children being referred for an evaluation, and they come back with a diagnosis: the child has delayed speech and language disorder. Delayed speech and language. म्हणजे पेशंट डॉक्टरकडे येऊन आई म्हणते माझ्या मुलाला ना ताप आहे. डॉक्टर म्हणतो ओके, युअर चाइल्ड हॅज फीवर. हे ट्रान्सलेशन झालं, डायग्नोसिस नाही. स्पीच बोलत नाही मुलाला, बाहेरच्या कंपाऊंडरला पण माहित होतं. तुमच्या आईच म्हणाली बोलत नाही माझं मूल, मग त्याचा डायग्नोसिस लिहून तुम्ही काय देता की डिलेड स्पीच अँड लँग्वेज डिसऑर्डर. काय चाललंय? सो प्लीज बी अवेअर अबाउट दिस. It's very easy for you to analyze the symptoms, match the approach, look for the comorbidities. It's not just the hyperactivity. Is there a speech problem? Is there a global delay problem? Is there a hearing problem? Is there a reading and writing problem? And hence do not initiate symptom-based therapy or intervention, because that will only be a rough work. रफू म्हणतात आपण डार्निंग. म्हणजे चाइल्ड विथ ऑटिझम डज नॉट मीन त्याच्या शर्टमध्ये 5 होल आहेत आणि रफू करत बसा, तसं नाहीये. ते रफू चक्कर होईल हे केल्याने. नका करू असं. प्रॉब्लेम एकच आहे. हे सिम्पटम्स अॅनलाइज करा आणि त्याच्यातून आपण अजून पुढे पुढे शिकत जाऊ. स्वतः बघत जा मुलांकडे. आपल्याला लक्षात येईल की प्रॉब्लेम इज नॉट सो डिफिकल्ट. लुक ॲट इट फ्रॉम सिम्पटम पॉइंट ऑफ व्यू अँड अप्रोच इट. सो दिस अगेन acknowledgements wherever I have learned it is on 2 platforms. One is my own center, where for the last 20 years with all these wonderful therapists, psychologists, special educators, other developmental pediatricians, remedial educators, this is where I learned. Fortunately, I did not learn in America. Fortunately, I did not learn this in a university. This was not even from the WhatsApp university. This was in the university of the clinic. But the second credit is to IAP, because for the last 12, 13 years, the ability to interact with pediatricians and share this knowledge, that is my lab. But the platform of learning, this is the classroom. Because I don't know how much you all have learned, but every time I interact, I understand better in the subject, and I suggest you do the same thing. Every time you see a child with hyperactivity, speech delay, or a learning problem, focus on what's happening. Call each other, call me, call another friend, look it up. Tell them to do something and tell them come back. Come back and tell me what happened. 1 month later, 15 days later. If it is not getting better, then surely refer, but don't refer and outsource. This is not my child, which is why after 12 years, 14 years of all of us working on autism, our collective knowledge is not much better. Because these children, my friend was telling me in the morning, देशपांडे मॅडम म्हणत होत्या, 50% मुलं परत येतच नाही आपल्याकडे. त्यांना सांगितलं जातं तुम्हाला औषधं देतील, वैक्सीन देतील, the वैक्सीन देने की वजह से ये प्रॉब्लम और बढ़ेगा. 50% of the children we refer don't even come back to us. How will we learn? If you refer a child to Dr. Amdekar, the great man, tells them मी तुमच्या डॉक्टरना सांगतो काय झाले ते, त्यांनाच जाऊन भेटा. Every child gets referred back to you, you learn. But you refer a child and he doesn't come back to you, you are not learning. Is there an agenda? Why is it that people don't want you to learn? Why is it that they just want you to send and not learn? Question that. So going ahead now, this is the book by Indian Academy of Pediatrics. It's available on JP. It was published or released in Pedicon. And finally, on behalf of Central IIP, we invite you for the 30th International Pediatric Association Conference along with the Pedicon to be held in February. And Chetan Bhai is also here. Please stand up and you can welcome everybody for the Pedicon, which is going to be held at Bhuj. We can hear you.
On behalf of Central IIP and organizing committee of IPA Congress, as well as our 60th Diamond Jubilee Pedicon, I invite all of you to our Mahatma Mandir venue at Gandhinagar in our Gujarat state. And as a representative from Gujarat state, it's my humble request and enjoy the hospitality of Gujarat. So, and there are a lot of other attractions too with the conference. Of course, we have a lot of international faculties as well as cream of our national faculties. So do come, do register at earliest. Thank you so much.
Thank you. And thank you everybody for a very patient participation. Thank you very much. Wish we could have done it slower, but thanks a lot. Thank you very much.
Thank you.
Thank you.
Thank
Back
you
to
very
the
much, sir, for the
chairpersons.
interactive session. So beyond the stage, I will request our chairpersons to felicitate Dr. Dalvi, sir. I will request Dr. Tanaji Khanade from Nasik to felicitate our chairperson. For our next session, I invite Chairperson Dr. Shyam Khandelwal from Aurangabad, Dr. Madhuri Kulkarni from Kolhapur on the stage. We are fortunate to have Dr. Camila Rodriguez from Mumbai as a speaker. Now zoom on to a deadly infection and scourge of mankind, Mycobacterium tuberculosis. We are in an advanced stage of removing this deadly disease from the face of the earth. So the antibiotic stewardship of anti-tuberculous drugs must be chosen judiciously. To know more about this, we have here Dr. Camila. Over to you, ma'am.
Thank you, and thank you for inviting me, Sharmila. Thank you very much. It's such a tough act to follow, Dr. Samir Dalwai. Really great talk. I'm actually going to quickly go through something that's kind of boring at this time, but I'll try and make it interesting. This is the tests that we have for TB. You have tests for exposure. You have tests for active disease. Believe me, tongue swabs are going to be the in thing next year. It's going to be the year of the swab where you can actually take a tongue swab and take out TB from that. Of course, we know about CAD and the second generation Fujilam from urine. Now I'm actually going to speak about the lab tests. And yet we all know that just 1 in 3 TB is bacteriologically confirmed. And sadly, 1 in 3 DRTB patients are put in treatment. Burden in children is huge and unrecognized. And just one point, overall 55% of estimated children are not being diagnosed. Yeah. So effective antibiotic stewardship, right drug, right dose, right time, right duration, because it improves outcome, decreases resistance and costs. This is the dedicated band of men who gave us the cure for TB. And sadly, we have failed to extend their achievement because, like I said, of the 1 in 3 patients who receive drug-resistant treatment, only 59% are cured. These are the new drugs. We have to preserve them and carefully guard them, bedaquiline, delamanid, and pretomanid, and the repurposed drugs, linezolid and clofazimine. So when you look at this seesaw, these drugs are on one side, but we have to be very careful how we inform our fellow doctors, the quality of drugs and how we dispense them. This slide, for those who are not in hospitals, who don't have an antibiotic stewardship, this is how we actually do it. The physician is in the center, and the rest of us actually report to him to allow this stewardship to happen. And guess what? For TB, the government of India has a really nice bedaquiline delamanid enrollment program. So if you want to put a patient on delamanid, a committee has to actually sign off. You can make that committee in your own hospital, but you have to have a committee who will sign off the prescription of these drugs. So it's not just about the regimen. It's before treatment, doing rapid diagnostics, knowing your surveillance, during treatment, optimizing PK/PD and duration counts. This is the drug susceptibility testing landscape. It's huge. And we are all familiar with Expert and TrueNat. This is LPA. But in 1 or 2 slides, I'll talk about targeted next-gen sequencing because I totally believe this is the way we're going to be. We're already doing it and submitting our results to WHO within 3 months. This will be a test, as you'll see. So the biggest impediment we all know has been the long wait for the right test and the poor accuracy of smear microscopy and the weeks to months long time for culture has really been the Achilles heel in TB. And we know about poor old LJ, and liquid cultures like midget are definitely better. And we did a study several years ago, about 15,000 samples, where we showed if you did put your samples on liquid culture, it was 9 days versus 33 days on LJ, and even for extrapulmonary, 13 days faster than 13 days versus 37 days. And not only that, it also grew better. But there are huge disadvantages. Today in TB, I really feel we no longer can wait because this is phenotypic testing. It takes time, it's demanding, it's expensive. You need to have a dedicated lab, etc. And you know what? The saddest thing is we have been, I hate to admit it, but the WHO and the global world has now admitted that we have been doing something that's really wrong. We have been testing at one single critical concentration that is known as the binary concentration. And this is what we just put out this year. Critical concentrations of rifampicin, the most significant drug in TB, has been set to high. That means that we have been missing patients. We have been calling them, the proportion of them as of R as S. And similarly for several fluoroquinolones, kanamycin, ethambutol, the breakpoints have been the critical concentration. There have been no breakpoints for TB. WHO is trying hard now, and a couple of months ago, they actually changed the breakpoints. But it's time to usher in a new era from the snail to the from the snail or tortoise to the hare. And clearly rapid and accurate tests is really fundamental. And TB, you have to really do drug susceptibility tests on the patient sample. So you know about expert MTB, 75% sensitivity in children, but they have limitations. You don't know the exact mutation. When you do RIF, we have a fair amount in our hands. We have about 14% of RIF sensitive that are INH monoresistant. In kids, we cannot pick up too well paucibacillary disease, and these you needn't worry about because in children, we're not seeing that much drug resistance. So the Expert Ultra was actually developed to improve the sensitivity. It's really a much better test. It can look at the way it has actually improved sensitivity compared to the old Expert. Overall 5%, in smear negative 17%, and in HIV positive 13%. And this is the whole range of differences, how it is better, because it has many more targets to pick up TB. So it really works well for pediatric TB. So please ensure that when you ask for an expert, it's not an expert MTB, it's an expert ultra MTB, because the amount you're detecting is 16 versus 131 organisms per mil. And the good, the bad, and the ugly of the next generation ultra is that if you look at pulmonary TB, it's really better performing for GL, sputum, nasopharyngeal aspirates, and stool. And the improved sensitivity is about 73% versus 66%. And in stool, the WHO meta-analysis actually reported a sensitivity of 53 and a good specificity. Whereas in GL, it was slightly higher, about 64 and 95. Oops. We all know the Hain. I'm not going to bore you with the details, but this is a test that is done by the government of India, and this is, we've written several publications how to interpret this particular test. If you have this mutation, you can't give INH, you can give ethionomide. If you have this mutation, you can't give ethionomide, you can give high dose isoniazid. So we have ways to actually tell you which drug you can give as per the LPA test. But the limitations of LPA are its inability to tell you all the mutations for resistance. This is the new kid on the block. This is the XDR TB. I don't know if any of you are using it. It just takes 1.5 hours. We are trying to apply it and do a feasibility study on children, but it takes, after RIF resistance, it's a great reflex test for INH, fluoroquinolones, and second-line injectables. And this is our Government of India management of DRTB. So we are actually doing it pretty well. We are doing all patients NAT, and when RIF resistance is not detected, we still do an LPA to look at monoresistance. And this is a pyramid that I've just made. So we've done this, this, and this. So I'm not going to bore you with this, but this has been a really outstanding contribution by the WHO. They've come out with a mutation catalog. And if you look at the gaps in the alignment between diagnostics, and here's diagnostic stewardship, and here's antimicrobial stewardship. Right test, right patient, right time, for the right antimicrobial and the right interpretation. So let's look at 3 cases that I'm going to show you. You have the right test for the right patient. This is Dr. Soman's case. This was a 2-year-old little girl treated elsewhere for fever, chest infiltrates, and was started on first-line AKT. 2 months later, she presented to us, to our hospital, with drowsiness, intractable seizures, ptosis, miliary TB, and tuberculomas, even in the spine. The child's mother was empirically put on HRZD for 5 months and was not responding. Regrettably, she didn't have a cough. The mother's sputum, the child's GL, and CSF was sent, and this was the mother's X-ray. All 3 samples were smear negative, and we couldn't do an LPA. So now we had this baby who was dying. And we had this mother with this chest X-ray. We did something known as pyrosequencing, and we could detect XDR TB in 6 hours. So what did we do? Our pediatric department put her on these medications and referred to ID for optimizing treatment. She was put on cycloserine and ethionamide. 4 weeks later, this was proven from a culture isolate, this was proven with the Hain test, and the XDR was subsequently confirmed both in the mother and child. And the good part is that if we had not believed that pyrosequencing report, I can assure you the baby would be no more and the mother would be much sicker. And I think this was the magic of technology today. It isn't us who did it. It was the physicians who believed in the technology, who put the treatment on that 6-hour test. Because the dilemmas in managing TBM are huge. You have drug resistance vital to exclude in Mumbai, where we have it's a hotspot. You have something known as a paradoxical response. And this always happens when you're weaning off the steroids, the patient does worse. And if you ramp up the steroids again and it is DRTB, you're in for trouble. Of course, there's adequate drug penetration issues. I mean, drugs like ethambutol don't even get into the CSF. And there are a variety of others. This is the penetration of the drugs. INH, rifampicin, and ethionamide are the best. And drugs like rifampicin, ethambutol are low penetration into the CSF. This was what we published maybe 2 years ago on CSF pyrosequencing to diagnose TB meningitis, and we found it had an excellent sensitivity to pick up indefinite cases. This is the second case, the right test at the right time. This was a 17-year-old female. She was treated with various first-line drugs, second-line drugs, twice in the recent past, including she had received clofazimine. The sputum, the gene expert showed RIF resistance was detected. She was severely breathless, cough, emaciation, and with high-grade fever. This was her CT scan, which just shows you that one side was practically gone, and the LPA showed that she had pre-XDR. Surgery was considered, but she was simply too sick to withstand the surgery. What treatment would we give her till then? So this is just to, I won't bore you with the details, but believe me that what we are doing currently is only detecting this much. I'm not even going to spell it out. We need to wait 4 weeks to find out the susceptibility to these other drugs, because this is what we need to know upfront. And today it's possible because we're doing something known as targeted next-gen sequencing. And just look at this, from LPA to targeted next-gen sequencing, this is how the result comes out, and all these drugs are there. And you can actually give this sheet to the clinician. You have read what you can't give, and all these drugs, linezolid, bedaquiline, clofazimine, capreomycin, you can give this patient. Not only that, this tells you that this patient had potential mixed infection, mixed infection was not detected, and the lineage was something we all fear, that is the Beijing type. So this is what we're looking out for. Targeted NGS directly from the patient sample takes 2 days, much cheaper than midget. It's faster than whole genome, but the disadvantages are possibly there are less information available yet. I look at it like this. In another 5 years, this is how we will be diagnosing TB. All TB patients will be put on expert, RIF resistance detected. We will then do one of these very fancy looking tests, which can be done easily. And we will tell you, based on a validated consensus mutation catalog, high level resistance, that is a mutation, don't use the drug. Low level mutation, use with higher doses. If it's just a variant that is not doing anything, drug can be used, and various other combinations will be able to be said. Because drug-resistant TB is a model example of precision medicine in ID. Currently, we are not so bad here. We are doing modified treatment and trying to be here somewhere. But this is where we need to be, because this is a therapeutic continuum and we have to perfect it because it's faster, precise, and far more effective. This is Tanu's patient, and this just shows you the case 3. This was a 5-year-old girl, fever for 1 month, no other symptoms. She was MAN2 positive, and as you can see here, she had necrotic nodes. The only history that she could get was 2 family members died of TB after long treatment. Nobody else knew anything. So she was a suspect of query DRTB. And the possible next steps were, should we give her first line? Should we attempt to biopsy the node? Should we do a CT biopsy? But none of them were possible due to various reasons. It was not feasible for the hilar node. EBUS was not possible due to age. Open biopsy was considered too invasive. And BAL was unlikely to be useful because there were no parenchymal lesions. And this very long chart shows you what happened to the child. She was started empirically on HRZE. Week 3, no response in fever, and her weight started decreasing. Luckily, sad to say luckily, but luckily she got a node which was biopsyable, a supraclavicular node. Histopath, expert, and TB culture were done. Month 2, expert showed MTB was, RIF resistance was there, and midget within a month came positive from this node. And the pyrosequencing that we did then showed that this was XDR TB, which was borne out on midget. But guess what? Though genotypically she was XDR TB, she was doing good. She was afebrile, 6 weeks, sustained weight gain, and there was partial regression of the nodes that we could make out. She was clinically fine. 6 to 9, she had seen about 3-4 pediatricians in the city, and everybody said, no, she's doing good. Let's continue with first-line drugs for 12 months. On month 10, she again restarts her fever, and the CT shows disease progression, and the child was then treated after 10 months for XDR TB. So the questions we really couldn't answer were, this was XDR TB, but why did it take 10 months or 9 months to show failure? This was possibly mixed infections, 2 subpopulations, and it took 9 months. How do we improve the detection of mixed infections? Either you do it in the lab like this. This is some patient's lung we took out, and believe me when I tell you, from each of these lesions, we could recover 4 types of mycobacteria when we actually genotype them and sequence them. So it's not just one. In patients who are failing treatment, you may have several strains infecting the patient. This is my last case, and this is basically the right interpretation. This was an engineering student who just got into engineering college, and he came to us. He was being treated elsewhere as pulmonary TB. 2 months later, he developed an abscess in his left palm. The pus was drained, ZN smear was positive, and the midget culture was negative. 4 months later, he developed an abscess in the right ankle. Some new drugs were added. He developed an abscess in the right forearm. Smear was positive, and the doctors were doing it right. They were sending it for midget. Every time it was no growth. 3 months, he developed an abscess from his right forearm. Again, the same story. So when he came to us, he came on a wheelchair. His father was desperate. Do something. My son has not been to college for 6 months. And they are all saying, this is drug-resistant TB. What do I do? Please help us. So what did we do? We took that pus and we ran this pyrosequencing. And to our shock, we found that no mutations were there. This was actually a drug-sensitive TB. But everybody, because he was not responding, everybody thought he was getting more and more resistant. They were adding more and more drugs. The boy was getting sicker and sicker. And eventually, we diagnosed malabsorption syndrome. All we had to do was increase his dose of rifampicin, and he dramatically improved. So finally, friends, if you see Expert Ultra, you have these tests, you have Expert XDR, you have pyrosequencing, you have targeted NGS that's here. We've currently just submitted about 400 DRTBs to the WHO, and this is being done globally. Once the WHO approves this test, targeted NGS will be available for all of us to use, which will tell us about every drug. Not only that, it will tell us about whether it's mixed infection or not. And the best part is, it's only going to take 2 days to give the result. How cool is that? It can be done directly from sputum. Currently, we haven't even looked at other sites. And the cost, I think, will be cheaper than doing a midget DST. Finally, TB innovations mean little if they cannot save lives. And this just shows you, by the time a patient seeks care, look at all these blue. He comes to the lab, he searches for the lab, he produces his sputum, he collects the report. This is what happens in the lab. But look at every maroon circle. It takes so long for a patient to finally get cured, and most of us are not cognizant of this. For me, I will only look here. For most clinicians will just write the treatment. But there is a lot that goes into helping a TB patient really recover and finally getting cured. So I'm going to stop here. Actually, I've got a couple of minutes. I'm just going to end with the last case. This was a right test with the right dosing. This was an adult. But just to show you, this was a 47-year-old male. Pain, he had severe pain in the back. He was losing weight. His current weight was 75 and he was 85 earlier. Biopsy showed normal bone. He didn't have RIF resistance. He was sensitive TB. 4 months later, he started having pain again. So this was extension to the iliopsoas, and you can see the abscess has really grown. This was Dr. Soman's case. And what we thought, he actually fit the criteria of identifying patients who can benefit from therapeutic drug monitoring. And this is work that's actually being, which you know that in the serum at a dose of 600, the value is 13.8, and in pus it can actually go down to 0.4, as it did in this patient. Dose of rifampicin was increased. The patient had symptomatic relief with abscess shrinkage by 30%. As you can see, it was so big, 30% in four weeks. So underdosing leads to suboptimal dose concentration and drives resistance. So this is something really interesting that we've come across, that when we actually give our patients INH and rifampicin and tell them, "Sube le lo," actually INH and rifampicin are antagonistic to each other. Because it depends on how INH is being metabolized. Indians are generally slow acetylators of INH. So INH remains for longer in them to antagonize rifampicin for a longer time. So all we did was, now we give our TB patients INH and the other drugs in the morning, and we'll give them rifampicin little later at lunchtime. So we separate out the dose so that the antagonism is not obvious. And this is what we've shown, that this is done by a biochemistry group. And they've shown that look at the patients who were in subtherapeutic range and look at the patients who were in the subtherapeutic range who developed resistance. I mean, this is really scary, because if you are not able to control when you give your drug treatment, if you're actually pushing for resistance while you're treating your patient, this is really a sad story. Thank you.
Thank you, madam, for enlightening us on tuberculosis. It was a very good and precise session.
Thank you very much, ma'am, for your informative lecture. May I request our chairperson to facilitate our speaker? Simultaneously, I request Dr. Vaibhav Pustake to felicitate our chairperson. There is one announcement. Our mixed bag session, we are going to conduct at our Tapon Hall. That is another hall. Here, after this, we are going to start with our capsule session.
I am Dr. Pradeep Gujar. I lost one bag. The bag is like this. Dhirubhai Ambani, something is written on that. It contains my some personal material. So whosoever got it, kindly either deposit on this counter, or that contains one QMR. That QMR on very first page, my phone number is written. Thank you.
Here we have a MMC observer, Dr. Bhaskar Jethe, sir. I invite sir on the stage. Yes, sir. Thank you. Simultaneously, I invite our organizing chairperson, Dr. Ramakant Patel, sir, on the stage to felicitate Dr. Bhaskar. Sir, stage. Thank you, Bhaskar, sir. At the fag end of the day, we will be taking up some quick 4-minute short yet powerful snip, and the speaker will have to enlighten us in a really short time limit of only 4 minutes. I request all our 8 speakers to sit in the front chairs. I would like to invite moderator for our this session, Dr. Pramod Jagg sir from Pune. I invite our first speaker. Oh, Dr. Sarita. So our 8 brilliant speakers will going to highlight a very important points on 8 different topics. Our first speaker, Dr. Mahindra Dhadiwar. Second, Dr. Amol Murkute from Nasik. Third speaker will be Dr. Sagar Sonone from Nasik. Next speaker will be Dr. Sandeep Kulkarni. Dr. Jog sir is here. So we all are waiting for you. Dr. Jog from Pune, please come on the dais and kindly moderate the session. Our first speaker, Dr. Mahindra Dhaliwal.
Okay. Maninder is a senior consultant in PICU and pediatric pulmonology at Amruta Hospital at Faridabad. He is a person with in-depth knowledge of most of the issues in PICU and pediatric pulmonology. And I request him to speak about the expanded dengue syndrome. So over to you, Maninder.
Thank you, sir. So I've got 4 minutes, so I'll keep it very, very simple. We are going to discuss expanded dengue syndrome. So medicine and science is advancing, and definitely with that, disease and bugs are also advancing. With that, we have to change. Evolution is bound to happen, and it is a survival of the fittest ultimately. We all know this diagram of dengue, where we know at day 3, the critical period starts in a dengue fever. And after that, either the patient will recover or he'll go into PICU with leak. So this was a famous classification of dengue, which showed dengue with warning signs. And then there is severe dengue. But in this 2009 classification, expanded dengue syndrome was not even mentioned. 1997 classification by WHO, no mention of EDS. 2009, no mention of EDS. Then 2011, first time WHO defined something which is called as expanded dengue syndrome. So this is the classification which was given by WHO, where the DHF grade 4, after that comes expanded dengue syndrome. This was defined in 2011. So again, just to harp on it again and again, that earlier there was no mention of dengue expanded syndrome. It was only dengue fever or dengue hemorrhagic fever or dengue shock. And after that, this is how the WHO classified an extra column over here, which is called as expanded dengue syndrome. So and even in Indian guidelines, if you see, the dengue, severe dengue is classified into dengue hemorrhagic fever, then grade 1, 2, 3, 4 shock, and C is expanded dengue syndrome. So, but we hardly use this term in our day-to-day practice. We call everything which comes into PICU as severe dengue. Whereas WHO has defined expanded dengue syndrome as unusual manifestation of patients with severe dengue organ, which can be single or multiple organ involvement, such as liver, kidney, brain, or heart, associated with dengue infection, which are not usually explained by dengue fever or dengue hemorrhagic fever. It may be associated with high-risk conditions like infants or diabetes, or even co-infections like malaria, typhoid, or as a complication of profound shock. So a patient who goes for shock for 2 to 3 days, or patient who has ARDS or dengue liver failure, hepatitis, it is all expanded dengue syndrome. It is nothing else. It is expanded dengue syndrome. And this is how it is classified as in neurological. If a dengue patient has encephalitis, encephalopathy, meningitis, hemorrhage, Guillain-Barré. In gastric, if it has hepatitis, ALF, pancreatitis, then it is expanded dengue. It can involve the kidney, causing acute kidney injury, even HUS, then it is expanded dengue. Myocarditis is expanded dengue. ARDS, pulmonary bleed is expanded dengue. But we used to call all of them as severe dengue. So this is what is expanded dengue. It includes co-infection, it includes high-risk group, it includes other organ damage also, which usually does not happen in dengue. So it is underreported, but we every day manage such patients, but it is very much underreported, and that includes, again to harp on it, liver failure, dengue ARDS, MODS, dengue HLH, dengue encephalitis, dengue AKI. And the treatment is organ specific and organ supportive. Thank you.
Thank you. Thank you very much, sir, for such a short and informative talk. Please be on the dais. Our moderate, Joke, sir,
Yeah.
kindly felicitate our speaker.
Okay. The next speaker.
Okay, we will do the joint felicitation.
Yeah. Supportive care in PICU. Dr. Amol Murkute. I will request him to start.
Presentation. Yeah. करून घ्या ना. मनिंदर सर.
Okay. Dr. Maninder has other session in some other hall, is it? Okay.
Joke, sir, kindly felicitate our
Okay.
speaker. Thank you, sir. Thank you very much. Now our next speaker, Dr. Amol Murkute from Nasik.
Yeah.
Over to you, sir.
So good evening, all of you. Good evening, all of you. My talk for this capsule session is supportive care in PICU. Many a times it happens that we dwell too much of time onto etiological or the main treatment of a particular disease, like shock, infections and all. But seldom do we address or give adequate attention to supportive care. Supportive care is that supporting actor or actress which can make a movie hit or flop with his or her acting. So there are these 13 entities, and I am glad that Zog sir is moderator, and I am having an acronym for that. It is a well-established acronym, Fast Hugs in Bed, Please. There are these 13 entities in this, and one may argue that there is more to supportive care than this, but I am sure that it will cover more than 2 standard deviation area under the curve of supportive care. The 13 entities are Fast Hugs in Bed, Please. It stands for feeding, analgesia, sedation, thromboembolism prophylaxis, head and elevation, ulcer prophylaxis, glycemic control, skin care. In stands for indwelling catheter care, and we should always ask, can I remove anyone? B stands for bowel care, E for environmental adjustment, D for de-escalation of drugs, and P stands for psychosocial supports. There are these 13 entities. I'll be really fast to cover all these 13 entities in short period of time. So feeding, as far as PICU is concerned, is relatively neglected part, unlike our NICU counterparts who are very eager to start trophic feeds. But dictum is not to starve any kid for more than 24 to 48 hours. And we should always make a habit to document the calories and proteins received in a particular day. So if you see the first table, as the age decreases, the minimum amount of calories required to run the show, that is for BMR, are increasing. Having said that, in certain conditions such as fever, trauma, sepsis and burns,
the caloric requirement increases by certain folds. Although in sedated and paralyzed kids, we should always try not to overfeed them because their caloric requirement is less. Analgesia sedation is again another neglected part in PSU, and there are variety of options available starting from paracetamol, fentanyl, and Dexem. So, like Sanjay Das from Vastav, kuch nahi to at least paracetamol hi de do. Thromboembolism prophylaxis is not routinely recommended in pediatrics, although surviving sepsis guidelines do recommend their use in post-pubertal kids with severe sepsis, and we have used this thromboembolism prophylaxis quite often during severe COVID pneumonias. That completes FAST. S stands, now we move on to hugs. S stands for head and elevation. That should be around 30 to 45 degree. It prevents microaspiration and decreases the ICP. And we should remember that during CPR and during hypotensive shock, the child should be flat. The head end should not be elevated. Ulcer prophylaxis with ranitidine and pantoprazole and other drugs. And these drugs are become as ubiquitous as ceftriaxone in our clinical practice. So in that sense, we are way ahead. G stands for glycemic control. Although the tight glycemic control, unlike adults, is not recommended in pediatrics, we strive very hard to prevent hypoglycemia, but seldom do we address give attention to hyperglycemia. So if the sugars are consistently above 180 despite reduction in GIR, we should consider starting insulin infusion. S stands for skin care, and it is usually meant for prevention of bed sores. Eye care with lubricating eye drops and oral care with chlorhexidine based oral care, especially for intubated kids, is equally important. This is NPUAP classification for pressure sores, and if you see as the staging advances, the management changes from protective dressing to debridement. IN stands for indwelling catheter, and we all know how to monitor ETs, CVP lines, and chest drains. But I take this opportunity to stress upon the fact that we lack to monitor our IV cannulae. So this is simple VIP score called as visual infusion phlebitis score. All of us, we all know this. It is readily available on internet. I would request all of you to take a printout of this and give it to your sisters. So at least once in their shift duties, they should try and plot this VIP score so as to pick up this thrombophlebitis and its complications early. B stands for bowel movement, E for environmental adjustments. So anything in the vicinity of the child that may give rise to agitation in a child should be addressed. Be it soiled bed sheets, very low temperature on air conditioners. So anything in the environment should be adjusted. D stands for de-escalation of drugs. In the morning, we had an excellent panel discussion on antibiotics. Think before you ink. Now, even if you have inked particular antibiotic, we should always try to de-escalate that particular antibiotic as fast as possible. Last but not the least is psychosocial support. The dictum is to counsel, 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 followed by documentation, documentation, and documentation. So these are the 13 entities under the supportive care. Fast hugs in bed, please. I would request all of you to practice these fast hugs so as to get good results in our PISU sick kids. Thank you.
Okay, thank you, Dr. Amol. Now is Dr. Sagar Sonavane for newborn screening for congenital hypothyroidism.
Good evening, all. All of us must be doing, I think, newborn screening for hypothyroidism. What is the objectives of this session? I'll just enumerate. Is the enumerate the etiology of congenital hypothyroidism. We all should know the causes of congenital hypothyroidism as a general pediatrician. And we should be understanding the importance and timing of newborn screening for congenital hypothyroidism. And other thing is timing and importance of rescreening. Usually rescreening most of the time is we miss. I have noticed in my practice also, sometimes we miss rescreening. So to which newborn we should be rescreening, that we should be knowing. And fourth one is able to interpret the result of newborn screening for congenital hypothyroidism. So what is the etiology? All of us know etiology is it is a primary hypothyroidism, which is from the thyroid gland, and other is central hypothyroidism, which is the cause is in the center, that is either in the hypothalamus or in the pituitary gland. So defect in the thyroid hormone biosynthesis is one cause which is very rare. But what is the commonest cause of primary hypothyroidism is thyroid degenesis. Is almost 80% of the cases are due to because of thyroid degenesis, and central hypothyroidism is which is rare, and which is I told you that it is from pituitary or hypothalamic origin. That is a rare cause. What is the importance of thyroid screening? Why we all should do thyroid screening? There are many other screening tests all of us know. We should be doing actually most of the screening which are now readily available. I have seen many prescriptions of gynecologists from Pune actually, and in Nasik also, those who are doing some screening test for each and every newborn. I came across that, but I am myself is doing thyroid screening. The incidence, but you must be surprised to know that incidence is so high. See, the ranges from 2000 to 4000 is to 1. There is one newborn from amongst the 2000 to 4000, and it is in 1.5 times or 2 times more in females than the males. In twins, so twins mean mandatory here. We should be doing all twins for screening for congenital hypothyroidism. In multiple births, in when the mother is older, that is must, and in preterm babies we should be doing. And newborns with congenital hypothyroidism have no or few clinical manifestations. So that's why the importance of screening. But very rarely will a child or newborn baby will present with signs and symptoms of congenital hypothyroidism at birth. Most of the newborn, they will present at around 6 weeks of age by the time the neurological damage has already occurred. So no, it will be the late treatment to start. So when you want to start treatment earlier, and the studies says that when you start treatment within 2 weeks of life, the global neurological or neurodevelopmental quotient is better. Therefore, we should be insisting on, or we should be practicing to do screening earlier in each and every newborn. Early and adequate therapy initiation, I just told you that prior to 2 weeks of life will increase the maintain the global developmental intelligence. Few subtle like scholastic and so mental developmental milestones may be delayed, which may be noticed even if you start treatment at 2 weeks of age. In severe congenital hypothyroidism, the outcome is more worse. So a few severe congenital hypothyroidism, even if you start treatment at 2 weeks, there may be some damage to the neurodevelopment will occur. Okay. What is the timing of screening? So ideal timing of screening is between 2 to 4 days of life. Okay. You all of you know that. Or if it is not possible, at least before the discharge of a child from a nursery or from a PNC ward, one should be doing a thyroid screening. जेव्हा आपल्याला चान्स मिळेल ॲटलिस्ट तेव्हा आपण TSH screening, thyroid screening करून घ्यायला हवं. There are 3 methods. One is which follow most, like TSH screening. This TSH screen will actually detect most of the cases of primary hypothyroidism, which is a more specific test for diagnosing primary congenital hypothyroidism. With TSH is then high, one should be doing T4 measurements. Initial T4 measurements will actually detect primary central hypothyroidism, but few cases may be missed, like with in which there is primary hypothyroidism because we did not have TSH levels, or in which when the T4 is normal but TSH is elevated, like those in subclinical thyroid or what you say mild thyroid cases may be missed when you only do T4. Simultaneous T4 and TSH measurements are ideal. I think the cost of the thyroid tests are not very costly. They are I think 250 or 350 rupees. We can counsel the parents. And if we collect a sample good enough, then we should be sending TSH and T4 measurements. That is ideal, but at least one should send TSH to detect primary congenital hypothyroidism, which is more specific test for it. When we should do repeat screening? Repeat screening, actually, I have also missed in few cases. That's what I know. I will tell you the importance of this. In low birth weight babies, in preterm babies, in sick neonates admitted to NICU, and if specimen is collected within first 24 hours, means cord blood is collected, and in multiple births, particularly in same-sex twins and in infants exposed to iodine. What happens in these cases? The TSH is suppressed because of immaturity of hypothalamic pituitary axis, and we may miss few cases of hypothyroidism in whom the TSH rises little later. So therefore, the importance of repeat skinning. In these cases, it must be done. When we should be doing repeat skinning? At 2 weeks, if the sampling is within 24 hours or preterm preterms less than 32 weeks or twins, then when it should be done in preterms, 1, 2 of 4 weeks, and at term, corrected gestational age or the day of discharge, whichever is earlier, or 2 and 4 weeks with monitoring. If the child is exposed to the iodine, then one should be monitoring that newborn baby at least till the age of 1 month, and we should be screening at 2 and 4 weeks as well. And the routine screening will be you all know it will be done at 2 and 4, between second and fourth day of life. Abnormal test always should be confirmed by obtaining venous sample and sending it for free T4 and TSH. So what is the interpretation? All of you know, still I'll emphasize. Low free T4 and high TSH is primary hypothyroidism. Normal free T4 and or total T4 and high TSH is compensated or subclinical hypothyroidism. If the serum TSH more than 20, one should be treating. And if it is between 6 to 20, observe and repeat TFT in 1 week. And if TSH remains elevated at 10 units per liter or more above 4 weeks, then one should be treating. And low free T4 and low or normal TSH, we all of us know that this is central hypothyroidism. Primary hypothyroidism with delayed TSH elevation and sick neonates or prematurity or in premature babies, and thyroid binding globulin deficiency, which are actually the TBG deficiency is a rare cause of one of the rare cause of hypothyroidism. What are the additional tests? Once you confirm that it is hypothyroidism, one should then be looking for the cause of hypothyroidism. If normally what happens, these causes which usually does not interfere with the treatment. Even if you don't do the treatment is must, so one should be starting the treatment, and you may try to find out the cause of hypothyroidism. So these are the tests which are thyroid ultrasound is very simple screening test. It can done. It will detect it will tell about the anatomy of the thyroid. The thyroid radionuclear scan will tell us about the site, where is the thyroid gland and how is it. The thyroid antibodies, like from the transient hypothyroidism, can be diagnosed with these thyroid autoantibodies, which are transferred from the mother to the baby. And serum thyroglobulin level, if they are deficient, and the urine iodine concentration in case of you suspect that the child has overexposure to iodine because of many creams and some medications. And radiographs of the knee, if required. Radiographs of the knee, I just reading, they mentioned that if the lower femoral epiphysis is absent at screening at birth, those children will have neurodevelopmental abnormalities. Okay, thank you.
Thank you very much, Sonone sir.
Okay,
We are going to declare the name of the winners for oral paper presentation. To felicitate them, I will request Dr. Ramakant Patil sir, Dr. Nitin Mehkar, and Dr. Tushar Godbole to be on stage. Oral paper presentation.
Jok sir, with
I
your
will
permission, sorry to interrupt you, but these students have to go back, and they are prize winners. So we are expressing our this thing, sorry and regrets to interrupt in between. But I will like to specially thank my paper and poster team, headed by Dr. Nitin Mehkarkar, Dr. Tushar Godbole, and many are there, Dr. Anand Makharia, Dr. Ravi Sonone, Dr. Vaibhav Pustake, Dr. Dhira Pustake, Dr. Vaibhav Gode, and Prasanna Parashare, Dr. Ravi Sonone sir, I said, Dr. Nilesh Aire, these are all faculties in medical colleges of MUHS like SMBT College, Dr. Vasantrao Power Medical College, then our MUHS College, and Pravara. So these have taken special efforts in assessing the paper and posters. 52 oral papers were there and 50 posters were there, and they have assessed. And even RCT paper was there from a PG student. Dr. Praveen Kale also had helped in assessing all these papers. Sorry to whosoever I have left, please blame it on my
reducing gray hairs and increasing age. So I request Dr. Tushar Godbole and Dr. Nitin Meherkar to give the prize distribution.
Okay.
Please, Anand.
I request all of three to be on stage only. To declare the winner for oral paper presentation, third rank goes to Nikhil Reddy from GMC Aurangabad. Second rank, Dr. Dheeraj Khandade from GMC Aurangabad. GMC Aurangabad is fortunate to have 2 prizes. Congratulations. The winner for the oral paper presentation is Dr. Georgina Elsa Joss from the Noble Hospital, Pune. Congratulations. मॅडम तुम्ही पण जॉईन होऊन जा फोटो आम्ही काढू. Three chairs for all of them. Hip hip, hooray. Thank you all of you. Sorry, jokes, sir, for the disturbance. We will continue our capsule session further. I will invite Dr. Sandeep Kulkarni to come on the stage.
Sandeep. He will be speaking on hemangiomas, infantile hemangiomas.
Good evening, friends. We all know, we all face many birthmarks in our OPDs. And can you slide? Ajay, I have to do it. We all see many birthmarks in our OPDs, and sometimes they are very tiny, and sometimes they are very big, and they are very scary too. So parents are very much concerned about that. Let us see in short how to go ahead with these birthmarks. These are benign vascular endothelial tumors, more common in females, commonly seen in skin and subcutaneous tissues. They grow rapidly in first year of year, and then in almost 70% they involute by up to 7 years of age. But in 30% they may persist till further life. Early proliferative lesion is very bright, irregular, and deep lesions, but deep lesions are very bluish and very irregular. Involution causes fading, softness, and shrinkage of the skin, leaving a crepe paper-like area behind. So infantile hemangiomas, there is no exact cause what causes them, but more common in firstborn babies, girls, premature babies, low birth weight babies, those mothers having preeclampsia or placenta previa, they may have, the children may have more amount of birthmarks. So incidence of infantile hemangioma is increasing over the years. Presently, we see many cases who are having these kind of birthmarks on them. Most of them are small and are have less concern, but 15 to 20% may have multiple lesions. Those on eyelids, earlobes, external nostrils, lips, tongues, palate, and around anus may have some functional concerns also. Large hemangiomas around face may be associated with face syndrome, where hemangiomas in liver or in groin or axilla, that is segmental hemangiomas, may need further evaluation. So Modified International Society of Study for Vascular Anomalies, that is ISSVA classification, where infantile hemangiomas could be focal, segmental, or intermediate, or congenital hemangiomas that is rich, that is rapidly involuting congenital hemangiomas, or non-involuting congenital hemangiomas. So there could be two types of hemangiomas. There could be other conditions such as tufted angioma, or pyogenic granuloma, or carposiform hemangioendothelioma, or spindle cell hemangioendothelioma. So lesion and its cause, as you see, there is a small bright spot under the eye, and that could be seen in, if you take an slide, it can be seen as a tufted endothelial enlargement below the skin. And if you see the course later on in their life, maybe around after 1 year of age, it involutes and leaves a very small scar. So this is the course. You can see multiple cases over here. You can see somewhere on the forehead, or near the umbilicus, or on the hand, or even on the legs. And in many cases, they involute almost completely. So why worry? If they are involuting completely, why worry? But in some cases, there could be very large hemangiomas, and those could be really scary, not only for parents, but sometimes even for us. So that could be on the nose, on the cheek, or it could be multiple hemangiomas over the body. So hemangiomas needing early interventions. One is periorbital hemangiomas, because they may there could be a deprivational amblyopia, or total obstruction of visual field could be there. Astigmatism can happen because of hemangioma. So we have to involve ophthalmologists in managing these cases. There could be large hemangiomas, or obstructing nasal passages, or under the in the mouth, palate, or tongue, that can lead to feeding difficulties. Or there could be skin ulcerations leading to hemorrhage or infection. Bacterial septicemia and necrosis can occur along with thrombocytopenia, that is Kasabash-Merritt syndrome. Or CCF can occur because of shunting of blood. Or there could be lumbosacral hemangiomas associated with with spinal defects, tethered cord, or lipomas, or other annular malformations. So all these hemangiomas need early intervention. What evaluations we need? Need to differentiate from other vascular anomalies and lymphangiomas. Careful examination is very much must in every case. We may support with CT or MRI for larger lesions around head and neck, like Face syndrome, or cardiac evaluation is like ECG and echo if there is any hemangioma, very large hemangioma causing shunting of the blood. CT, MRI, abdomen, or for ruling out hepatic hemangiomas. Neurological and developmental assessment can be done, and other investigations such as CBC with platelet counts, liver function tests, renal function tests, and thyroid function tests are must. Treatment: almost 70% of infantile hemangiomas involute, and they just need assurance. So we have to sit down with the parents and assure them, and we have to monitor the progress of this hemangiomas in early life of the baby. Infantile hemangiomas are very small, without any deformity, and maybe in hidden areas. So that can be assured, and the parents can be counseled that this will go with the time. Medical line of treatment for localized, and surgery and laser surgery will go one by one. So medical line of treatment, propranolol is the right drug which we use. Vascular endothelial growth factor inhibitor, because it associated with this. So 1 to 3 milligram per kg usually is given for up to 6 months, and it is effective in almost 85% of proliferating hemangiomas. Short-term side effects such as hypoglycemia, hypotension, hyperkalemia should be observed, and long-term side effects are very much not known. Potential rebound after weaning of the propranolol are seen in some cases, and topical antibiotics are given for all infected infantile hemangiomas. Oral steroids, that is prednisolone, 2 to 3 milligram per kg per day, single AM dose for 1 month, slow tapering for 3 to 4 months, and effective in 75% cases which did not respond to propranolol. Short-term side effects, as we know, hypertension, weight gain, Cushingoid features, live vaccine restrictions are seen, and long-term side effects such as growth retardation, bone demineralization, and adrenal suppression. Potential rebound are also seen in these cases, and other drugs such as interferon alpha 2A and vincristine have some promising results. Laser therapies are needed in all cases where there is some large hemangiomas on the face. So they are less painful. They promote healing and ulceration of ulcerated lesions. There are special FPDL, that is flashlight pulse dye laser, which is very useful in superficial strawberry hemangiomas. And it is also less scarring. YAG laser is needed for those who are rapidly growing deep and mixed hemangiomas. 75% respond to it as it penetrates to the depth of 5 to 6 millimeters and requires local and general anesthesia. Scar formation is more frequent in these cases. And surgical resections are needed in very few cases, which are very large hemangiomas. Those are affecting functions such as vision, breathing, eating, hearing, and all. Ulcerated large hemangiomas and hemangioma causing mass effects and cosmetic deformities and complex reconstruction may be needed in some cases. So in summary, though throughout examination, thorough examination and reassurance is needed in many cases, appropriate investigations for classification, no associated conditions and extent of lesion in few cases, detail of management protocols, short-term and long-term side effects of the medical management should be discussed with the parents. Laser and surgical treatments, if necessary, should be discussed at length with the relatives, and self-esteem of the child and financial support of the family needs to be reconsidered in planning the treatment. Thank you. Thank you very much.
Thank you. Thank you. Thank you, Sandeep.
Thank you.
I would request all the speakers to kindly restrict to the time of 4 minutes. We have a saying that he came, he saw, he conquered. So similarly, he or she comes, speaks, and conquers. Okay, so that's the purpose of 4 minutes. So Trupti, you can speak about EQ. The topic has been given to a female pediatrician because it's related to emotions.
Good evening, everyone. So I will be talking about emotional intelligence, and I would like to begin with a famous quote of father of emotional intelligence, Daniel Goleman, that in a sense, we have two brains, two minds, and two different kinds of intelligence. One is rational and second is emotional. So in my encapsulated talk about EI, I will be giving glimpse of concept of emotional intelligence and its implications in children. So what are emotions basically? These are the inward feelings which are expressed outwardly via behaviors. And what is emotional intelligence? There are various theories and models about EI given by different psychologists and researchers. The scientific definition given by Salloway and Mayer is emotional intelligence is the ability to perceive emotions, to access and generate emotions, to understand emotions and emotional knowledge, and to reflectively regulate emotions so as to promote emotional and intellectual growth. Emerging neuroscience studies have revealed that there are several emotion-related regions in the brain which correlate with different EI skills. These regions are anterior insula, amygdala, orbitofrontal cortex, and so on. And now, why emotional intelligence is being so talked about and so important in children? Because EQ is not something we are born with. It is a learned skill. Research says that emotions either hurt or heal children. Emotions matter in every important aspect of children's life, and so is the emotional intelligence. We know today children are facing many new era challenges. There is a big list of these onerous and thorny challenges. Gadgets on the top are acting as an unbeatable new era monster, which keeps on haunting the brain continuously for the dopamine release. And what are the implications of all these challenges? We also know that 21st century is an era of mental disorder epidemic. As per WHO, there is no health without mental health, and we cannot overlook the fact that emotional well-being is one of the 3 important core competencies in mental health definition. If we look at the magnitude of mental health problems in India, As per study published in Indian Journal of Psychiatry 2019, at any given point of time, around 50 million Indian children suffer from mental health disorders. Around 40 to 50% of adult mental disorders originate between the age group of 4 to 14 years. And major chunk of these mental disorders is contributed by emotional disorders. So where is the missing link? Is this piece of emotional intelligence is missing from the jigsaw puzzle of lifespan development of today's children. Ample research has been done regarding direct connection between emotional intelligence and mental health. EQ has been identified as a predictor of mental wellness. Unfortunately, studies have revealed that since last few decades, IQ of children has risen significantly, but children are deteriorating on all the 42 indicators of EQ and improving on none. Now, IQ versus EQ, which is more important? Literature says that when it comes to success in life, only 20% of the contribution is by IQ. Rest 80% success depends on your EQ. But of course, it doesn't mean that IQ is not important. EQ is not opposite of IQ. It is not a triumph of heart over the head. Rather, it's a unique intersection of both. Now, my last part. Can EQ be really enhanced? As EQ is only partly genetic versus IQ, which is fixed and genetic, research says that EQ can be enhanced via unlearning, learning, and relearning. Various studies have been done across the globe regarding effectiveness of emotional intelligence enhancement programs. Though the limitation of these studies is lack of generalizability, there are promising results of enhancing EQ scores across different age groups. I would like to conclude with this take-home message. In this era of overwhelming complexity, when more and more artificial intelligence is coming in, to bulletproof the serenity, creative magnificence, productivity, and prosperity, having a critical balance between cognition and emotion is of utmost importance. With this, I would like to end my talk. Thank you so much.
Thank you. Now we have Dr. Tushar. Dr. Tushar, you are requested to speak on vitamin D. What has changed?
Good afternoon, or rather good evening. So in next 4 minutes or so, I'm going to speak about what has changed with respect to vitamin D. So why this vitamin D guideline 2021? There was a guideline in 2017 by IAP. So what has changed after this 2017 guideline is the CNNS, the nutrition survey data came, which gave us a huge data on how children across the country behave with regards to vitamin D. What is the prevalence? More studies are now available with regards to the prevalence of vitamin D, the treatment modalities of vitamin D, the safety data, and effect of maternal nutrition is also now available. And the earlier guideline did not speak about maternal supplementation, the specific role of preterm and LBW supplementation, and sunlight quantification. But why do I get to talk about this talk? Because I am one of the contributors for the current guideline. So what has changed and what has remained the same is the definition has remained the same, where the deficiency is defined as less than 12 nanograms per ml or less than 30 nanomoles per liter. Sufficiency is above 20, and anything between is considered insufficient. But these cutoffs are based on the PTH deflection point and the skeletal benefits, not the extraskeletal benefits. The sources of vitamin D are overhead sunlight, mushrooms, fatty fishes, and dairy products. Most of them are unreliable, expensive, and unpredictable. So barring 2 states in our nation, one of them being Kerala, where they eat plenty of seafood, rest of the India, including the northern states, are extremely deficient in vitamin D. So what is routine supplementation? The guideline clearly states that all infants until first birthday should receive 400 international units per day. Doses above 400 IU do not provide any additional skeletal benefits. So doses like 800, 1200 do not give any additional support, and 1600 and above given per day is in fact toxic. Points to ponder: Do children beyond their first birthday suddenly start walking and going into the sunlight? So one must think that do we have to extend it until the child goes out in the sunlight? With regards to preterm babies, 400 is safe but may not be effective. There are studies from Europe, Canada, as well as AIMS New Delhi, where they have shown that preterm babies, ELBW babies, may not get sufficiency with this 400 IU dose, and there is a scope of giving 800 international units per day for preterm ELBW babies, provided you have documented the baseline value, and that is less than 10 nanograms per deciliter. Children and adolescents, of course, we want them to go out in the sunlight, get sunbathing, and no routine testing and no routine supplementation is recommended. Point to ponder again, cities like Delhi have prevalence of vitamin D to the tune of 90% because of pollution. How do we treat vitamin D deficiency? Daily is preferred over weekly, and oral route is always preferred over injectable route because it's more sustainable in terms of the levels. Below 1 year, you give 2,000 international units per day. Above 1 year, you give 3,000 international units per day. For 12 weeks, that's 3 months. And there is an option or provision for giving 5 doses of 60,000 international units per week for 5 weeks. Sunlight exposure, it's recommended that you go out in the sunlight, infants for 17 to 30 minutes and older children for 30 to 45 minutes. Overhead sunlight is what is recommended. So out of these 3, the darker ones require more sunlight as compared to the fair ones. So these are the needy ones who require vitamin D from outside with liver, kidney disease, children on anticonvulsants, children on transplant therapy, and having debility or skin diseases. And children with Cushing's or hypogonadism are also the candidates to be given continuous preventive dose of vitamin D. So these are the resources for you to read. And with this, I thank all of you.
Thank you, Tushar. Now, Amol, your turn. You are going to speak on reading our own lipid profile.
Thank you, sir. So we'll be looking at the components of lipid profile first. So it is total cholesterol, high density lipoprotein, low density lipoprotein, very low density lipoprotein, and triglycerides. So before that, we should look after this borderline and abnormal levels. Normal we know, acceptable we know. So the total cholesterol between 170 to 199, we should be watchful for it. And more than 200 is abnormal. Low density lipoprotein, the borderlines level is 110 to 129. Then non-HDL cholesterol, the level is 120 to 144, and abnormal is more than 145. In triglycerides, depending on the age group, between 0 to 9, the borderline level is 75 to 99, and more than 99 is, more than 100 is abnormal. And between 10 to 19 years age, the borderline level is 90 to 129, and abnormal is more than 130. So we should be watchful for these levels, and we should look for controlling it by various means. We will look after that. And the high density lipoprotein, so the borderline should be between 40 to 45, and less than 40 is abnormal. So it should be acceptable is more than 45. So this is the flow diagram of pathway of lipid metabolism. Because of time constraint, we will not go in details. So these are various conditions in which these components are increased. So mainly the commonest is familial combined hyperlipidemia, in which the LDL and VLDL are increased. And another one common is familial hypertriglyceridemia, in which LDL has been increased. And the rare ones are other primary hyperlipoproteinemia, then familial hypercholesteremia, endogenous hypertriglyceridemia. These are various pathologies according to these conditions. So these components are high density lipoprotein. So this is a scavenging molecule, and the higher levels will protect against the atherosclerotic cardiovascular diseases. And the low HDL is associated with obesity and sedentary lifestyle. So how to improve the HDL? So we should carry out regular exercises. Then we should decrease our carbohydrate diet. And the next one is alcohol. So we should limit our alcohol. It should be 30 to 60 ml per day. Sorry. It is it is 30 to 60 ml per week actually it is. So the next is LDL, and it is the main lipid causing atherosclerosis, and the desirable levels are less than 100, and when the treatment is needed. So less than 190 without risk factors, treatment is needed. And if it is with risk factors, then with one risk factors less than 160, then treatment should be initiated. And with two risk factors less than 130 is the level in which treatment is needed. But the ideal one is less than 100. So we should look after it. And how to improve it? So dietary means and various statins and this new molecule, ezetimibe, then triglycerides. So it contributes to atherosclerotic process. The target should be less than 150. There are secondary causes of hypertriglyceridemia, that is diabetes, thyroid, then drugs and HLH. And primary causes are genetic mutations in lipoprotein lipase. So the treatment is dietary changes, dietary modification, and fenofibrate, which decreases the cholesterol levels. So the diet advice for healthy lipid profile is, so you should reduce the saturated fats and trans fat. You should increase the polyunsaturated fatty acids and medium chain saturated fatty acids. Then you should limit the alcohol, and you should increase the fiber in diet. You should increase the omega-3 intake, and you should reduce the dairy products, also increase in plant sterols. Thank you, one and all.
Very good. Okay, and now last speaker is Dr. Vinayak Patil. He will speak on management of constipation.
Good evening, friends. With changing lifestyles, I think the patients of constipation in our day-to-day practice have increased significantly, and sometimes they are difficult to treat. So, the prevalence of functional constipation is 3% worldwide. It is defined as difficult or infrequent bowel movement, deviation from normal in frequency, painful defecation, passage of hard stools, and/or sensation of incomplete evacuation of stools. There is no underlying systemic cause or anatomical defect. Etiology, mostly one episode of painful defecation can precipitate constipation. In toddlers, it can be dietary changes, excessive toilet training. In older children, it can be unpleasant toilet facilities, sexual abuse, trauma to perineal areas, and voluntary withholding while playing. This is pathogenesis chart. For evaluation, this Rome 4 criteria is used. There are few red flag signs which we can differentiate or which can help to diagnose organic cause of constipation, such as delayed passage of meconium, where we can think of Hirschsprung's disease or cystic fibrosis, failure to gain weight, coarse faces, or prolonged jaundice help us to diagnose hypothyroidism. Coming to main part of treatment, fecal disinfection is the main treatment of the constipation. Orally, we can give this polyethylene glycol 1 to 1.5 gram per kg per day, dissolved in 10 ml per kg water or flavored beverages, which should be continued twice daily for 6 days until child has passed large amount of stools. Sometimes these rectal enemas can be used. If a patient is not taking orally, we can give polyethylene glycol through nasogastric lavage, also 25 ml per kg. In older like more than 2 years old child, we can use stimulants like Fena or Bifacodil, or sometimes we can use Bifacodil suppositories in child below above 2 years of age. Glycerin suppositories can be used also. Coming to maintenance therapy, PEG is the main drug of choice. 0.4 to 0.8 grams per kg per day for at least 3 to 6 months of age, 3 to 6 months. Behavior modification is very important, like regular toileting with rewards, toilet fitting after meals, family education. Dietary modification is very important. More fluids, balanced diet, eliminating cow's milk, sorbitol containing juices. For maintenance, also PEG is the main drug of choice. Sometimes this magnesium sulfate or sorbitol can be used. For treatment in infants, rule out organic constipation first. Increase fluid intake, may respond to sorbitol containing juices. If these measures are unsuccessful, lactulose or PEG are safe. Glycerin suppositories can be used. Avoid enema, stimulant, laxatives or mineral oil in treatment in infant. Coming to discontinuation, may begin weaning laxatives once the child has regular soft daily stools and no episode of acute constipation or fecal incontinence for 3 to 6 months. The laxative dose is gradually decreased to a dose that will maintain 1 to 2 bowel movements per day. If recurrence occurs, weaning has started, it is important to have a rescue plan. Sometimes relapses are very common. And in that condition, ensure that the parents are mixing the drug properly. Briefly increase the dose of oral laxative, double the dose. Use enema to empty the rectum. Give 1 or 2 doses of stimulant laxatives. If the intervention is successful, treatment is continued with maintenance osmotic laxative, but at higher doses. 60% of the children with functional constipation have successful outcome with laxative therapy after for 12 months. 30% require long-term therapy. Key points are majority of the childhood constipation is functional. Detailed history and examination are sufficient to differentiate functional and organic constipation. Presence of red flag signs suggests organic constipation. PEG is the principal laxative for maintenance therapy and for treatment of fecal disinfection. Both education and behavioral intervention are important part of treatment. Treatment may be required for months to years depending on the severity. Thank you.
Okay. Very good. So we have completed all the speakers and now I will just summarize whatever they have said. First of all, I congratulate all of them. सगळ्यांनी भरपूर अभ्यास करून presentation केलेला आहे. Now, most important things that you should remember. So the first speaker, Dr. Maninder, he gave us information about the expanded dengue syndrome. What you should remember is that those features which were thought to be uncommon have now become common. So liver failure, acute kidney injury, myocarditis, lung involvement, brain involvement in the form of encephalopathy or encephalitis. Earlier people did not think that these are very common, but now they are included under expanded dengue syndrome, and associations like say malaria or maybe enteric fever, they could be associated with dengue. So all these are included in the expanded dengue syndrome. Baki management तर सगळ्यांना आपल्याला माहितीच आहे. The second speaker, Amol, he gave unmol information about supportive care in PICU, and he made the discussion very easy by giving the mnemonic fast hugs in bed please, and he has given the information about each of these components. The most important thing you should remember is that critically ill children should not be kept fasting for more than 24 to 48 hours. तेव्हा आई म्हणते बा पोरग खात नाही तेव्हा आपण म्हणतो नाही खाईल खाईल तसं नाही. क्रिटिकल इल असेल तर you have to be very very careful. Then third speaker, Dr. Sonavane. Sagar Sonavane, I will call him Jnana Sagar Sonavane. So Jnana Sagar has given lot of information about newborn screening. What you should remember is that every newborn, before he goes home, has to have his TSH done. Okay. Baki काय केलं नाही त्याचं TSH नक्की करा. Because if you delay the diagnosis beyond 2 weeks, he will have delayed global development. Global याचा अर्थ सगळी ब्रेनची डेव्हलपमेंट खलास. म्हणजे not only one selective, but global development will be affected. So you must see that the diagnosis is made before 2 weeks. If the values are abnormal, please try to confirm the values and then try to find out the cause. And this is newborn screening when the child is coming into world. And I always say that even check the thyroid level of the new employees coming to your office. Okay. म्हणजे those like receptionist हे वगैरे तुम्ही अपॉइंट कराल तेव्हा त्यांची पण थायरॉइड लेवल बघा. अदरवाइज युअर लाईफ ऑल्सो विल बी व्हेरी व्हेरी horrible. Okay, so don't appoint people who are idiotic look and then developmentally delayed with a constipated face. Okay, this is thyroid is very important in life. Okay, then Sandeep gave a detailed account of infantile hemangiomas. He told us that the incidence is rising. Remember, hemangiomas in the beard area, okay, they are always dangerous. They are associated with some internal problem, palate वगैरे तिथं कुठे hemangioma असतो, which may produce laryngeal obstruction. He gave so many other examples where vision could be impaired, so on and so forth. And management includes either propranolol, surgery or laser therapy. मॅनेज करायच्या फंदात पडू नका, प्लास्टिक सर्जन कडे रिफर करा, त्यांचा पण धंदा चालला पाहिजे. ओके, देन नेक्स्ट इज ऑफ कोर्स द डिस्कशन ऑन EQ बाय तृप्ती. रिमेंबर, इमोशनल इंटेलिजन्सचा अर्थ काय, की पोरांचा इमोशनल क्वेश्चन तुम्हाला वाढवायचा. म्हणजे साने गुरुजींचं पुस्तक वाचल्यानंतर आपल्याला रडू येतं, तसं त्या पोराला रडू यायला पाहिजे. दॅट इज इमोशन. ओके, नाही तर पोरं कार्टून बघून उड्या मारतात, कोचावर नाचतात. दॅट इज ओके, वन हॅपीनेस. ओके, सो टू इम्प्रूव्ह दोज डिफरंट इमोशन्स इज व्हेरी इम्पॉर्टंट. आनंद आहे किंवा दुःख आहे. सो शी हॅज ऑलरेडी टोल्ड अस दॅट लर्निंग, मेमरी, अटेन्शन, रिलेशन्स, डिसिजन मेकिंग अँड फिजिकल अँड मेंटल वेलबिंग हे सगळं इमोशनवर अवलंबून आहे. You may not be knowing, but at very high level offices, the person who is employed is not checked by his IQ, but he is checked by EQ. आपल्या लोकांशी कसा वागतो, सबऑर्डिनेटशी कसा वागतो, त्याच्यावरनं युजली त्यांना अपॉइंटमेंट्स म्हणजे वरच्या मिळत जातात. I congratulate her for bringing out many points. And the last thing that she told was EQ can be increased. म्हणजे जो इमोशनल क्वेश्चन आहे तो वाढवता येतो. But I personally feel it all depends on the parents and the teachers as role models. आपण स्वतःच जर एखाद्याला काही प्रॉब्लेम आल्यानंतर आपण रडलो नाही, आपण जर वेदना दाखवल्या नाहीत, तर मुलं कशी दाखवणार? सो इट इज टोटली अप टू अस. देन नेक्स्ट स्पीकर वॉज ऑफ कोर्स Hushar Godbole. I call him not Tushar but Hushar. Okay, and then he gave us the vitamin D guidelines. The earlier guidelines I was involved, these 2021 guidelines were a modification of that. Remember, the level of 12 nanograms per ml is important. Okay, and there are many endocrinologists who keep debating about this between 12 and 20. That is because below 12 you need to treat definitely. But there are people who say, if it is below 20, you treat it, and so on and so forth. And remember that he told us some new things, like the sources of vitamin D are unreliable and unpredictable, but the dose remains to be 400 units per day. And then the prematures may be given 800 units, provided the deficiency has been proved. And then you have to have your exposure of the skin for 40 minutes a day. And those who are at risk, like those who are on anti-epileptic therapy or some other disease, they definitely need to be supplemented vitamin D. And I suppose obese children also, they require vitamin D more than usual, because the vitamin D usually gets into the fat. Okay, and then the next speaker, Amol, he gave information about lipid profile. He, of course, faltered while giving the exact amount of alcohol. So I was keenly listening to him. But remember that he gave the talk, and probably it was at the end because organizers wanted you should not eat a lot during banquet. Okay. And then some important points. Cholesterol bhaga ghari gelyanantar. If it is more than 200, it's a red flag. Low HDL less than 40 is bad. Okay. Because all of us are, having a sedentary lifestyle, and some of us have developed obesity. LDL less than 100 is ideal. And the main lipid causing atherosclerosis is LDL. तेव्हा लिपिड प्रोफाईल निदान वाढदिवसाला चेक करा. मी माझ्या मित्राचा कधी वाढदिवस असेल, HBD, that is HBD is happy birthday, but hypertension, blood sugar and dyslipidemia या 3 गोष्टी हॅपी बर्थडेला बघायच्या. बरं, मी जातो मित्राच्या घरी, त्याला झोपवतो, त्याचं ब्लड प्रेशर घेतो, त्याचं ग्लुकोमीटरनी शुगर मोजतो आणि लिपिड लेवलसाठी ब्लड पाठवतो, आणि त्या लिपिड लेवलचे पैसे असतील ते बिल मी भरतो. सो दॅट इज दी गिफ्ट विच आय गिव्ह इट टू माय फ्रेंड. तो तुमचा जो कोणी बेस्ट फ्रेंड असेल, बेस्ट फ्रेंड तुम्ही तुमची बायको पण असू शकते, तेव्हा तिचं तरी करा. ओके, नाहीतर आपण आपल्याच लोकांना निग्लेक्ट करतो. त्याच्यासाठी हा टॉक ठेवलेला होता. बरोबर बोल. ओके, and triglyceride target should be less than 150. And remember to then reduce the saturated fat. ते बाहेर सगळं डेझर्ट्स वगैरे ठेवलेले असतील तिकडे जायचंच नाही, पाठच फिरवायची. रिमेंबर दॅट डेझर्ट्स आर नॉट केप्ट इन द बँक वे, असंच लक्षात, ते आपल्यासाठी नाहीच असं लक्षात ठेवा, तरच खाणार नाही. नाहीतर तिथला एक सुद्धा पदार्थ तुमच्या कामाचा नाही. लिमिट अल्कोहोल अँड इन्क्रीज द फायबर. ओके, ठीक आहे. मे बी whatever I said may be true after Monday for many of you. But okay. And the last one, of course, is the talk by Vinayak, and he spoke on functional constipation. Functional constipation याचा अर्थ काय आहे की गॅस्ट्रोएंटरोलॉजिस्टने क्रायटेरिया देऊन ठेवलेत कॉन्स्टिपेशनचे. तर त्यासाठी तुम्हाला एक शॉर्ट फॉर्म सांगतो. दर्द भरी कहाणी ऑफ खडी बडी हार्डी. म्हणजे काय लक्षात ठेवा, हार्डी नावाची मुलगी दर्द भरी बिकॉज इट इज पेनफुल वेन द चाइल्ड इज पासिंग मोशन. Khadi, that means they pass motions in standing position. Badi, that means they are big stools, okay? And hardy, that means they are hard stools. तर ह्या जर समजा क्रायटेरिया फुलफिल झाल्या तर त्याला फंक्शनल कॉन्स्टिपेशन म्हणतात. ओके, सो इट इज पेनफुल. दर्द भरी कहाणी ऑफ खडी बडी हार्डी. रिमेंबर दॅट. सो द मॅनेजमेंट व्हॉट ही हॅज इन्क्लुडेड इज बेसिकली इन 3 different Ds. वन इज द डाएट, गाजर, काकडी, हिरव्या भाज्या, जे काही फायबर ज्याच्यात आहे माहिती ते सांगा. फर्स्ट डाएट. Second is complementary to it is drug. Only drug therapy is not the answer. Drugs with diet. Drugs म्हणजे peg a day keeps doctor away. Peg म्हणजे तुमच्या डोक्यातील तो नाही. Peg म्हणजे polyethylene glycol. Once in a day at bedtime. Management includes disinfection first. पहिले 10 दिवस polyethylene glycol रात्री पाजा. 10व्या दिवशी व्यवस्थित सगळी मोशन वगैरे झाली की नंतर 5-6 महिने मेंटेनन्स थेरपी लागते. कारण ते जे हार्ड स्टूल्स, बिग स्टूल्स आहेत, बडी, त्याच्यामुळे सगळी लॅक्सिटी रेक्टमची गेलेली असते, ती परत यायला इट टेक्स टाइम फॉर ऑलमोस्ट 5 to 6 months. ओके, सो क्रॉनिक कॉन्स्टिपेशन नीड्स टू बी ट्रीटेड फॉर अ लॉंग टाइम सो दॅट द रेक्टल इलास्टिसिटी कम्स बॅक. सो दॅट इज सेकंड D. सो डाएट, ड्रग्स अँड डिसिप्लिन. Discipline means as the child becomes 2-year-old, you have to see that he goes for toilet training. त्याला पॉटीवर बसवा. तो बसलेला असताना त्याचं टीव्ही लावून अटेन्शन डिस्ट्रॅक्ट करू नका. तर त्याला तुमच्या बोलण्यात गुंतवा. त्याला गोष्टी सांगा की बुवा असं असं आहे वगैरे. गोष्टी कुठल्याही सांगा. आई असेल तर ती नवऱ्याच्या गोष्टी सांगू शकते, हा कसा वेड्यासारखा वागत होता लहानपणी वगैरे. तिथं त्या पोराला काही कळत नाही 2 वर्षाच्या, तो हा हा हू म्हणतो, ते आपला राग काढून घ्यायचा. एनिवे, तर रिमेंबर दॅट द चाइल्ड शुड बी मेड टू सिट ऑन अ पॉटी आफ्टर 2 years. फार लवकर टॉयलेट ट्रेनिंग देऊ नका. 2 years इज ओके. So that is discipline. Okay. Also see that they use Indian style, that is squatting posture, because the level of the knee joint, तुम्ही जेव्हा कमोड वर बसता तेव्हा level of the knee joint should be above the hip joint. Okay. Then only that force comes. आणि तो जो अँगल असतो रेक्टम आणि सिग्मॉइडचा तो सरळ होतो. तुम्ही जर खाली नी जॉईंट ठेवला आणि जर समजा हिप जॉईंटची लेवल वर आली, तो अँगल बरोबर राहत नाही, मोशन्स होत नाही. दॅट्स वाय फॉर अ चाइल्ड टू हॅव द नीज अबाव, त्याच्या पायाखाली स्टूल ठेवा. कीप अ स्टूल अंडर द feet if you are using a commode type of a toilet. Anyway, with all that, I must congratulate all the speakers. There is a lot to be said about each of these conditions, but they have really put up the information in nutshell, and I congratulate all the speakers again, and I must appreciate the idea of the organizers to have this capsule. I think there is one more capsule tomorrow, and you will learn more about some other conditions. Thank you.
Thank you. Thank you very much, sir, for summarizing all 8 talks. Now I request Dr. Ramakant Patil, sir, to felicitate Dr. Jog, sir. And I will request remaining 7 speakers to be on dais. Please wait, don't leave the hall. Dr. Hemant Joshi, sir, is going to guide, is going to give very useful tips for the pediatricians. All the speakers to be on stage, please. Dr. Kulkarni, please. Dr. Amol Murkute. Dr. Sagar Sonawne. Dr. Amol. Dr. Amol Pawar. Dr. Tripti. Last but not the least, our Hushar Tushar. Thank you all of you for your excellent talk. We will have a group photo. All speakers with the moderator and Dr. Patil. Thank you. Thank you, all of you. Dr. Hemant, sir, please.
Slide. नमस्कार, मी तुम्हाला सगळे VIP इथे आहेत, सो आय एम हॅपी अबाउट इट. उद्या सगळ्यांना एक कॉफीचं पॅकेट मिळेल आणि एक कागद मिळेल. वी विल चेंज न्यूनॅटल मॉर्टॅलिटी विथ अ नेस्कॅफे पॅकेट. टेक इट टुमॉरो. आम्ही एक आरोग्य ज्ञानेश्वरी तुमच्या बॅगमध्ये टाकली आहे. त्यात सगळं मराठीमध्ये कसं बोलायचं पेशंटशी हे छान दिलं आहे. त्याने तुमचं कम्युनिकेशन वाढेल आणि प्रॅक्टिस वाढेल. IAP ने माझ्यासाठी काय केलं विचारतो, पण IAP ने आपलं भलं करावं म्हणून महाराष्ट्राने IAP वर 100 वर्ष राज्य केलं पाहिजे. शिवाजी महाराजांनी शून्यातनं साम्राज्य उभं केलं आणि मग मराठ्यांनी 100 वर्ष देशावर राज्य केलं. तर आपण तसं करूया. मराठे एकजूटने इंग्रजांशी लढले असते तर जिंकले असते. आपण पण असंच करतो, आपण एकजूट करूया. म्हणून आपण 100% कलिग्सना IAP मेंबर करू, 100% मतदान करू आणि आपण व आपला माणूस IAPचा प्रमुख करू. आणि फेल्युअर इज नॉट बट लो एम इज क्राईम हे सांगू. आम्हाला इच्छाच नसते, मी नाही होणार IAP प्रेसिडेंट, मी नाही होणार सेक्रेटरी, मला ट्रेझरर नाही व्हायचं, आमचे नाही सुरू होते. सो लेट्स गो टू यस मोड. आणि महाराष्ट्रामध्ये निस्वार्थ काम करणारे खूप आहेत. तर आपण एवढं एक निस्वार्थ काम IAP साठी करू, मग IAP आपल्याला खूप डिव्हिडंड्स देईल. दुसरं, NFHS 5 आता राष्ट्रीय परिवार सर्वेक्षण झालं, त्याचा अहवाल वाचा, तो खूप महत्वाचा आहे. तो सांगतो की आपण सगळे आहेत तरीही 10 पैकी 9 मुलं दुसऱ्याच वाढदिवसाला मालनरिश असतात, इरिव्हर्सबली. ते 6व्या महिन्यालाच असतात ऍक्च्युअली, आणि आपण ते बदलायला पाहिजे. डॉक्टर म्हणतात आपल्या बाळाला HI ग्रेड 3 आहे. बाबा आईला सांगतो, डॉक्टर म्हणतात एकदम मस्त बाळ आहे. सो वी फेल इन कम्युनिकेशन अँड वी फेल टू अंडरस्टँड की वी फेल. तर रिसेप्शनिस्टने पुस्तक द्यायला पाहिजे की डॉक्टरचं बोलणं समजायला ही डिक्शनरी वापरा म्हणून. आपण कन्सल्टंट सल्ला देतो, त्याचे पैसे घेतो, पण किती टक्के लोकांना कळतं आपल्याला माहित नाही. असा विचार करा की 10 गोष्टी सांगून 100 रुपये घेतले, 10च्या 10 गोष्टी कळल्या तरच 100 मिळाले पाहिजे, आणि 5 कळल्या तर 50 मिळाले पाहिजे. आपण एका रात्रीत रस्त्यावर येऊ. प्रॉब्लेम काय आहे की KG to PG इंग्रजीत शिकल्यामुळे आपल्याला मराठीत समजवायला जड होतं. आपण मेडिकल पण इंग्लिशमध्ये शिकलो, त्यामुळे नकळत इंग्रजी बोलतो. म्हणून बाल आरोग्य मराठीत वाचायला पाहिजे. त्यासाठी आम्ही तुम्हाला आता एक आरोग्य ज्ञानेश्वरी दिली आहे. त्याच्या मागे QR कोड आहे. त्याच्यामध्ये WHO टेक्स्टबुक ऑफ पीडियाट्रिक्स मराठीमध्ये आहे, त्याचं QR कोड आहे. ते एकदा प्लीज वाचा. स्टाफला शिकवायला खूप सोपं आहे आणि मोफत डाउनलोड आहे. सो डाउनलोड करा आणि शेअर करा. WHO पुस्तकाचे 303 मिनिटाचे व्हिडिओज पण आहेत ग्लोबल स्टँडर्ड केअरचे. त्यांनी स्टाफ खूप हुशार होईल आणि तुम्ही पण चांगलं बोलू शकाल. आणि कम्युनिकेशननीच 90% प्रॅक्टिस आहे, ती चांगली होईल आणि आनंद होईल. तुम्ही सोप्या मराठीत बोलाल. करून बघा, आई बाबा पाया पडतील तुमच्या. आम्ही प्रत्येक मेडिकल कॉलेजला 11 पुस्तकं कुरिअर केली आहेत. तर मेडिकल स्टुडंट्स पण सोप्या मराठीमध्ये डॉक्टर म्हणतील आणि छान होतील. हे सगळ्यांशी शेअर करा. आता आम्ही मेहनत करून खूप औषधावर मराठीत नाव आणले आहेत. सगळे ब्रँड मराठीत लिहिता येतात. लिहा बरं मराठीमध्ये. आमच्या शेपट्या वाकड्या झाल्यात. इंग्लिश गार्बेज इन इंग्लिश गार्बेज आउट. आणि मराठी इंग्लिश लिहायला फास्ट आहे, सवय झाली. कठीण आहे सवय बदलायला, पण बदला. एक्सरे चांगला आहे असं मराठीत लिहायला किती अक्कल लागते. तर रिपोर्ट मराठीमध्ये पाहिजे. जो 4000 रुपये देतो MRIचे, त्याला MRIचा रिपोर्ट नाही कळत. हे ट्रॅजेडी आहे देशाची, ही आपणच बदलायला पाहिजे. रिपोर्ट्स मराठीत यायला पाहिजे. मराठी ही महाआयपीची अधिकृत भाषा आहे, पण आम्हा कोणाला माहित नाही आहे. मीच तो ठराव मांडला होता. एक मराठीची शपथ आहे, माझ्यासोबत घ्या बरं जरा. मराठी असे आमुची माय बोली, जरी आज ती वैद्यकीय भाषा नसे. इचे पुत्र आम्ही इचे पांग फेडू, जगन्मान्यता ही सारपू प्रतापे. आपण करूया. नाशिक चांगली जागा आहे. मी भोसला मिलिटरी स्कूलला शिकलो. मला एवढा अभिमान, कौतुक आणि आनंद आहे. जय हिंद.
Thank you. Thank you very much, sir, for the thought-provoking message. I will request Kasliwal sir to felicitate Hemant Joshi sir. Thank you. Thank you very much, sir.
Thank you for being there.